Hello? Hello, can you hear me?
Hello.
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 Fine. Nice to meet you.
Nice to meet you too.
Thank you.
Can you hear me?
Yes.
Okay, thank you.
All is good, I guess.
Mm.
Okay, great. Hello.
Hello, hi Liana.
Hi.
Hi.
Nice to meet you.
Same here as well. Hi Mugita.
Yes, hello. How are you?
I am fine. How are you?
Dubla. I'm fine, Dubla. How are you?
Yeah.
Good.
Nice to meet you here.
Yeah, nice to meet you too.
I can see Dr. Rohit Roshan as well. Hi, Dr. Roshan.
Yes.
Hi, Dr. Raghunath.
Hello.
Hi.
Okay. It seems everything is okay. Yeah. Okay. Hope everything will go smoothly. Okay.
So
Yeah, sorry. Hello. Yeah, I just made a mistake. Just saying. Sit.
Okay. Hello, everyone. Nice to meet all of you, and I'm honored to welcome you and to say good luck to all of us, good luck to speakers. I hope very much that today's event will be useful for all of us, and all of us will gain knowledge, will gain skills to be used in our everyday pharmacovigilance practice. So today's theme is review of current pharmacovigilance system, processes, and progress. So I'm again welcoming you and saying, let's start. Just to introduce myself, I am Eliana Gaisian. I'm hosting today's webinar. I'm a head of Medical Affairs and Pharmacovigilance Department at NOIMED Contract Research Organization. I am Global Pharmacovigilance Society Ambassador for Armenia. I am member of International Society of Pharmacovigilance, ISOP. I'm International Editorial Board member for the Journal of Pharmacovigilance and Drug Research, GPADR, board member of Armenian Association of Specialists in Evidence-Based Medicine, and co-founder of Do Not Harm Health NGO. So saying again, welcome all of you, and let's start our today's first session. I would ask Chinmaya to change slide and to introduce the next. Okay, so here you see Dr. Chinmaya Mahapatra. Actually, we together organized today's webinar, and I'm very thankful to Dr. Chinmaya for all organization stuff he did for doing all this for today's webinar to go smoothly. So Dr. Chinmaya Mahapatra in master degree, completed his PhD from Bahampur University. He's having 10 years of experience in field of pharmaceuticals and pharmacovigilance. He had started his career as quality assurance officer in Psur Drugs, Mumbai, and moved to IPCA Laboratories Ltd. With an experience of 4 years, he started to pursue his PhD with TST-inspired fellowship. Currently, he is working as Pharmacovigilance Associate for PVPI IPC Chazabad, Ministry of Health and Family Welfare of India since 2017, being posted at Ministry of Health and being posted at MKCG Medical College and Hospitals, Berhampur. He has published a book, Fundamental Concepts of Pharmacy, its application, and 5 research articles to his credit. He is certified member of Association of Clinical Research Professionals, USA. He had accomplished his post-graduation diploma in clinical research from Bangalore in 2009. Since last 2 years, he is leading the International Peer-Reviewed Journal of Pharmacovigilance and Drug Research, GPADR, as editor-in-chief, and he is also a founder president of Global Pharmacovigilance Society. Actually, we organized today's webinar together. I mean, Armenia and India and this Global Pharmacovigilance Society headed by Chinma is one of the organizers of today's webinar. So thank you very much, Chinma, for all your efforts for today's webinar. Thank you.
Thank you so much, Dr.
Okay. So the first session of today's webinar will be moderated by Dr. Maya Sharma, a Global Medical Director in Medicare, Modi Mundi Pharma in Signuta, India. So she's chairperson of Global Pharmacovigilance Society, medical doctor with 2 decades of diversified experience across healthcare spectrum, clinical, pharmaceutical, nutrition, medical devices, and brings an experience mixed of multinational and Indian measures, established businesses and startups, AstraZeneca, Abbott, Philips. Currently associated with Modimund Pharma, Win Medicare, Signuta as Global Medical Director. She is chairperson, Global Pharmacovigilance Society, GPVS. She advisory board member, Journal of Pharmacovigilance and Drug Research, GPADR. She is executive board member, Accreditation Council for Medical Affairs, ACMEA Asia. She is Patron Science Advisor Society for Prevention of Cancer, SPOC, and founder of Nutrigenomics Club of India. So let's welcome Dr. Maya Sharma, and I will pass the floor to her to start to moderate the first session.
Hi, thank you so much, Liana. I mean, it is a delight to be here again with all of you, where we have the who's who of pharmacovigilance from around the world. So thank you so much for giving this opportunity. And without much ado, I'll start with the first speaker for today, which is Dr. Raghunath Dule. So can we have her introduction slide as well, please? Yeah, so Dr. Raghunath is a QPPV pharmacovigilance officer in charge at AbbVie, India. He has a bachelor's degree from Ayurvedic Medical Science from Rajiv Gandhi University of Health Science, Bangalore, and a post-graduation in clinical research from Bilcare Research Academy. He is a Six Sigma and ACRP, which is Association of Clinical Research Professionals certified, with more than 13 years of experience from Allergan or AbbVie India, ClinChoice India, Seniors Health, George Clinical, and IQVIA. So currently working as Senior Pharmacovigilance Manager with Allergan India, is responsible for the operations of pharmacovigilance in India, Sri Lanka, Nepal, Bangladesh, and he was backup also for other Southeast Asian countries. Not just pharmacovigilance, he comes with a rich experience in clinical trials, regulatory compliance, and pharmacovigilance. And he has been a peer reviewer for multiple articles for Journal of Pharmacovigilance and Drug Research. So I invite the first speaker to talk about pharmacovigilance regulations for marketing authorization holders. When we talk about regulations, it is said that there is nothing magic about regulations. Too much is bad, too little is bad. And good regulation should be conducive to the business and provide customer protection. So with that, I welcome Dr. Raghunath Dhule for his first deliberation.
Thank you so much, Dr. Maya, for the beautiful introduction about me. Hi, everyone. Good morning, good afternoon, good evening to all. As Dr. Maya mentioned, my name is Dr. Raghunath Dhule, and today I'm going to speak about regulations for pharmacovigilance requirement for regulatory requirement for the pharmacovigilance for the marketing authorization holders. I hope you all will take the opportunity to learn, to gain the knowledge about this, and you can implement. And moreover, the presentation which I'm going to present today, that is in general, that is not specific or relevant to the any country regulations or guidance. So it is in overall in general. Let me share my screen to you all. uh, can anyone confirm that my screen is visible?
Yes, it is on. Okay.
Yes, it is.
Thank you, Dr. Chinmaya. So current topic, as I mentioned, that pharmacovigilance regulation for marketing authorization holder. So disclaimer, this presentation is only intended for education purpose only and do not replace independent professional judgment and statement, fact and opinion expressed or those of the participant individual, and unless expressly stated to the contrary or not the opinion of our current organization or its co-sponsors or the committee. My current organization does not endorse or approve or assume the responsibility for the content, accuracy, completeness of the informed presentations. So let me start. Why PB guidance or regulations for MAH? Just a second. Let me minimize this. Yeah. So the importance of PB guidance or regulations to smooth functioning of pharmacovigilance activities in the pharmaceutical industries, and many countries regulators, they are emphasizing or having a guidance for running a smooth pharmacovigilance or activities in their organizations, specifically for marketing authorization holders for their pharmaceutical products. The guidance document also provides assistance to the marketing authorization holder on establishing and ensuring effective pharmacology system at their site. Uh, I, I can hear the disturbance. Can you mute all, Liana or Dr. Chinmay?
Everything is fine. Go ahead.
Please, please, everybody mute your microphones, please.
Okay. Yeah. The focus of the guidance and regulation is to ensure identification of risk associated with the pharmaceutical products and establishment of an ideal pharmacovigilance systems to mitigate the risk. So these are the key requirements for the marketing authorization holder. What you can see here, the first thing is pharmacovigilance system master file. Second is a preparation and submission of PSUR, audit and inspections of PV, collection, processing and reporting of individual case report form, and quality management as well as submission of risk management plan. This is the minimum requirement for each and every marketing or authorization holder to have these criteria or these process or system is in place. But this is not limited to, it's depend upon country specific regulations. They could have more, but these are the mandatory, I can say, compulsory requirement for the all the marketing authorization holder who marketed their product in the in their respective countries. So let's see about pharmacovigilance master file. So what it covers. The first point I would like to speak here is that QPP or pharmacovigilance officer in charge. See, it's again depend upon the individual country guidance or regulations. QPPV person must be having experience in pharmacovigilance, must be more than a 10-year, or maybe it's depend upon the country regulations. And QPP person also should have complete exposure. He will be the point of contact to the various distributor, manufacturer, partners, and also for the regulators as well. The second point is PV organization structure. Yes, it is a mandatory to have PV organization structure, how the hierarchy is, how many member we have, who is taking care. So this is how pharmacovigilance structures that is required, that should have each marketing authorization holder. When I go to the next one, marketing authorization and CRO point, see, pharmacovigilance master file should mention about roles and responsibility of marketing authorization holder, what they are gonna do, what their responsibilities, and certain marketing authorization holder, they outsource the work to the CROs. So in the pharmacovigilance master file also should cover the roles and responsibility of the CROs, including their point of contact, their responsibilities, that also need to be covered. So when I talk about sources of data, see, every company must have contact us a wording on their company website. So that is one of the sources of data where all the people can report. Also the medical inquiries system also is in a place, so people can generate their query to the medical department, so that they analyze. So there could be a chances of having adverse event. And apart from that, there are other sources like a digital medias. If the company is having a digital media page, then there could be a chances of reporting of adverse event on a social media as well. So there are multiple sources of sources for the receiving of adverse event. So company or marketing authorization holder should utilize that as well, and they need to be process the case, report to the regulatory authority as per their country regulations and guidance. So when I talk about the process, that is ICSR collection and SOP, yes, each and every marketing authorization holder should have a process system is in place for the collection of adverse event, as like I have mentioned just now, from the from the various sources of data, and also should have a SOP is in place with clear roles and responsibilities, who is going to do what kind of product specific conversion case processing. So all these things that need to be covered in the PV processes. And when I talk about composite system and database, this is one of the important, the what kind of system, what kind of database that we are going to use for the PV system. That is also, one of the important criteria. There are multiple, database in the market. You can, you might have heard about the Scepter, Argus, Eritrea. There are so many, multiple database. That is also need to be, explain which version that we are using, composite system database for the PV activities at marketing authorization holder. So when I talk about quality management system, yes, it is must and should, important, and all marketing authorization should have in place how you are documenting your PV document, what is the archival period. So it's depend upon again, company SOPs and record control as well. Yes, of course, training is a must and should, that should be is in place. So it's a responsibility of all marketing authorization holder to provide the training to all their company employees, not only the company employees, even the contractor, which include, manufacturers, distributor, patient support program, as well as even, marketing research, who will be the vendor for the marketing authorization holder. So that training process and system is in place. So whoever, managing, marketing authorization holder product, they should aware about marketing authorization holder product, and they should report adverse event as and when they come to know within 24-hour timeline. Again, the timeline is depend upon, country specific guidance and regulations. So also marketing authorization holder should have a auditing system is in place. So there could be chance that, audit there could be internal audit or external audit. So by maintaining all these quality system is in place, so marketing authorization holder should be ready at any point of time for the auditing, as well as I can say for inspections. So by combining all these, QPP or PV organization structure, roles and responsibilities, sources of data, this is how we can, keep up to date our database, or I can say a pharmacovigilance system master file, so that anyone can come and audit. If you have everything, clear structure, so it would be easy for the auditor as well, and also for the inspectors. So when I talk about the annexures, so this is one of the, important part of PSMF. So annexure should cover, list of products. So marketing authorization, whoever, marketing the product, they should have attach list of product to the pharmacovigilance master file, and also the contract agreement. Once again, when I say about the contract agreement, so contract would be with the distributors, manufacturers, and others with, PSP vendors, or a market for the market research and audit, as like just I have mentioned, and the task delegation. So it's again one of the important criteria. As a marketing authorization holder, you might have outsourced a certain task to the CROs. Say for example, literature screening, you might have outsourced this activity to the, one of the CRO. So what their roles and responsibility, that that list also need to be attached along with which company is handling. So all these things that need to be attached as an annexure. And apart from that, yes, one more thing, audit reports as well. So how many audit as of now undergone by the marketing authorization holder, and what will be the CAPA action items and upcoming audit. All those things need to be attached as an annexure to the pharmacovigilance master file. So I have explained about this is how it looks like, pharmacovigilance master file. Again, this is not specific to any country specific or country specific regulation, but these are the things what I can say based on my experience. These are the mandatory requirement the pharmacovigilance master file should cover as a marketing authorization holder. So let me move to the next slide. Just a sorry. It's about collection and reporting of an ICSRs, collection, ICSR processing and reporting. The first thing will come into the picture, that is collection of ICSR. How you will collect? As like previously mentions, medical inquiries. So every department, every marketing authorization holder must have a medical inquiry department who managing the queries from the in general whatever they receive. So there could be a chances that as a consumer they write an query to the medical department mentioning that for the so and so product I am facing so and so kind of issues. So there could be involvement of product, there could not be involvement of product, or there could be a just a kind of information asking like how can I use this particular product. So medical inquiry department, if they come to know that if any adverse event reported, then it's their responsibility to pass those adverse event to the pharmacovigilance department. So pharmacovigilance department can process that case, can do the follow-up with the consumers. So this is how relationship between the medical inquiry department as well as the pharmacovigilance department. As like I have mentioned, contact us email. This is contact us email, something that is should be present on the company website, where the general public or even a kind of doctors, who anybody can report an adverse event. And this contact email ID should also be screened on a daily basis. If anything comes reported adverse event, that also need to be transferred to the pharmacovigilance department, so that further they can take the action as per the SOPs. And marketing authorization employees, here there could be chances that even marketing authorization employee can also report an adverse event. For example, the medical representatives, the medical representative job is to daily visit to the doctors. So at certain point of time, even doctor may mention that they have faced an adverse event for so and so product. So it's the responsibility of even marketing authorization holder to report an adverse event. And contract partners, as like mentioned, distributor, manufacturers, PSP vendors, also they also need to be report, or they also responsible for providing adverse event to the pharmacovigilance department of marketing authorization holder. And again, digital media, certain companies have a digital media page, maybe a Facebook or Instagram, so there could be a chances of getting of adverse event there as well. And also solicited report, literature screening and follow-ups, that is also need to be follow by the pharmacovigilance department. This is how, these are the criteria, or these are the I can see sources for the collection of ICSR. And once we receive the ICSR at a pharmacovigilance department, what we do, we process the case as per the product specific conventions and as per the SOPs. And also it is important to do a follow for the pharmacovigilance person who is processing the case, to get the more information as early as possible. The basic element, as you all know, that patient detail, drug detail, event detail, and reported detail. These are the four basic, requirement. And apart from that, it would be always, I would like to urge all the pharmacovigilance associate, they can follow for the for the information like batch number or a lot number and date when the the reporter has come to know about an adverse event. These are the specific follow-up that is required to do, so it would be helpful for the further assessment of the case. So next step will come to the reporting part. Yes, reporting part, it's again it's depend upon as per the country specific rules and regulations. Certain company certain country regulators require CONs forms, and certain countries regulation speaks about the XML form. So let me move to the next slide. So preparation of a PSUR. this is also again, again in general, it is not specific to the any country rules or regulations. So these are the certain basic things that required to, required for preparation of a PSR and also for the submission of a PSR. It says about the title page, introductions, and current worldwide marketing authorization status as well. So if I tell about current worldwide marketing authorizations status, so it's a responsibility of the person who will be the taking care of PSR. They need to provide the detailed information about, is there any clinical trial conducting for that particular product, or is there any clinical trial has been stopped, or I can say, is there any there could be chances that regulator may stop marketing the product. All those things that need to be covered in the current worldwide marketing authorization status. And also update the action on the action for the safety reason. Is there any action has been taken for that particular product, that also need to be updated. And changes to the reference safety information like PIL or a CCDS, I can say, okay, this is company core or data sheet or product information leaflet. And also it should cover presentation of an individual case history. And apart from that, estimated patient exposure means how many number of patient got exposed, how many patient has a similar kind of adverse event. And apart from that, if I say about studies, a study is nothing but, again, it's talk about the clinical trial and post-marketing studies and other information like lack of efficacy, overview of a signal, and conclusions. So let me move to the next step. Just a second. Sorry about, just a second. Sorry, this is about quality management by the by the marketing authorization holders. So as like just now I mentioned about pharmacovigilance system master file. Apart from that, also marketing authorization holder also should have, quality cycle of a PV system, quality objective of the pharmacovigilance system, responsibility for the quality system within the organizations, training to the marketing authorization personals, facility and equipment for the pharmacovigilance, PV compliance management, and monitoring effectiveness of a PV systems. So this is all about talks about a quality system that should be managed by the marketing authorization holder. Yeah. Well, coming to the next steps, audits and inspection. This is the most important what I can say. Marketing authorization holder must be ready for the audit and inspections and ensure that the PV system is in place, ensuring that the appropriateness and effectiveness of the implementation and operation of the PV system, including quality system for the PV activities. Ensure that marketing authorization holder should have a qualified personnel, robust system, and facility to conduct the PV audits and inspections. So this is a big topic what I can say, but just I have covered this is one of the requirement for the marketing authorization holder. Let me move to the next slides. Submission of a risk management. There are several ways to categorize an effective risk management process, and marketing authorization holder should have again this process is in place. So here, when I speak about submission of a risk management, the activities involved like identification of a risk, measurement of a risk, examining the solution, and implementation of a solution, and monitor the results. So this is how goes this workflow go as in submission of a risk management. So you'll be this is again a vast topic, and yeah, you'll be in order to know more about that, you need to be go through into the details. So just I have covered the basic requirement for the marketing authorization holder from pharmacovigilance perspective. This is all about me. If you have any queries or concerns, you can contact to our organizer and we'll get back to you. Thank you.
Do we have any questions for the speaker?
I will have a question. May
Please.
I?
Yes.
Please.
Okay. Thank you very much, Raghu. Really very, very informative overview of pharmacovigilance system and QPPV work. What I would ask is, so I have several questions. I will try to organize my questions in a best way to be clear. So when you say qualified person for pharmacovigilance QPPV, my question is about location of this person in the European Union or in Eurasian Union. Should this person be located in the country where the drugs are marketed, or it doesn't matter, or what is the rule for this? And there are local pharmacovigilance person and QPPV qualified pharmacovigilance person. I would just ask you to describe a difference, and also from the point of view residing in the country or not. This is the first question. And the second question is about communication between marketing authorization holder and healthcare professionals, HCPs, for gathering information about case reports. This is, I know this is an important part in pharmacovigilance to get an information about case reports, because in overall, all over the world, this is a problem to get case reports, information from healthcare professionals, from patients. So what is the general rule for marketing authorization holder, whether they obliged to have trainings or speak with HCPs, patients, to promote and to get this information about case reports? Thank you.
Yeah, thank you, Liana. I'll answer to your second question firstly. See, marketing authorization holder should try as much as possible to collect the adverse event through the various process, as like, training is the one of the important factor. If you look at, I can see there is no robust mechanism is in place currently in all over the world, and many people, they are hesitant to report an adverse event. So in that situations, the first point of contact is medical representative. So marketing authorization holder can train to the medical representative in such a way that as and when, they can encourage to the doctor to report an adverse event. One thing I would like to tell you that, in order to encourage reporting an adverse event, marketing authorization holder can place a system is in place, like say for example a kind of visiting card which can be easy to carry in their pocket, and that visiting card a size of visiting card should have process for collection of adverse. They can collect the basic information for reporting an adverse event, like patient detail, drug detail, event detail, and reporter detail. So it would be easy for them to carry, and as and when they come across about an adverse event, they can immediately report to the pharmacovigilance department. I would say this is one of the major thing that we can play. As a marketing authorization holder, anyone can place this activity so that we'll be getting a more adverse event. So this is the one of the thing. And apart from that, I would like to say, as like I have mentioned in my presentation, it's about there are some companies having a digital media pages like Facebook and also on Instagram. So people used to report an adverse event there only. There should be someone, person from a medical marketing authorization holder perspective. They can look into those pages and they can immediately respond, or they can immediately collect the required informations for, which is required for a, processing of an adverse event. This is how I feel where we can get the more adverse event, and we can encourage the people for reporting adverse event. And also marketing authorization, in fact, they save their product, they can assess their product safety profile. So this is how it helps. I hope I answered to your second question.
Yes, thank you. I'm just thinking that actually there is no regulatory obligations for these processes. Yeah, so it's like just up to marketing authorization holder, like to gather this information, but they are not obliged to regulatory agencies for these processes. Yeah.
You're right, Liana. It is more of voluntary in nature.
Right.
But I think, as Dr. Raghu has described, that if we can create a system in which we are not just providing training to the medical representatives, but also in a way when on a visiting card or your introduction card, if some detail about that, if the SCP encounter any kind of adverse drug reaction, which is the point of contact they should reach out to. If it can be mentioned on the visiting card, I think it would help even build the credibility of the organization better, and doctor would know how to report, whom to report, right? I think on this, Dr. Mulugeta has something to say. So Dr. Mulugeta Russo, maybe you can put on your video and unmute yourself, please.
Okay, thank you, Maya.
Yeah.
Thank you, Dr. Dehle. It was really very insightful presentation. I have one or two questions for you. The first one is some people argued that PV might not be required for market authorization holders that produce, especially for the generic manufacturers. I would like to have your impression on this. And the other is, what's really your experience in other countries, maybe from India and other countries, or maybe anybody else who is in this platform might comment on it. How is the participation of the generic manufacturers in identifying or in monitoring safety of the products they produce? And what platforms they usually use? Is it online platforms, or do they have paper-based platforms? That's what I want to know. Experience of other countries. Thank you.
Thank you so much, Muruge, for your questions. Yes, I'm happy to answer your questions. See, as like I mentioned, not only the marketing authorization holder, this again rules and responsibility also applicable for even distributors and as well even for manufacturers as well. It's their responsibility as well. It's depend upon the country regulations, country guidance. In the guidance they might have mentioned that even a distributor or even a manufacturer, they also should supposed to be report in adverse events. It's depend upon a country's regulation, I would strongly say. If guidance is not clear or regulation is not clear, nobody is bothered about reporting adverse event. So in order to again improve the culture of notification of adverse event, these things need to be is in place. So it's depend upon again generic company depend upon generic companies how they want to be involved in the PV activities. Yes, I would like to say moving forward, not now, but I have seen from my past era, in my past experience, regulator become more and more stringent for bringing new rules and regulations, not only for the marketing authorization but also for the manufacturers as well. So I hope moving forward, even PV system also been built in the generic companies as well, and this is one of the important, I would say, for patient safety perspective, I would like to say. Yeah.
Yeah, I agree to that because it is mostly country specific. So there are health ministries and country specific drug controller generals who bring out these kind of regulations. And if I would say as responsible citizens, we worry about this and we want, something new to bring about for better patient safety. I think we should participate, with the governments and regulatory bodies to come up with some kind of a regulation to make sure that even generic companies are regulated, so they have a system and process in place to notify, the regulatory bodies about adverse drug reaction, and a system, to make the people aware about it. And, that would do good to everyone. So I think the citizens should actually participate if the existing system is not in place.
I agree. Yeah.
Dr. Amir had something to say. Dr. Amir, maybe you can unmute yourself.
Sure. Thank you, Dr. Maya. I appreciate that. First of all, thank you, Dr. Raghunath. It was a great presentation. I appreciate that. I have a quick question. In terms of BLA submission for FDA, I know it's not like for Europe. Can you just give a quick differentiation with, MAH for EU versus FDA? Like what are the core requirements for the PV for BLA submissions? And the follow-up question is that also, if a company is outsourcing the PV activities, like, case processing and everything, that company, when they write SOP, do they also include those outsourcing activities as part of a core SOP, or you have to just have an extra addendum to that? Thank you in advance.
Thank you very much, Amar. So again, I would like to say here is that with regards to submission of this PSUR and all, so country regulator, they might have guidance document, so they have mentioned content of the requirement of a submission of a PSUR. So the respective countries need to be follow as per their process. If I say about India, there are certain specific content that need to be covered into the PSUR submission, so that need to be followed. So the
I said BLA, Biologic License Application, BLA.
Sorry, I do not have an idea about it. Can I get back to you later, Amar?
Sure. Okay, no problem. Thanks.
Thank you.
And
So much.
regarding the SOP, if you can answer that, if the company outsource the PV activities and has a part of submission of the document, if they have to write SOPs, does they need to include all those the CRO responsibilities in the core SOP or not?
It would be always, usually it is covered into the SAW, that is, in the agreement or kind of agreement. What will be managing by the CROs and what will be the managing by the marketing authorization. So that covers the roles and responsibility of CROs and also marketing authorization. I would say it would not be included into the SOP, but it is in the agreement.
So will that be sufficient in terms of SOP? So that's like a safety management plan, right? SMP. That's what you're describing. So will that be sufficient? Sorry.
Yeah. If it is a safety management plan, if safety activity is going to take care by the CROs, then it's their responsibility to follow, that SOP. But the safety part that should be covered into the agreement, I would say. That is the agreement which is between the CROs and between CROs and MAH.
Okay, thank you.
Thank you.
All right. Thank you so much, Dr. Raghu, for the wonderful presentation and for clarifying the doubts of our audience. With that, I think we will move on to our next presenter. So can we have the introduction slide, please, for Dr. Luca Cariolato? Yeah, so it's a pleasure to introduce Dr. Luca, who's the head of Signal Management, Vifor Pharma, Switzerland. Dr. Luca obtained his master's in pharmaceutical biotechnology in 2004 and a PhD in life sciences in 2011. After a postdoctoral in cancer immunology, he started his career in pharmacovigilance as a consultant. And for more than 8 years, he was involved in medical writing and in particular in signal assessment reports, aggregate reports, benefit risk assessments, and updates of product information. Currently, Dr. Luca is the head of signal management at Vifor, and his main goal is to build a robust safety surveillance process that ensures the safety of the patients and the evaluation of the benefit risk during the entire life cycle of the drugs. So I invite Dr. Luca to share his wealth of knowledge on signal detection and management, suggestions to have a more efficient process. Dr. Luca, kindly unmute yourself and put on your video, please.
Hello, can you hear me?
Yes, absolutely.
Can you see me?
We can see you, and we would also like to see your slides.
Thanks. Just I wanted to be sure that my camera is working.
Yes,
I will share my slide. So where should I share? Just a second. Voila. Tell me if you can see my screen.
Yes, we can see.
I will put in the presentation mode. Okay. So, good morning, good afternoon, everybody. So today I'm going to present a quick presentation about how we can increase our efficiency in signaling. Signaling is, I would say, the beating heart of pharmacovigilance. So we are collecting data to identify any new safety information. However, from the single case report, we don't know exactly if we have a new data. Often we need to analyze the data in aggregate manner and try to understand if there is an increased trend, if there is a cluster of events that can be explained by a similar way to cause the event, for example, a risk factor, concomitant administration of a drug, so drug-drug interaction, etc. This is a critical process, unfortunately, because there is not one correct method, I would say. And based on my experience, sorry, mute yourself, please. Can you please mute all your, thanks. So there is not a correct method, there are different methods and based on the nature of our database and our drug. So this is just an explanation of how we can improve the process based on my experience. For this reason, this is just for all of you, is all the opinions and idea that will be present in the PowerPoint slide reflect my idea, my thought, and not the company. So what is a signal? A signal often per definition is a new safety information. I take the two most important definition from EMA WHO. EMA say that a signal is an information from one or multiple sources, including observation and experiment, which suggest a new potential association or a new aspect of a known association. Similarly, the WHO said that a signal is a reporting information on a possible causal relationship between an event and a drug administration, the relation being unknown or incompletely documented previously. So a signal, as described by EMA WHO, is a new safety information that suggests a potential link between the drug use and the event. Of course, this interpretation, sometimes there is two ways. There is a pragmatic interpretation and sometimes a conservative interpretation. For this reason, an exercise very nice, if you give 10 cases or 10 events to two different persons that work in PV, they will find potentially different signals. Because of course, depends of the sensibility and sensitivity about the person who reviewed the data, they can judge an information as new or not. So again, also here, there is not a standardization, even if it should be clear what is a new safety information. Typically, when we are reviewing data, and we know that in our daily activities, we review 100 of cases every day, every week, every month, depending on the volume of the database. What is a new signal? Typically, when we have a new adverse event that suggests a new potential risk that was not known, important is also the drug interaction. Often drug interaction is not coded when we process our cases, but can be clear that the two drugs can cause an event when they are taken together, or can worsening also some other events. Another important thing here is that the signal can be also medication error. Our label should give a clear explanation how we should take the drug to have a correct and the desired benefit. Of course, when we have some issues like medication error that lead to important events, probably is because the medication error is caused by a wrong information in our label, and we should correct. Lack of efficacy is another important signal that we can identify if in particular there is a cluster of cases that suggest lack of efficacy. Lack of efficacy, in a way, there is a threshold that is accepted. Nobody taking a drug have the same effect. This is quite clear and known, but of course we should identify this threshold of lack of efficacy, and when it's not acceptable or when there is something that is unexplained, also we should understand why, if there is a reason behind the lack of efficacy, and act to minimize the risk. Off-label use is very important. We know that for during the pandemic, there were a lot of off-label use of the drug that in a way were accepted due to the emergency, but often an off-label use, when it's not justified, can be not accepted and increase the risk of the health of the patient that takes the drug. And finally, we have product quality. Also, product quality, even if they don't directly suggest a signal, are important to continue to monitor. And finally, I put just side effect. Side effect is any effect, not even an adverse event, something that also can be unexpected benefit. But also on this side, we should monitor the positive effect, because again, this could lead to in future off-label use that is not what we want, and we are not able to monitor and take the control. About the new information, sometimes when we have a risk, when something is listed in our label, we tend to not forget what to say, okay, is known the risk. True, but sometimes from the review of the data, from the review of the literature, we can identify a lot of new information, like a new potential mechanism that was not known, and help to characterize the risk and take some measure to minimize the risk. We can have also risk factor. We identify from a publication or reviewing the data there is a new risk factor. Again, also here, we should characterize this new risk factor and probably act to reduce the risk, the impact of the risk. And we can have a specific population that can be affected. So again, we can focus the risk based on the specific population and act to reduce the impact of the event and of the risk. Changing severity, this is very challenging, because often when we receive cases, they don't specify the severity. They specify the seriousness, but not the severity. But again, in case we can have this information, which is very useful, we can also act and as a new safety information that minimize the risk. Then we have the increased frequency. We know that an event is very rare. Then we start to see that become rare or uncommon, change the frequency, or different evidence for a new potential risk. Sometimes we have potential risks that are still not identified. We still need some evidence to confirm the risk. Every single piece of information could be important to collect, analyze, to identify if the risk can then become an identified risk. So these are just an introduction that I think you know very well. Just here is a different methodology when we do signal detection. As I said, we have data sources, line listing from our database, published literature, clinical data, data mining like Pharmacovigilance, EVIDAS, and FIRES from FDA. We can have pharmacoepidemiological data, and all these data must be reviewed in different methods. We have semi-quantitative method, for example, from line listings and review case series. Qualitative methods, review of ISSR or published literature. And as I said before, quantitative method from the database of health authorities. The most known is FIRES and EVIDAS, but there are also other BG-based, etc. All the analysis of these sources with the methodology should suggest if we have a signal or not. As I said, also product complaint, product quality must be reviewed, even if they don't identify a specific signal directly, but can suggest a potential signal. If we have a product quality issue, we can build hypotheses and then try to monitor if there will be an impact of this product quality issue. For example, we have a bad taste or something like that, so we can anticipate and identify if in the future analysis, this quality issue could have an impact on the signal. So these are all the methodology that are often explained, all the data source review. But what are the challenging now in industry? As I said before, the first challenging is the quality of the data. Unfortunately, we know that the information from the ISSR, in particular from spontaneous sources, are not very complete and accurate. So it's already difficult to sometimes understand if the information is relevant or not. Then we are also problem to access to follow up the information. We request follow-up queries, we don't receive sometimes answer. So still we remain with cases that are poorly documented or not well documented. I would say sometimes they are very well documented. They are documented, but the relevant information like the time to onset is not there, and then it's difficult for when we review the data to say if the drug caused the event. And then, of course, sometimes we have a lot of in publication, we have more and more publication, and the case report are excellent tool, but sometimes also the case report are not well documented. So they provide important information, but that can be misinterpreted because it's a publication for sure, but still there's a lot of evidence that are needed to identify a signal or not. The other challenging is the volume of data. We know the cases are increasing every year, and we start to have more and more cases in the future from different sources, solicited and unsolicited. For me, already to put in the same basket the two cases from the two sources, solicited and unsolicited, is not very accurate. Solicited data, unfortunately, they over-report events that the unsolicited they don't. They also increase the number of publications to screen, and again, we should have more resources. All the publications are important, but then we have to have a method to classify which publication is relevant and which is less relevant. I would say not non-relevant, but less relevant. And then we have also new sources to review regularly, like health authority website, PRAC meeting minutes, PRAC agenda, etc. That in a way also provide a lot of information that must digest. And again, then the appropriate strategy. When we go to review this data, we have different ways. We have a qualitative review. If we have few cases, but if we have a lot of cases in our database, we cannot continue to have a qualitative review. We should move to a more structured quantitative and semi-quantitative analysis. For this reason, we should be able to define threshold to minimize the noise, which is not very easy and not very sometimes applicable. We have, for example, what is important for me also to understand that sometimes we have initial events that should be summarized in summary tabulation. We have line listing that initial and follow-up cases should be reported. We have clinical event that should be considered to calculate the statistics, and exposure must be reflect the correct periods of the event. One example, often we try to make a reporting rate. We take the number of events collected during a month, and then we divide it by the product sold during that month. And this is not accurate, the period is too short, and of course the people that took the drug on January 2022 are not the patient that experienced the event in that month. Probably they will experience event in February, March, April. So sometimes it's also difficult to make this decision and to establish a correct process and accurate. Another thing here is that we know signaling cannot, even if in the regulation there is not really the major regulation, they don't define timelines. It's not like PSUR that you have to write in 70 days after that log point or 90 days after, depending how long the product is in the market. But still, we have to do it in a reasonable time manner. Cannot take the data of December 2021 and analyze in July 2022. It's too long, and there is a risk that the patient that we miss an event and the patient were at risk. So we should do this activity as soon as possible. So we should identify just a few months after the data log point to complete the analysis and take already some action. Also, this implies that also we need a lot of resources sometimes. We need a lot of people that work on signaling to do a good analysis in a time manner that is justified. And also the analysis. The analysis must be performed in a comprehensive manner. We should not just review the line listing. We should review line listing. We should review the literature, other data, and then try to understand if there is a signal. Otherwise, we can risk to take false positive or false negative for signals. And also, one important thing is that when we produce a report or an analysis, it's not just for that moment, but we should take back when is needed for audit inspection to try to explain why certain decisions were taken. So should be also used the data to discuss in future or to retrospectively understand why historically certain decisions were taken. So there are some suggestions that I would like to provide for these and that I took during my experience. For the qualitative manner to review cases, we should have always a qualitative review, but should be performed first on daily activities. We know that every day there is a medical approval of the cases that were processed. So really the physician that review these cases should be trained to identify signal, because they review cases every day. They can already identify if something comes from these cases that they review. And the other way is also to select some topics. If we have too many cases, already select some topics. For example, we should review every day all fatal cases, so we don't take too long to take a decision. All designated medical events should review on daily basis, or all the events that are under close monitoring. We know that for certain drugs, there is certain potential risk that can be caused. So we should have a methodology to extract every day these cases and review, because this can suggest a new signal, important signal, or on the contrary, we try to monitor if the activities that we put in place minimize the risk. On the other hand, we have also the quantitative analysis, and the quantitative analysis then should be done when we review a particular period. If we have a signal detection done every quarter, a signal detection report, it should be important to have a good quantitative analysis that stratify the cases to review. So, for example, to stratify events, serious, non-serious, listed, unlisted, to stratify the cases spontaneous versus unlisted versus litigation, which is also sometimes case that report a lot of information, and to identify a new risk that report also an increased frequency or a new population. So we should have a good approach when we arrive at the end of a period to identify the quantitative analysis. Another way is that to have this graph. So on daily review, we should, as I said, have a better, should be reviewed by the same person or his deputy. So the daily review of the cases is done always by the same eyes, I would say. We cannot have 10 persons to review on daily basis, because we can miss information or not to have the same approach when we do the daily review of the cases. When we have the review of a period, weekly, monthly, that is in a short period like this, we should review the relevant information, summary for the relevant information, or literature article or other information. We should have always an intermediate report between the daily and the period, something that we try to reconcile the daily activities and identify if there are important information to report. And then we should have the periodic detection. And as I said before, this should be done based on quantitative analysis, and keep always a slot for the qualitative review for events that come from this quantitative analysis or cases that are considered relevant and we cannot miss, like fatal, designated medical event, and close work event under close monitoring. So ideally, we should have a balanced approach to have continuous versus periodic review. The periodic review, like aggregate analysis that combine different sources and methods, versus continuous review, that is the daily review or the weekly review or summary. You should have a good balance, not too much quantitative and or not too much qualitative. Otherwise, we are not on the good, it's not well balanced. In addition, the tool and process should be flexible. Signal detection and management, we know sometimes we can have no signal for 1 year, and then in 1 month, 5 signals. So we should have a flexible, so to be ready to review any important information in a timely manner, and to communicate these with the other documents like RMP, PSUR, and of course should be also flexible. If we have 2 products in our company, one product that is very old, that the safety profile is well known, for this product, we can have a review every 6 months of the data, a periodic review. But at the same time, in our company, we have a new product that is launched in the market less than 1 year. For this product, we should have every month a periodic review, because there the information, the new information, of course, are more frequent. So the process should be also flexible. So we should not have just one period for review for all products. We should adapt to their safety profile. And as I said, signal detection is not in a silos activities. We should communicate with other important document. For example, PSUR, PBRER. So should have a direct link with these 2 documents. So the question here is why we are not already performing certain activities that are done for PSUR during our signaling activities. For example, we should already perform an analysis of medication error every period. We should already review the new information about the important identified risk, missing information, important potential risk. So then when we arrive at the PSUR, we can have a real good summary of the activity and surveillance done during the year, during the 2 years, during the 3 years. So we should always try to think that this data in a way will be used for an aggregate report. And not only, is also for an RMP. Signal detection activity and RMP should communicate regularly. There are a lot of activities in the RMP that tend to, of course, minimize the risk. We should monitor during our signaling activities if a risk is reported more or less. And also, more importantly, the activities that we review every time, we should understand if we should also reclassify the risk. Maybe is an important risk, and then we see that there are a lot of, based on the risk minimization activity that we have put in place, the impact of the risk is less, and we can also say that at the end of a long period, the risk was important. Now, based on the activity that we minimize the risk, is not anymore an important risk. It's still a risk, but not important. And finally, of course, there is the part of management. When we identify a risk, we have the 2 steps, the signal validation and the signal assessment. Also here, we can do a lot of things to increase the efficiency and the quality. From my point of view, when there is a signal validation, the signal validation is the process that should suggest us if we have sufficient data to support a signal that was detected, and to perform a complete assessment. So if we have a process, or even better, a tool, it should provide quickly an analysis of the number of the cases in the database, try to understand how many articles we have in the published literature, if there are other medical references, and also a prior assessment. So we should analyze this data before to say if a signal is validated or not, because otherwise we continue to validate signal, and then, of course, when we arrive to assessment, we should provide clear information also when we arrive in the assessment. And in the assessment is, of course, a process to evaluate the causal relationship. So a tool should not just establish if a drug can cause an event, but also should understand which are the risk factor and the contributing factor. And a tool, a process should select the relevant publication to support the assessment and any other relevant suggestive sources. So in conclusion, to take a home message, any tool or process can provide the relevant support if there is a robust signal detection and management process. An efficient process and tool must combine in a smart manner the quantitative and qualitative approaches, and any process and tool should be flexible and adapt easily to the safety profile of the product. So I like to thank you, and this is my mail in case you need some clarification, if you need some question.
Yeah, thank you so much, Dr. Luca, for this presentation, quite an enlightening one. I have a question. So when we talk about signal detection, which is typically done for the new product launches, when a new molecules comes to the market, are there pre-specified timelines, and whether these timelines for signal detection depend upon the molecule, the organization, or the country regulation, that for how long signal detection will be done for a specific molecule?
When is no, there is the point in the module 9, for example, that is the typical is like our main guidance for the signaling. There is not any timelines to perform signaling, but of course, it's an activity that is requested. And when there is any inspection, they will look and understand if it's appropriate the timeline. So sometimes the authority, even if it's not specified the timelines, can challenge the timelines. For example, if it's a quarter early review and is done in 3 months, this quarterly review, they can have questions, say, no, for us it's too long. So you should reduce the time to perform an analysis. So there is not an established timeline. It's not like PSUR to submit in 70 days or 90 days. It is not. But of course, it's very important and taken seriously by the inspector. It is not because it's not described that we can take years to perform an analysis. They want, or month. They want to do a good analysis, clear logic analysis in the shortest period.
And can you give some example of, the molecules and how the timelines for signal detection and the length of it varied?
As I said, for a product is new in the market, and we review again every month the data, should not be completed. Then the extraction of the data and the analysis of the data not more than 1 month after the data log point. So we should be sure that all the data that we analyzed are well done, and also that the analysis is robust, no more than 1 month from my point of view, in particular if the product is really new in the period. Also because we have to also keep in mind that the data, in particular at the beginning, it's not like an established product, but the volume of cases is start to arrive. Of course, the product is new, is not launched in all the countries, so still the volume can be manageable. So we should analyze quickly the data. For a product that is not an established brand, but is already 5 years in the market, I think that already we can analyze data every 3 months, and of course, no more than 2 months to analyze the data. But every three months to analyze the data is sufficient, because the product is 5 years in the market at least. So we have already 5 years of experience, and we also try to reach. We already reached a kind of known safety profile. Of course, we can only discover new things, but we start to have already an experience. Then for products that are more than 15 years in the market, 20 years, the products start to become an established brand. So again, depend on also the safety profile, but if the safety profile is well known and addressed, we can move also to 6 months to review the data.
Okay.
But again, this depends on the safety profile.
Sure, thank you so much. Dr. Ahmed had a question?
Yes, thank you, Dr. Maya. Dr. Lucas, it was a great presentation. Thank you again for that. I have a couple of questions and one comment. First one regarding the literature, as you mentioned that as a part of the challenges that there's a lot of articles coming in, and there is no way you can review a lot of articles, and you were thinking like how we should deal with it. But my point is that without looking at the articles, you cannot really say, right, which articles are important, which are not important. you need to review it, and a medical professional has to review that. Because signal can come from any articles, right? You cannot categorize high impact articles and low impact articles, right? What's your thought on that?
Yeah, as I said, we cannot really establish if a journal or an article is more important than another journal or article. So I think that we should be clear that we should review, of course, every article that suggests at least that there is a source, but it's more on the methodology to interpret the data, I would say. To bring to the signaling meeting for when we discuss signal detection. Sometimes we tend to be very conservative. What I'm asking you is always to challenge the article. There is a case report, is one case report. Try to always to understand if really is well documented the medical history before to bring to the attention. Otherwise, I so many times, in particular authority, that they flag articles that could be important. I'm not saying that are not, and they consider it a signal confirmed for them. While if you review the article, it's still one case report. There is not, for me, for a case report, because for an article, I start from the hypothesis that should be extremely well documented, a journal, if something is published in a journal. So be critical when you bring this to the attention of the signaling meeting or to the signaling team. Try to really challenge, is it the concomitant drug well detailed? The medical history is well described. Often when you have a case report, you should have also good diagnosis. Is it that the method that diagnosed the event well known, gold standard? So this is what I mean. Of course, we have to review everything. We can't say, oh, this I don't review. But when you have to bring to the attention to the signaling, be very critical, because again, then we can also the senior meeting discuss. But be very critical with that to not have false positive.
The non-confounding case to the management, right? And I think it's expected that the person who's reviewing it should have a sound knowledge of the product and safety profile. That when he brings it, he should know that this is a triggering article. Could be a signal. All right. My second point was. Go ahead.
Sorry, go ahead.
Yeah. The second point was you was telling that as a part of challenges that. We should have some flexibility in terms of signals. I know that sometimes we get a lot of at one point of time, but again, if you're looking from a patient perspective, that if you take a lot of time, that could be an urgent situation, and patient health can be jeopardized, right? So we're coming from a patient perspective. End of the day, the industry can ask for flexibility, but we cannot stake patients' health, right? I think that's why the regulatory is very stiff about that. You're right. There is no specific timeline for signal reporting, but again, it's also depend on the company by company. They can have an SOP. We can mention specific.
Yeah, well, flexibility. It means also on the methodology to write the signal assessment. For example, if we have again, we know that we have 5. We can have 0 signal for 1 year, 5 signal in 1 month, and we have, as you said. So the priority already should define which should be done immediately and which can, because are non less serious, less important signal, can be done. So there are signals that have high priority, like emergency safety issue, that must be done immediately. So really to have a good way to identify when is an important signal to be done in 1 month, a standard signal to be done in 3 months. And a low important signal can be done in 6 months. Already these give you a possibility to place the appropriate times for the importance of the risk. The other thing, also the flexibility in the process. I was thinking sometimes we should have a possibility to document the signal in a different manner. We can have a good signal assessment report where we interrogate different sources, and the document is very long. But sometimes the signal, we know we don't have cases, a lot of cases in the database, should be mainly the literature review. So in this case, we should have a different template that can satisfy our needs to investigate the signal, but in a different manner, a more quick manner.
Right. but these has to be done by the company, right? From the industry
Yeah.
perspective, they
Yeah.
will not give it that leverage. It's end of the day,
Yeah.
it's company or sponsor responsibility, and they should train the employee in a very well fashion that they kind of figure out all these things, right? So I think again, my thing was like we have to make ourselves better, right? That's my point here. The third point was regarding the increased reports that we get, was solicited and not. So I think we also have some of the tools out there which does DPR, like reporting ratios, where we can have this threshold and you can see these cases only after a certain number has been reached, like EBO 5, and there is PRR. That would also kind of resolve some of those increased number of reports. We don't need to look every one, but you can aggregately we can use the threshold.
Yeah, this is a good example. Of course, also for this report, we should understand that certain reports, certain events, we should not wait for the ERR more than one. If, for example, is pancytopenia. For pancytopenia, maybe it's important to review everything. For alopecia, we can have aggregate the statistical tool that help. So again, yeah, I agree.
Right.
So we should have again, we should contextualize the event.
Correct. I agree with that. Yeah, you should have this important identified and versus under surveillance that can go for the statistical tool, and the one which are very important, you can look every case because that really
every
matters.
case. All the fatal should be reviewed. All the fatal, all the depend on the company and depend on the volume. But I think this is my suggestion.
Sure. Sounds good. Thank you.
Thank you so much once again, Luca Cariolato, for your critical evaluation and presentation on signal detection management. I'm sure all of us have learned a lot. And also would like to thank Dr. Amer for your questions and inputs. Thank you so much. So we now move on to our next presenter, which is Dr. Muruge Russo. Can we have the introductory slide, please? And Dr. Muruge Russo is actually the CEO at Eritrean Pharmacovigilance Center, East Africa. And he is a pharmacist, a pharmacoepidemiologist, and a pharmacovigilance specialist with 10+ years of experience in medical product regulation, health systems strengthening, project management, strategic planning, program review, and building a full-fledged pharmacovigilance system from scratch. He's the head of Eritrean Pharmacovigilance Center and also advisor to the WHO on medicine safety and COVID-19 therapeutics, and a team lead of the Working Group B of the WHO member states mechanism for combating substandard and falsified medical products. Dr. Muruge is a well-versed on investigation of AEFIs and product quality defects, benefit risk assessment, evaluation of implementation problems, quality management systems, and formulation of institutional development plans. He's an experienced medical writer, associate editor, reviewer, and editorial board member in several international journals, and has published more than 45 scientific articles in peer-reviewed journals and 4 books in local language. He's a member of a dozen of international professional societies and consultant on medicines regulation and pharmacovigilance, as well as an international trainer in signal management. With his leadership, the Eritrean Pharmacovigilance Center is currently recognized as one of the top performing countries in Africa. He's a young scholar with extensive experience in resource management and outstanding leadership qualities. And currently he's completing his PhD in pharmacovigilance and pharmacoepidemiology. So I invite Dr. Muruge Russo to talk about monitoring adverse events during immunization campaigns, strategies that can be adopted for COVID-19 vaccine deployment. I mean, such a relevant topic for the current situation. So, Dr. Muruge, please, it's all yours.
Thank you, Dr. Maya, for the good introduction. Okay, let me share my screen. Can you see the screen?
We can see the screen, yes. Also would request you to adjust your video a little so that we can see your face as well clearly.
Okay.
Yes, that's better. Thank you.
Thank
yes,
you.
Please go ahead.
Okay, thank you, Dr. Maya. My presentation here is on monitoring adverse events during immunization campaigns in Eritrea. Basically, it is country's experience, which is aimed at sharing some of the strategies that we have been deploying during the immunization campaigns, and this can be, which might be adopted for, by countries having similar socioeconomic status for COVID-19 vaccine deployment. Okay, this is a disclaimer. Of course, I work for the National Medicines and Food Administration. I'm a regulator. I'm head of the Eritrean Pharmacovigilance Center, and I don't have any conflict of interest. Okay, it's very obvious that pharmacovigilance is very important during immunization campaigns, even though it is also important during routine immunization, but its importance during campaigns is also significant. Maybe we might explain several reasons for that, but the first reason is, during immunization campaigns, there are many people who can participate during the campaigns while they are not really qualified or maybe well trained on immunization practices. And as a lot of people are exposed to the vaccine, several adverse events might be encountered, and this might create outrages in the community, or we might get serious adverse events from immunization during, which can be really coincidental, that really requires investigation of these adverse events. And also it's likely to get clusters of adverse events and so on. So it's really very, very important to have a good surveillance system during immunization campaigns. Having such kind of surveillance system with effective risk communication is very important also to ensure public confidence as well. In Eritrea, we have had two recent immunization campaigns. One is for measles and rubella, and the other is MNA campaign, the meningitis A vaccination campaign. For example, for the meningitis A vaccination campaign, about 70% of the population was immunized. So it was really a huge mobilization, and we had full participation during the immunization campaigns. But just before I take you to our experience during the immunization campaign, I will just give you what opportunities we had for this, for being involved to the immunization campaign. That is, first of all, we have a strong pharmacovigilance system in the country, which has been recognized by different international and national entities as a center with a groundbreaking progress in pharmacovigilance, and also we have good integration with the immunization program since 2016. And accordingly, we have established the National Immunization Safety Advisory Committee. We call it ISAC, and also we have zonal investigation, AFI investigation committees at all levels from the outset. So the aim of this presentation is basically to share our just experience that we have gone through the MR and MNA vaccination campaigns. First, efforts were made to identify key strategies for the prevention, detection, reporting, investigation, and management of AFIs. When we think of deploying the immunization campaign, we started our preparation 8 months ago, and our our goal was not only to identify safety issues, but also to prevent them to the best of our capacity. So we decided to institute or to deploy a smart safe surveillance system in the country. And this presentation will highlight how we have gone through it. Okay, first let's see how we planned it. The Ministry of Health from the outset involved the Eritrean Pharmacovigilance Center, and 3 of its staff were members of the National Technical Committee, which was composed of 17 experts at national level. And this technical committee established the AFI Surveillance Committee at national level, which was really 8 months before. And it was authorized by the technical committee and the Minister of Health to design and execute AFI surveillance strategies for the immunization campaign. So as part of its preparation and as part of the requirements for the smart safe surveillance, we decided first to evaluate and register the vaccines. And during this evaluation process, we have strictly reviewed the safety profile of both vaccines, and we have also mobilized resources from the Ministry of Health and other partners for the safety surveillance system, and structured the strategies that were outlined were published in the field guides of the MAN-A and also MR vaccination campaigns. We have also developed and printed working documents such as AFI reporting form, a guide for management of anaphylaxis, because anaphylaxis has been suspected to be one of the serious adverse events that might be encountered during the field visits, and healthcare professionals, especially at their lower levels, might not have the knowledge on how to manage anaphylaxis, and if we fail to manage the adverse event, it might cause fatal outcomes. So we had a guide also for management of anaphylaxis, terms of references for both the committee and also the focal points at all levels, focal point for the surveillance system, AFI line list to be used by the zonal or regional offices, and also field guides. Therefore, first, a TOT was provided to the health facility managers, zonal administrators, and also EPI coordinators, and so on. And a cascade training was organized by the zonal offices to all vaccinators. And AFI Zonal Investigation Committee that were established years ago were revitalized and refreshed to ensure that they have the capacity and capability of investigating adverse events following immunization. And also instruction was circulated to the zonal offices to identify focal points at all levels, at the vaccine as what we call vaccination post, health facility, district offices, zonal offices, and so on. So we have identified, instructed the zonal or regional offices to identify that. When you come to the implementation process, I will start with the prevention of AFIs. Orientation was offered to all vaccinators on what to expect with the respective of the both vaccines, especially the serious adverse events that might be encountered during immunization and how we can minimize them, and risk mitigation strategies, and the storage and handling of the vaccines, immunization errors that might be encountered, and so on. And also the zonal AFI committees of all the regional offices, they trained healthcare professionals or the vaccinators on the management of anaphylaxis. It was one presentation, but it highlights all the processes of managing anaphylaxis, and strict supervision was conducted to minimize immunization errors. This is sometimes, you see you strictly follow how they reconstitute the time that from reconstitution to the administration and so on, and also the queues and so on. And also this surveillance committee it was mandated to ensure that emergency medicines are available in the country. Not only emergency medicines also, but equipment. And if some of the equipments, after we did the inventory, if some of the equipments or emergency medicines are available, budget was already available for procurement. So we were able to procure also some of the unavailable medicines, and we have to ensure also these resources are fairly distributed to all regions and vaccination sites. When it comes to the case detection and reporting, AFI focal points were nominated and oriented at all levels, and reporting forms, AFI line list template, which we prepared it in Excel sheet, and terms of reference, field guides, and protocols were distributed with the vaccines to all zones and district offices. And AFI reports were being shared on daily basis from the remote areas to the zonal levels. So as you know, the zonal offices have the mandate, or the accountability to put them into the AFI line list to track, which will enable them to track, what we call cluster of events. And the AFI focal points, they were responsible to ensure availability of the reporting forms and documentation of AFIs in all sites. When it comes to the case investigation, cases that deserve investigations were forwarded to the zonal AFI investigation committees. We had very strong zonal AFI investigation committees. For whenever you hear maybe be it via telephone or email that a safety issue is encountered somewhere, and if you call them, it was very likely that this committee were investigating the case. So the cases were being investigated within 5 days, and investigation reports were, as per the protocol, forwarded to higher levels, so zonal level and national levels. Accordingly, risks were communicated to the family and individual levels. I mean, family, individual, and community levels by the already designated spokespersons. When it comes to supervision, intensive supervision was carried out to avoid anxiety-related events. This is basically due to open queues or immunization errors. For example, if somebody is getting immunized, the other children has not, they should be away. They should not be in front of the one who is going to be vaccinated, because they might develop anxiety. So we had strict procedures or protocols of organizing the queues, and also efforts were made by the supervisors to ensure that every event was detected, managed, reported, and investigated. Okay, we have seen some of the strategies that we have used, and was it really impactful, or what was really the output of or outcomes of these strategies? Around 1,000 and 3,600 plus AFIs were documented during the MNA and MR immunization campaigns, respectively, which is in about 2 weeks' time. And majority of the adverse events were minor, and several of the serious cases were timely investigated, and they were found by the zonal AFI investigation. I mean, they were timely investigated, and some of these serious adverse events were found to be coincidental. We had very serious events. Following investigation, we ensured that majority were coincidental, which is really very important. Unless, if there was no system for investigation, it would really compromise the credibility of the immunization program and also public confidence. So as such, issues were immediately communicated at family and community levels as appropriate, and all AFIs are logged they were logged in a line list. Also, they were entered to VigiFlow. So when you see, sorry, okay, this is for MENE. When you see the global WHO global database, which is VigiBase, Eritrea's contribution in detection or reporting of adverse events related to MEN-A or meningococcal A was significant. About 80% of such reports in the global database are from Eritrea. And similarly, measles and rubella, of course, MEN-A, we usually use many of, it's mainly localized in Africa, and it is mainly deployed by African countries, but measles and rubella, it has been used by many other countries as well. So if you see visit the global database or VigiBase, Eritrea's contribution in detection and reporting of what we call adverse events related to measles, measles and rubella was also significant. Of course, the surveillance system or the project was not without limitations. We had some critical and minor limitations. The first limitation was, even though, you know, while we were during the earliest times of integration, the what we call the conception or the perception of the immunization program on the pharmacovigilance system was not, no, it was not good. They were assuming that we are a fault finder, so they were running away out of the pharmacovigilance stuff. And then, but later on, with lots of advocacy and also training programs, they assume or they come to understand that we are allies of the program, not, you know, fault finders. So we had the assumptions that we had no problems of such misconceptions, but during the immunization campaigns, we have seen that there were some confrontational difficulties during investigation, especially during case investigation. But after, you know, we have completed all the processes, they build a confidence on us, and they see how, you know, reliable we were, and or the investigators were, and they were very confident enough on. We know that AFI cases are really, or safety issues of immunization or vaccines are very fragile or delicate. So we were handling these safety issues in strict confidence until investigations was carried out. And then we have been using afterwards a transparent approach. The other problem or challenge were investigation of reports, where, I mean, the investigation reports were forwarded to the national office, maybe sometimes after several months, because they have done the investigation, but they have some, maybe they had some problems with documentation and so on. And also this was in late 2019, and in early 2020 there was emergence of COVID-19. This has really impacted the assessment sessions, because the Immunization Safety Advisory Committee should have what we call the assessment or investigation reports in order to proceed with the assessment or signal assessment of the safety issues. So with the delays that have been observed in reporting the investigation reports from zonal office to national offices, also this caused a delay in assessment of the investigated cases by ISAC. The lessons learned we have, I mean, from this surveillance program, of course, integration was really a master key for our success, because we had good integration with the immunization program. This really helped us to succeed on the following programs. And we really appreciate for this, the genuineness of the EPI program in involving us and the confidence they build on us to take part in the immunization program. And establishing AFI focal points at all levels was also found to be an effective strategy for detection and reporting of AFIs. The other important thing that we have learned is timely reporting and investigation saved the API program from being jeopardized by the coincidental events, because majority of the adverse events that we have investigated were found to be coincidental. Thank you very much. This is Asmara.
Yeah, thank you so much, for the wonderful presentation. And I actually marvel, Muruge, on how you moved from the area of confrontation to now cooperation with your stakeholders, and my compliments to you for the kind of scope of work you are doing, both in quantity and quality. I'm sure all these learnings can be applicable in COVID-19 vaccine deployment as well. So thank you once again. Do we have any questions for Dr. Muruge?
Yes, I will ask a question.
Please.
Okay, thank you very much. I'm really impressed what you are doing in your country. It's like an example for other countries to follow your experience in this. It's very important. Thanks for that. My question is about adverse event active surveillance process, for vaccination. So you are doing adverse event following immunization surveillance, and I'm about adverse event of special interest surveillance process, like active surveillance. Do you have any proposal, or are you doing this now, or are you planning to do adverse event of special interest surveillance process as well, active surveillance in your country? Because it's important to know special adverse events and causal association with the COVID-19 vaccine. This is the first question. The second question is about investigation reports. You said that you have reporting committees, but my question is how you gather this information. Do you have investigation form, which is a part of reporting form, based on WHO recommendations, or you are using reporting forms and gathering this information with reporting forms? So, is this processes are separated? Investigation, going to sites, speaking with patients, with vaccinees, doing investigation of storage, distribution, administration to figure out the causal association, or you are just relying on a reporting form? Thank you.
Okay, shall I proceed with the questions?
Yes.
Okay, thank you, Dr. Liana. As for the active surveillance system, of course, we have developed a protocol for that during the preparation stage, and not only for active surveillance of what we call the vaccines, but also we have been experiencing, or we have been exposed to several active surveillance systems of drugs of interest. For example, artesunate injection. We have about 5 years court event monitoring that we have deployed for artesunate injection. And also we had such kind of active surveillance systems for artesunate, the isoniazid preventive therapy. We have also similar active surveillance is going on for dolutegravir-based ART regimen. So, in collaboration with partners, we have been conducting several active surveillance plans. But as for the immunization program, we need to, we started this with the capacity we had at that time. But from 2019 up to now, we have been, improving our capacity and capability in materializing such active surveillance systems, and our understanding on the smart safe surveillance system, which is not really well understood globally, is also improving. So in collaboration with partners, we are aiming to develop some sub-guidelines or protocols for active surveillance of drugs of interest or vaccines. As for the investigation reports and the reporting forms, of course, we had the reporting form. We had a separate reporting form, but for the sake of immunization, of course, we don't use the written immunization form that we have in the health facilities, just to or to avoid overuse of the reporting forms that are distributed all over the countries. We have distributed new printed reporting forms to all countries, and then with this AFI reporting form, healthcare professionals are accountable to this or responsible to report adverse events from different corners to the respective focal points, or we have a clear reporting channel, by the way. The AFI reporting channel in Eritrea is through the AFI focal points in the country. The AFI program, or the EPI program. The API program, it has what we call grassroots, or it has a cascade programs. It is deep, what we call, until the district points, they have a well-structured channel. So reports are expected to come through these AFI reporting channels, through this channel with clear timelines. And what we usually do is, whenever an adverse event is reported to the district office or to the zonal office, the district officer or the zonal office and the zonal pharmacy in the district and the zonal offices, they collaboratively evaluate the reports, and whenever, according to our guideline, whenever the cases deserve investigation, they inform the zonal AFI investigation committee, and the zonal AFI investigation committee, they have also the AFI investigation forms. It is adopted from the WHO AFI investigation form. It's a 4-page investigation form, and then they went to the field and investigate, and accordingly they have to report the investigation report within 7 days to the national office. This is basically the protocol we have. Thank you.
Yes.
Okay.
So what is the vaccine coverage in your country?
Which vaccine?
COVID-19.
COVID-19 or COVID-19? COVID-19 vaccine, Eritrea is in fact among the countries that are unable to deploy COVID-19 vaccines at the moment.
Okay.
Yeah.
And how about meningococcal A vaccine and measles and rubella that you just talked about in the presentation?
You mean the coverage?
Yes, please.
Immunization coverage overall in Eritrea. Eritrea has been awarded for its high immunization coverage for the last many years. We have a very strong immunization program, but the immunization program previously, it was criticized for not having a surveillance program for AFIs. Though they were high immunization coverage, their quality of services were being criticized for not incorporating vigilance systems into it. This was really a good opportunity for us to enter into the platform. So basically, immunization coverage in Eritrea is very high, more than 90%, but it depends from antigen to antigen, of course. But as for the main A and what we call measles and rubella vaccine, with the strategy to leave no one behind, they have the coverage was estimated to be greater than 93%. But later on, they also did a catch-up campaign in order to ensure better coverage. But I don't know really exactly after the catch up the current coverage for immunization. Thank you.
Okay, all right. Thank you so much for all that data and inputs, Dr. Muruge. And
yes,
in the interest of time, we now move on to the next presentation. And the presentation is on artificial intelligence. And that brings me to the famous saying that a year spent in artificial intelligence is enough to make one believe in God. And to present the examples of the use of artificial intelligence for the development of risk minimization plans, we have the CEO and co-founder at Biologit Ireland, which is Dr. Nicole Baker. She is an early stage, working for this early stage technology company developing artificial intelligence for life sciences. Immunologist and pharmacovigilance professional with extensive knowledge in global legislation and guidelines for medical affairs, regulatory affairs, pharmacovigilance, and clinical trials, gained by leading and managing global multidisciplinary teams. She brings an extensive experience in providing strategic, operational, and scientific leadership throughout the product life cycle from preclinical to post-marketing. Dr. Baker has experience gained supporting or fulfillment the role of European Economic Area's qualified person for pharmacovigilance and responsible person for pharmacovigilance in clinical trials over the past 8 years. She has an overall experience of more than 20 years gained within academia, pharmaceutical industry, regulatory authority, clinical research organizations, and most recently with a startup. She holds a professional diploma in strategic growth, biotech and pharma industry, as well as a PhD in medicine and rheumatology. And she's also an MSc in biomedical sciences, immunology, and a BSc in biological sciences. So thank you so much, Dr. Nicole Baker, for joining us, and we look forward to your enlightening presentation.
Thank you. And sorry, I don't have my camera on, but I think you can all hear my voice. Yeah?
Yes, please. Please go ahead.
Thank you. Thanks. And thanks for the introduction. And I think this is a very good initiative. And thanks very much for inviting me to be part of this this webinar. And I'm very happy to be here. I would like to talk a little bit about examples on how to use artificial intelligence for development of risk management plans and risk minimization plans as well. And I have a couple of questions here where I started to, when I was starting to put this presentation together, I was thinking, what are the kind of questions that you would ask when you're writing a risk minimization or risk management plan, and how can AI help that? We do have a couple of examples of how we've done it in the past using our technology, but I would like to brainstorm a little bit with you on this presentation. So what are the ways? So what is how do you develop a risk management plan? So basically, going back a little bit, we look at the data that we have, that the company will have regarding their products, and what is known, from clinical development to if they have any post-marketing data, which they wouldn't if it's at the very beginning. But they're also going to look at the class. So a drug class is very important in that way, to kind of try to understand what happens in the class. So you could use artificial intelligence to look at data coming from the drug class, and to try to understand a little bit better the known and unknown risks coming from the drug class, and also from the risks that you already have identified doing your clinical development, and that about them, and you also would know the new ones that will be coming into effect, because in clinical development, you're able to find some of the risks, but when you have the drug in use by a larger amount of people, you will end up finding new possible or new new risks that are going to come to light. And then you have the epidemiology data, where you want to see, you want to understand a little bit better the disease, and you want to understand the use of other drugs with the disease. You want to understand where, in what countries, what age, and you want to understand the how the disease progression happens, what age, and all of that, and the numbers. And we have been working on building a platform for us to be able to do that. So we believe that there's a lot of data out there. So there's for you to do that using, you could use your own data, and you can put together your data and then use AI models to identify the risks in your own data. But we have been looking at data coming from the literature, and the literature is a very rich source of information. There's a lot of data out there in terms of how was the use of the drug classes and things like that. But we also think that it's very important to be able to include the local journals, or the regional, we call them local journals, but you could be calling them regional journals, where information is coming not only from what is indexed in larger databases, but also what is not indexed coming from other countries, like let's say Africa or South America, any other region that doesn't have some of the journals indexed, because that is a very rich pool of information that can help us to understand the use of the drug in certain countries. That might be the use of certain, because you might have areas that are more likely to have certain diseases, and using the drug in the same way that is approved, or in other ways, in the ways this is not approved. So I will give you a little bit of an example of work that we have done with our team. And I see that some people from my team is there, and that they have been doing a wonderful work doing the research. And one good example that we did was for PPARs, agonists. So we went back and then we looked at the class, and we looked at the risks for PPARs, and we tried to identify as many as possible. So we expanded the search not only for the product, but for the whole class. And we were looking at the known and unknown types of events that we're finding from the literature. And the literature, when you move then to our AI, and I can explain a little bit more on how we do the AI itself, what models we have. We were able to identify the known and unknown possible risks, and also break it down by what was data coming from, for example, from patient reports, coming from animal, in vitro, elderly, using in children, and things like that. So we were able to stratify the data a bit more. And I think that's where if we have all this data to look at, and we don't have a way of helping us to look at the data in more detail, we can get lost in a lot of information. And then the other way that you can do, and you can manage your risk minimization and your risk management, is using your own data. So using data coming from clinical trials, data coming from registries, and so on, and then put that into our our source of real world data, and then use the AI models to filter that. So basically you are enlarging the amount of data that you have. So you put it into the database, and you enlarge the amount of information you have, and then you use AI to filter out what is important and what is not. And then you will need to have some sort of workflow to be able to help you to find that information. So things that we can look at. So we can look at investigation of medicinal products, medical devices, combination of drug and device, and then the drug class and compounds, kind of expanding into the class. I think it's interesting because you can have a much bigger vision when you expand into the class, especially for products that are not well known. The specific product that you are looking at is not well known, is not fully developed yet, or is in a niche population that you have very little information about it, that it makes it difficult for you to find a lot of information because it's so niche and so small that there isn't a lot of information out there. But then when you expand it to the class, you might be able to find a bit more and correlate with the information that you have for your own product. So we can look at, and then things that we can look at once you have the AI, and the AI is not a magic wand. You need to create the models, and as you create the models and the questions that you want to ask, you will be able to get the answers for the questions that you're asking. But a lot of work goes behind the scenes for building up the models and being able to ask there's not the right and wrong question, but ask the question for you to be able to answer the question. So things that we have looked at and we have developed models for is for looking into the class effect, efficacy. Efficacy it's important as well, because as we move into the use of the drug, we also want to know if the drug that has been developed what is the efficacy, what is the use of the drug, for example. Is the drug being used properly? Is the drug being used off-label? Is the drug actually working? Are we finding lots of reports of the drug not working, for example? And then you can look at animal in vitro studies, toxicology studies, interventional studies, misuse, abuse, counterfeit, medication errors, off-label use that I didn't put in here, pregnancy, lactation, elderly, and pediatric use. See, these are some of the models that we have managed to put together to answer some of our questions. And when we do that, this is the way that the model was built. And I think it's important to talk about, when we talk about AI, that we talk about how the models were built, what was the question that we were asking, and where do we go from a large amount of data into smaller, more specific data that we want to look at, and then how are we making that decision on what it is and what is not, and what gets classified and what doesn't. So because we have a database where put data together, so we do have a large amount of data, and we are trying to build data coming from a database with data coming from open source and data coming from the regional data as well, and also the main big databases, the likes of PubMed. And we are enriching the amount of information that we have, and doing that, we hopefully in a point we will be able to cover global information, where we will be able to have enough information to say that our database is truly a global database that can give us information about what is out there in the whole world, not only the likes of Europe and the US. So when you have all this data, if you don't have a way of managing this data and reviewing this data, that will give you a lot of work. If you go from having, let's say, 1,000 articles to review to having 10,000 or 20,000 articles to review, that's a lot of work, and it will give you a lot of hours in front of a computer. But so we said we want to enrich, but we also then want to be able to filter the data to help us to be able to review this in a more kind of usable way that we can do the reviews. So the first thing that we started doing on the first circle there is to start looking at possible adverse events. And another important thing is, when we do searches on databases, we usually use keywords, and the keywords that we're looking for are keywords that we hope that have been also indexed in the journal, so they will be found once we do the search, that we will find that word. And we think that this, as AI, NLP, machine learning develops, we are seeing more that the use of keywords for doing searches might not be the best way, because if certain words are not indexed within the journals, then you might not be able to pick that information back once you do the search. So we use NLP and artificial intelligence to read the unstructured text and be able to find if there is an adverse event, so we call it suspected adverse event, within the whole context of the words, not only the keywords, but in the whole context of the sentence or the abstract, for example. And we start with the abstracts, and with the abstracts, we look at things that are implicit and explicit, because sometimes you have the abstract that is talking about something, but is not really giving you clear indication that there is in the full text description of, for example, misuse of the drug or use in certain populations and so on. So we try to teach the machine to find implicit and explicit suspicions of having a possible adverse event in the text in general, but not in the keywords that we are looking. We don't look at the specific keywords. And then we start breaking it down. We go down another notch, and we look at identifiable patients, for example. And in that way, we can identify if it's a case report, if it's a case series, studies. You can see if it's used in animals, for example, or in vitro. So we start stratifying where the data is coming from. If it's, and then we go down in saying, is it an elderly, pediatric, and so on. So we start with a large volume of data, and then we start kind of focusing in more and more into the type of data that we are interested. And another model that I want to create is the efficacy model and the use of the drug. So for indications, and then is this drug, what is the drug used for, what indications this drug is used for. So that's another model that we might create in the future. And then in terms of artificial intelligence, you need to have a question. So once you have the question, you can start developing a new model, and you can start teaching the machine how to find to give you an answer for the question that you're asking. But I think it's not as, I want to go back and say, this is not magic wand. We don't create AI models overnight. This takes a long time. So we did that when we were at the university as part of doing the research for this project. And it took over a year and a bit to create models. And another thing that is important for me to say here, we didn't want to create models that are specific to certain drugs. So we wanted to be able to look at the data and have and be able to find possible suspected adverse events or anything that we're saying here in any drug. So we trained models that are generic, so they can find information about paracetamol or about COVID vaccine or vitamins or anything that you want to look at, but it's not as specific to one specific drug, and it can find the way its drug is a generic model for any drug. And then we needed to create a workflow to be able to help us on how to identify the things that we were looking at and how to filter the data. So if you have, we have examples where we started with a very large volume of data, and then we started doing the reviews and finding the tags, what we were looking for, and trying to answer the question. So I want to give you a little bit the examples of things that we have done. So the PPAR was I've mentioned that before, was an interesting one. So we started with the known risks and the information that is known, the known adverse events. So we can use data that is already available, like the SMPC or the product information. we can also go back and look at risk management plans that are published and see what are the table of risks, and then we can start with what is known. So when we developing an idea, so what is known, so this is what we know, this is the risk management plans for the products that are in the class. These are the types of adverse events that we see in the label of the product, and then we start looking at our own product that we are doing the investigation for. So we expand it first, and then we look at a kind of a bigger picture, and then we start looking inwards into the products that is the product of interest, for example. And then the product of interest is a product in clinical development, for example, and then you will have the data coming from your clinical development. You know what are the possible risks. You might not have it, you might have identified risks already, or you just might have possible missing information and so on. And then you start looking at the data and try to correlate what is known for the class and what is information for your own product. And then if it's something that you see very strongly and it's very likely to happen in your own product, but you only see it, for example, in a very small percentage of a patient that is has another condition, for example, and that might be a predisposition to having that adverse event, but then we move to our class, our own product, and that is not likely to happen, then these are the kind of things that you're going to have to look at in terms of deciding what you are going to include and what is important or not. So I think it's very important when you're doing the looking at the literature that you have a very good understanding of your product and also about the class, so you know how the product actually works. So this is a very specific, type of searches that the physicians in the company or the scientists will be able to do, and it's kind of fine tuning and combing down the information to find what is important for you. Another example that we've used was for risk minimization. So it was for a drug that is an antibiotic, and it comes from a very big class of products. Again, a lot of data. When you expand to the class, when you do the search on the class, you get a huge amount of data. And the question was a very specific question on the use of the drug during pregnancy and lactation. So with our models, we were able to filter that, to screen and find information specific for the product itself and is specific for the class, and start filtering them and being able to give an answer. You know, is there a risk? What are what happens, you know, with a patient if she's pregnant? What happens to the baby if she takes that antibiotic during pregnancy? So that was an interesting work as well. And we started with a very high volume of data, and then we filtered it down to more relevant information, and we end up with a couple of articles where it was well described, and we were able to use that. And there are lots of other examples as well where you can use it on an ongoing risk minimization. So for example, if you know the risks that you're already looking at, and you know what information you're looking at, so you can start filtering for that. So for example, if you're using one example with back to antibiotics, where we're doing quite a lot of work, is like, you know, resistance. You know that there is resistance to certain types of microbes, and you want to start filtering for that and start monitoring resistance. So you can do that, and using the tool that will help you to even show resistance, for example, in vitro or in animal models and in culture and things like that. So there's a lot of information out there. That's what I have, and I think everybody knows that. And it becomes even richer once you start using data coming from regional, local data, because the use of drugs is not the same across the globe. So different countries will look at data in a slightly different way, not look at the data in a slightly different way, but the use of drugs might be slightly different in different countries. And the types of diseases and treatments and the type of drugs as well are different. So if you think about Ireland, for example, we might have certain drugs here that are not available in other countries. And then we might not use certain products here because we're using another one that is more available or paid by the government for us to use, for example. So I think it's interesting as well to be able to separate it by looking at trying to stratify the data to look at the use of the drug as well and find more information about that once you have more information coming from larger amount of sources. So this is what and I'm trying to remember any other use, but I open to the audience if they want to ask any questions in terms of what types of questions we can ask the models and what kind of questions, what kind of problems we can try to solve using AI. But before I answer that question, we need to make sure that we build the model before we need the models, AI models need to be built before we can have answers. So it might take a while, but that's why we wanted to have models that are generic enough so we can answer as many questions as possible. I go back to this when I answer I think there are a couple of questions. Let me see. Maybe not.
Dr. Amir, you may ask questions.
Sure. Thank you, Dr. Maya. Hi, Dr. Nicole. This is very enlightening presentation. Thank you so much. It's thought-provoking. I appreciate that. I have some couple of questions.
Yeah.
First one is that I work in clinical development and I work with first in human and first in class. So how does the AI work in this situation? Do you have any thoughts on that?
So first in human, we have looked at first in human data, but it might not be first in class, so we can expand into the class. So if it's first in human, first in class, there will be some development done in animals, or you can look at the toxicology, or even you can look at the molecule, how the molecule works, what are the expression of what cells are expressing the molecule, for example, if it is an antagonist or agonist, or what is the mode of action. So you will understand the mode of action of the product when you clinical development, for example. So you can start from there. What's the mode of action? What is it binding to? And then you can start doing your questions from there. We haven't done any yet with what we have at the moment with the AI models that we have. We haven't yet used mode of action, for example. So how does that molecule, what it binds to? And that would be probably a very interesting place to go to look at clinical development, even going back before you go to first in human. If you're doing preclinical work, looking at in vitro and in vivo, and try to understand what the risks are. So we haven't gone back that much into mode of action, but that's a very interesting question, and I think that is something that it is doable. But you might have, then you will go to the problem of having a lot more data to look at it and start filtering it down.
Right. So from the tox data, and animal data, we can get some of the risk, which is like a potential risk, which can fall as a known risk. How about for the unknown risk? Like I know this, like you don't know, but is there any way that AI can, come up with some suggestions or, trends that they can say based on the mechanism of action, not necessarily it has been done? I'm just thinking out loud, in my mind, that is there options?
So going back to the AI models. So for AI models, you need to have a question. So if you say, I want to find unknown risks, that is not a question that the AI will be able to answer. But what we are doing is trying to look at any So our first model there, which is the suspected adverse event, is just that any suspicion that is something that could be going wrong, that is not quite right. So that's what the machine is reading. So there's a suspicion that something here didn't work quite well as it should, or that is a possibility that this can cause harm. So that is what the model reads, the current model that we have. So if there is a possibility of having a risk, it will pick it up, because there's a possibility of something that have been gone wrong, for example. And it can go down all the way to, for example, then you find that the in vitro work that they've looked at the cells, and there was mutations in the cell. So then it would pick it up, for example. So you know that in that cell work that somebody did, found mutations in the gene. I'm just using an example, and then that would be a possible risk of you if you don't know anything about the class yet. It's new. It's brand new in the class.
Because we are also seeing so many new things that was not seen before. Even in the literature, something, because that's how the products things evolve. So I think I was coming from that perspective, like cross integration, like not necessarily class to class, but mechanism of action somewhere else. Okay. Yeah. I think next question. So with your module currently, it's kind of similar to the Oracle Empirica, where you also have the database where this data was dumped into it, and it can categorize a lot of the events, like it's emerging events, suspected events, things like that. So how is it different with that tool?
It's different because with Oracle Empirica, it's an analytic database. So it's taking the data and analyzing the data and giving numbers. So what we are doing, it's slightly different because we don't, for example, this is not a signal detection tool as such, because it's not going in and we're not coming back with. So there's a little bit of work that the human in the loop needs to do. So you need to say, I want to look at this article. So when you go on the workflow and you see this is a possible risk, you need to actually you need to be involved in the process of saying this is a possible risk and make that decision. But with Empirica, it will give you, and the data that Empirica is looking at, it's cases. So it's coming from, and it's already coded, for example. So in this instance here, the data is not yet coded, and it is something that we can look at in terms of if we find adverse events, possible risks, we code the events to be able to analyze them a little bit better. So we have been working in collaboration with other companies and companies that are looking into signal detection and trying to bring their own data. So you would have the likes of Empirica, and then the data that we're bringing from the literature, for example, from any registries, and putting them together, because Empirica most of the time will have the data that was given to, like if it's a dump from fairs or from Vigibase, and then the data from the database. So I think for new, what we're doing, it is different because we are not, we are looking at the text in general and trying to find context on what is said in the text of the articles, for example.
Natural language processing, correct?
Yeah, I think so.
Last question before I let you go. Have you compared the time taken by a human person to review an article and find something versus your tool finding that articles? Did you see any difference, and what was that?
Yeah, and we see about 70% of difference. So if you start, we have started with let's say we started with 20,000 articles to review, and we end up with 5 articles at the 5 articles that had useful information for us. So, but then the AI is filtering out quite a lot. But it depends your level of confidence on the AI models, that it will, because they will give you the data in a way that you can make a decision, or you can make exclusions on the filtering. So there's lots of options. So if you say, I only want to know information coming from a patient, for example. So you can start filtering, saying, I want to only look at, so it will bring all this information and you will choose, I want to look at patient data and using elderly. So then you're filtering just for patients and elderly, for example. So it will help you to be able to see the use of the drug in elderly. So you can exclude everything else, and that the machine can do for you. Or use, like I said, the use case that we did for pregnancy and lactation. So we used the pregnancy and lactation models, and then we found only events that were in that population, for example. And then you can start all the filters. For example, we are looking into being able to identify where the report is coming from. And then we could do it by region, and that would be interesting. Say, let's look at Africa, let's look at Eritrea, let's look at a specific region for what types of events are happening in each area. But that can be misleading, because if the articles are published through, let's say, the US, then most articles will show that is coming from the US, but you need to look at the who is the author and find that information, for example.
Right, makes sense. Yeah, I think this is very interesting. I would love to volunteer, and I have some ideas in signal detection, kind of correlating with several things. So I'm not sure if I can get your contact information. Maybe I can volunteer.
Yes, definitely. And then another interesting thing that we want to do is to expand it in SMQ. So when we start coding the event, let's say we're looking at the specifics. I don't know what I want to say. Like, I'm not going to use the Stevens-Johnson. I think Stevens-Johnson is the most used one. And then paracetamol is always my example of the drug. But yeah, so if you trying to look at a specific event, but you want to expand it to see if there's any of the symptoms and signs, and we can look at that as well. So the model is looking at symptoms and signs. So the next one, I think it's something that we can start looking. So if you want to say, I want to look at the symptoms, and it will start helping you to find the symptoms. So if you have, let's use Stevens-Johnson. So you have Stevens-Johnson, you are worried about starting with a rash, and then you can start looking for rashing symptoms and signs and classify it that way. So the interesting thing about AI and doing models like that is that you start using classifiers to help you to filter and to put the data that have the information that you want together.
Yeah, I think you can also use this kind of module in terms of grouping the events in label. I think I'll stop for now. Yeah, thanks. I think people want to ask questions.
Yeah, thank you, Dr. Nicole, for a nice presentation. I would like to invite Jinosian to say something. She has raised her hand, I think.
Yep. Thank you everyone for a great presentation.
Sorry.
And yeah, can you? Hello?
Yes, we can hear you. Go
Yeah,
forward.
thank you. So actually, like partially my question was already answered, but mostly I want to know like how these algorithms would work for new products, because like if it's based on like already available data, it's clear and we can use like complicated algorithms. But will it be useful? Do you think will it be useful for new drugs that we don't have too much information? How dependent it's on like database and data amount?
Yes, so for new drugs, then we tend to look to expand it to the class. So if it is a drug that doesn't have a lot of information about, or even if it's a compound, so let's say it's a compound N234, there's not a lot of information about that compound. But if it is part of a class, then we can do that. And the way we built the model was by looking at all the classes. So we expanded the searches into different classes, and we trained the model to understand that adverse event is not. So and I think that's one important thing. So when we look at possible adverse event, we don't know if that adverse event is related to the product or not. And within the text, you could have actually like 5 or I don't know, different drugs in the text. So, and we are looking at one specific drug, and that event is happening to that drug, not to the other 10 or 5 drugs that are in the text, for example. So for a new drug, we expand it to the class.
Okay, thank you very much. And maybe another question, like, is there any evidence, like validation compared to like human signal detection compared to the one that AI is giving? Is there a big difference between, or it is just the matter of time and by AI it's more quicker or it's more precise too?
Yes, so and that's something that we are working really hard to be able to show the results. And so it depends on the recall of the data. So for example, it's not easy, but it's easier to do the collection of weekly ICSRs, because the ICSRs that you do in the collection on a weekly basis is more black and white than signal detection. So you know that you have if there's a reporter, if there's a drug, if there's an adverse event and a patient, then it's an adverse event to your drug, and you classify that and you select that. But looking for new signals is a little bit more subtle than that, because you need to have the knowledge of the product. You need to have it's not about having an adverse event or not, but it's looking at the potential of that event to cause harm or to be a risk. So I think it's still the human in the loop for signal detection is much more important than for your weekly ICSR screening, for example, because you need to ask the question. You need to look at what you want to answer and look for any, like the unknowns. So you will find the unknowns. You will find rare events coming from the literature, things that are not yet in your label, for example. So these are the ones that you want to look at. So the unknowns and the more and the risks that you already know. For example, let's say Stevens-Johnson for your drug, you already know and you have it classified as a possible or a risk. So when you see that and you start seeing a higher volume of Stevens-Johnson's coming from the literature, for example, then you definitely have to act on it. But there might be. I want to go back to the pregnancy, because that's where pharmacovigilance started. So that's a good one to use the literature to possibly start looking at class effect on that. If there's any risks coming from even toxicology, going back to cell work or animal work, that could have an effect on the embryo, for example. And then we bring that back to the product and make sure that there's no risk at all for using pregnancy, for example.
Thank you very much.
We're not going to be certain if it hasn't been seen yet.
Thank you.
All right, thank you so much, Dr. Nicole Baker, for your exciting presentation and for showing us that how artificial intelligence comes to the rescue in terms of developing the risk management plans. So thank you once again, everyone. Liana, you have a question?
Yeah. Just please, before going to the break, Nicole, thank you very much. Very, very informative. And I think that with huge volume of data, pharmacovigilance, contemporary pharmacovigilance, I cannot handle any kind of activities without help of artificial intelligence. This is for sure. My question is about the unexpected events and like signal detection and artificial intelligence. Connection process of connection of two kind of activities. If I'm wrong, just correct me. But my question is about unexpected events. New safety information which is coming from signal management activities. So you said that you should have question for doing kind of artificial intelligence activities, yeah? Or you have to know the adverse event to look for. And signal detection is a bit different. We have to find out new safety information. So am I right that they are not they are not replaceable? I mean, the process of signal detection.
They are fine. So our model will find any possible risk, any possible event, because it expands in a way, is restricted to the keywords that you would have, like I don't know, adverse, for example. So even if there's no mentioning that this is an adverse event within the abstract, the AI will find that, because the AI has been trained to find any possible risk. So even if it's unknown, the AI will find it. So that's why that's how we started asking our question, because we didn't want to miss anything. So we are very cognizant that we don't want to miss anything. We want to be quite protective of the data that we're looking at. So we expand the search in a way that increases the amount of data. So it's different from when you do your keywords, where you put a certain keywords and you reduce the number of information that you get. So what we do is the opposite. We expand the amount of data that we are going to look at. And then with AI, we start using the model. So it's not only one model. One model is to look for possible adverse events, or possible risks. So that's the first question that so on that big circle, that's the first question that we look. So we don't miss anything, because we are looking at possible risks.
Okay, so but you are looking at abstracts. So I understand that you are looking for suspicious adverse events somehow, not to miss anything from. I mean, but the whole program is based on abstracts. So what is the percentage of missed data? Because you are not looking at full texts, but on abstracts. Do you have such data?
We have. So we can show, we have done quite a lot of different projects where we look at the data. So we can say that by looking at the abstract, we can probably find more adverse events than by looking at keywords, because the model is looking at the so we are looking at explicit and implicit information. So if within the information of the abstract, there is any sentiment, because the AI looks at, it analyzes the way the words are constructed. So if there's any sentiment within the abstract, that there's a possibility, even a faint possibility of having an adverse event or a risk, it will tag as a possible. So we don't say for sure it is an adverse event. We say there's a possibility of having an event or a risk on this abstract, and then you can move on to the full article and find. But we get asked that question a lot, and that's something that we will probably move into looking at the data coming from the full article. But full articles sometimes are behind paywalls, so that is more difficult for us to do, because if we have to purchase 10,000, 20,000 full articles to read, that is not doable. Nobody will be able to do. And that's something that we are working very hard, is to be able to look at to kind of democratize information and scientific publication. So that's why we go back and try to pull information coming from the regions, and also to any, to open source is what we obviously there are articles that you have to pay for, but we will try to present the free what is open first.
And the last question, do you have language constraints? I mean, are you looking only in English publication, or do you have some range?
That's a very interesting question, and thank you for asking. So what we found is that most abstracts, even if the full article are in another language, the abstract would be in English. So that's what we found. And then what we do, if we find an abstract or an article in another language, it might be moved to the side, and if we cannot translate it. But if we can, we use machine translation. And then when we use the name of the product, for example, so the name of the product or the class, we use dictionaries for that. So we will expand to be able to pick it up in any language. So let's say paracetamol. So paracetamol will be, we will use all the synonyms of paracetamol, and it will expand to any language or any. So it will find a possible adverse event for paracetamol in any language. But if the machine cannot translate the language that, or it's not giving the right answer, so what it does, it puts to the side. So you have a tab on the workflow that will say, I think it says another language or something like that. So that wasn't used. So it means go and read it yourself. But yeah, but what we found is that a lot of the abstracts are in English, even if the full text is in another language.
Yeah. Okay. Thank you so much, Nicola. Really very impressive presentation, and very nice talk on this important topic, or artificial intelligence and pharmacovigilance. Thank you so much.
Thank you. Thanks.
Yeah, thank you once again, Dr. Nicole Baker. It was absolutely amazing to see, the involvement and how everybody was giving ideas. And I'm sure it will help in tremendous mutual learning. So with that, I would request you to stay back, and also our other speakers, which is Dr. Raghunath, Dr. Luca, and Dr. Muruge for a small panel discussion with Dr. Chinmay Mahapatra. Thank you so much.
Thank you, ma'am.
Yeah, sorry to say that we are a bit over time, and I can stay not more than 5 minutes if you have important questions for me, because I must disconnect. Unfortunately, I will stay 5 minutes more if you have questions about signaling. I think that one point that I wanted to say about the artificial intelligence is great tool, and I think that help a lot in our safety evaluation and safety detection. However, I would say that we have always to handle with care when we enter in the artificial intelligence, because we should understand the basis behind. Otherwise, we can risk that we don't understand the stratification, how the tools stratify the event, and we have the risk that then we analyzed behind all the data analyzed already by the artificial intelligence. My thought was about EVIDAS, for example. When there was EVIDAS, there were a lot of information there in the listing that we were able to retrieve, but I think that few people really understood how to use correctly this information. And now it happened that people in PV, they do a lot of work, they invest more time that was supposed to do, and now there is a risk that people have overdone a lot of things and they missed the important information that can come from EVIDAS, for example, or fires. So the thing here, artificial intelligence is important for our activities, but what is more important is to understand behind, so like this we can use properly the tool and more efficiently. Otherwise, it can be a paradox, but can cause more work and at the end, not useful work, I would say.
Thank you, Doctor.
I totally agree. Yeah, because without using the brain, I guess it's impossible. So I mean, both should be here, and technique, artificial intelligence, and human brain, because otherwise you cannot proceed. Luca, may I just do one question, and because you are leaving, as I understand. I'm regarding non-clinical data. You mentioned clinical trials and all other kind of. Yeah. What about non-clinical data?
This I didn't probably include, but I put like other sources. Of course, non-clinical data are very important. Again, I think that non-clinical data, to identify a signal directly, we can always ask ourselves, they can identify potential risk, because then we don't know what's happening in human. So if we find that in some animal we can have a certain reaction, it doesn't mean that automatically we'll have it in human, but they can identify important potential risk that then must be carefully evaluated when we do the analysis in humans. So for sure, they can identify signal non-clinical data. What is important about non-clinical data, I think, nowadays is, as I said, to identify potential mechanism. for a reaction. If a drug we discover that can interact with a particular signaling pathway in the cells or on other contexts, so we are trying to explain how the mechanism work, and we use, of course, preclinical model. This would be very important because it can open again other important scenario for other events. So is to better characterize the risk, is better to understand the context of the risk. So non-clinical data is extremely useful, and I love to use non-clinical, but for signaling particularly, we can use it in a different way, as I said, to already characterize the risk or to identify potential risk. Doesn't mean that will be a risk to identify. Then we have to check in human if this happen. But it's very important to identify important potential risk or potential risk.
Yeah, I think
are you adjusting it to be human equivalent? Are you adjusting this data? I mean, the doses, for example, which used for non-clinical data, are you adjusting for human to be human equivalent?
Yeah, this for sure. But if, for example, also we have some toxicity, if we want to identify the toxicity of a drug, and of course we check in preclinical, and we identified certain potential risk, is extremely important to use, and extremely important every time to will be great. Often in post-marketing, we don't do that anymore, but will be important to request when we assess a risk to check data in preclinical again, and new data with new hypotheses. What I'm saying.
Yeah, for the clinical trials, you can also use the data to support your signal, right? That gives you a potential signal. But having said that, if you experience few events, you can retroactively go back and link up that this is a possibility, and you can call it as a signal. So that is a big deal, because in clinical trials, that's all you depend. There's no other data you support to signal validation. So that's the main thing. You either use it as a risk identification, or you use it as a rationale why it is happening in your data.
Exactly, the rationale is important to understand. And again, when we then often, for example, this week I was looking for a signal that was done in the past in the company where I'm working now, and I checked this potential signaling pathway that can explain the risk. And of course, will be very important to come back maybe with some preclinical methods to check, because this could explain a lot of other, not risk, but also how we can minimize the risk more efficiently, not just to report the information in the label, like this drug can cause this risk. This is just an information, it's not really a minimization way, but really to more efficiently challenge the and maybe with some corrective treatment really minimize the risk. So this is extremely good, the preclinical, and the thing that I completely agree that they can identify signal, but of course we have always to remain on the level that preclinical and doesn't translate directly. It happened, we observe in certain experiment, this will happen in human, or this can be applicable 100% to human. So we have always to keep this.
That's why the clinical is more complex, because you're working in a gray area. You really have to have critical thinking with this rationale coming out. So that's a very tough thing, you need a medical judgment. Thank you.
Thank you. Sorry, I must disconnect. If you have any question, I just sent my email and my LinkedIn. I will be happy to answer your email or to have a discussion. Unfortunately, I had already something scheduled after this morning session, so I hope that I can connect in the afternoon. Unfortunately, I need to disconnect now.
Just to say thank you very much.
Thank you. Bye.
I have one question to Dr. Raghunath. Like, as the trend is shifting towards looking for the social media for the adverse events report, right? So do you think that is a valid report, or the MAH should look into that, because we don't know whether that is a correct one or a fake one, or anything anyone can post, right? So what criteria we can create that these things should be included or not, like that? Means that is just a question. As the world is shifting towards social media, so that is the main question what I have.
I totally agree, Dr. Chinmay. I can understand your question. See, we have something called QA, Quality Assurance Department, in place. So if somebody report an adverse event and that medication has a fake, what we can do is that we can, ask the person to sift the used bottle or use a sample to us. And from that, we can make out whether that is a counterfeit medicine or what, that we can make out. So it's not like that just, looking into the pictures and we can, consider this as an adverse event or this is a product quality complaint. That we cannot make it. And there are certain follow-up questions, designed follow-up questions we have to ask, to get a further information. So based on that, we can make a judgment and proceed accordingly. Yeah.
Right. See, suppose it is a big companies, they are having that things in place and they can follow up with the patient, right? But for generic drugs, they don't have any PV system in place in their company. So in that case, who can take the responsibility and who can do that work?
See, ultimately, it's responsibility again lying with the product owner, whether it is a manufacturer or marketing authorization holder. It's their responsibility to evaluate.
Right. So we all have to work on that area also, right?
Correct. Yeah.
Thank you. And I'm having one question for Dr. Nicole also. Artificial intelligence is good and it is good whatever you have presented, and really means if we collect the data from the literatures and we can get to know the profile of the drug in much better way, right? But still, do you think that artificial intelligence is having any impact on PV audits, pharmacovigilance audits?
In terms of how the systems are being audited, or to support auditing, having better. So there is we haven't developed, and that's something in the back of my mind that I would like to develop, that is related to audits and compliance, that you have some sort of a checklist to use AI to make sure that you're compliant. That is a possibility, and I don't see it being too complicated. But in terms of going back to what we have developed, you need to make sure that the model that you're developing, you can explain. So it's the explainability of the model behind. So you're able to, if there's an auditor, you're able to explain how you created that model and what goes in it. And then there's all the software documentation, so and then it will comply with GXP and FDA CFR guidance, and there is quite a bit of guidance coming out now in terms of software development and AI development on how to do that across the industry. So Transcelerate has published and what types of models we want to create. I think going forward, a lot of companies probably want to go towards the more interactive learning capacity of a model, that the model can learn as they go along. But and it's not a rigid model. But I think for now, with the way the legislation lies, we are still working on static models where we can prove how the model was created, so how the model was developed. Because if you keep developing the model further,
process,
you're changing the process, so you're on an ongoing basis changing the process. So I think for a while we're still going to have static models where we create the model and we know exactly what is doing, and we can have the knowledge of how it's working. But in terms of compliance, softwares and how to use AI for compliance, that's I think it's already used. I was in a call yesterday, and they were looking at taxation. and they already using, which is similar, but they're looking at numbers. So they are using AI software on finance to make sure that the numbers match, that the taxation is done properly, that and then flagging certain errors based on the knowledge that he has from lots of inputs. But it's already been used in legal as well. So I think in pharmacovigilance, we don't need to always reinvent the wheel and start something from scratch that hasn't been used before, but we can look at the other industries. So we can look at aviation, we can look at financial, legal, and try to bring systems that work well for them into our industry, and obviously make it suitable for what we do.
Thank you so much. Dr. Liana, you can take over.
Okay, just maybe we're out of time actually, Chinmaya. Maybe we can announce some break for 10 minutes and
Yes.
we'll start again. Yeah, okay.
Yes.
So, dear participants, we will have break for 10 minutes and we'll start at 2 p.m. Armenia time. Thank you. Stay with us. Okay. Hope all are with us. It's 2 p.m. Armenia time, so we need to start the second session of today's webinar. I would ask Chinmay just share the slides to present the first speaker. Chinmay, are you with us? Let me to check. Okay, I guess Chinmay, please go forward. And the first speaker is Dr. Liana. Can you just change the slide? Yeah, okay. Thank you. So I'm honored to present the next speaker, Dr. Liana Gross-Martirosyan, alternate Prague member from Netherlands, Medicine Evaluation Board of Netherlands. Topic of today's presentation will be pharmacovigilance in the European Union, the role of Pharmacovigilance Risk Assessment Committee. Dr. Liana Gross-Martirosyan is an alternate Prague member from the Netherlands Medicine Evaluation Board at the European Medicine Agency Pharmacovigilance and Risk Assessment Committee, Prague. Liana is a medical doctor with a master's degree in public health and a PhD in pharmacoepidemiology from Groningen University in the Netherlands. She has been with the Medicine Evaluation Board since 2012. Before joining it, Liana worked as an epidemiologist advisor to WHO Europe. So I'm passing the floor to Liana to start the presentation. Liana, are you there?
Oh, good afternoon. Good afternoon, everyone. Thank you very much for the invitation. I'm pleased to speak at this conference. I'm just hoping that sharing my screen will work out. I'm sorry, I do not have much experience with Zoom.
Do not worry, we will let you know when we will see your shared screen.
Do you see now my slides?
No,
No.
not yet. You shared your screen, but you have to open these slides, I guess. Yeah.
Yeah, is it now okay?
Yes, all is okay.
Okay.
Thank you.
Okay. Sorry very much for this hiccup. Yeah. Well, being an alternate member of PRAC committee of the EMEA, I would like to use this opportunity to provide some insights in our work in well, providing pharmacovigilance activities in the European Union. And I would like to start with a slide which shows the increasing importance of pharmacovigilance nowadays, because as you know, in many well, continents, including US and also in Europe, there is an increasing tendency to bring important medicinal products quicker to the market. And this is especially important for critical medicinal products where there is an unmet medical need to provide early access to important medicines for the patients. And that means that there is a possibility to give marketing authorization to products that have a rather limited set of data in the clinical development program on both efficacy and safety. Well, however, considering the unmet medical need, there is a possibility to bring the product somewhat earlier with limited data set, although the efficacy needs to be proven and safety should look okay. But because of the limited safety database, it's even more important in such situations to have good plans how the safety of such products will be monitored post-marketing, because obviously in such cases there is a lot more work to be done by pharmacovigilance professionals than in case of full marketing authorization. And well, I can imagine that the audience of today's conference is very much familiar with this concept, so I will just provide very brief introduction that in general, the safety of the medicinal product is determined by both type A and B adverse reactions, and type A is more predictable by us because it is more dependent on pharmacological mechanism of action of the drug, while there are other factors that may influence the safety of the product, and this is related to the way that patients may use the drug, which may impact the safety. It has to do also with the severity of the underlying disease, and also, well, we have now more and more increasing information on the role of different genotypes in terms of safety when a product is used by different patients. There are also, of course, usage and prescribing related factors that affect safety. Well, normally we may not expect certain safety events if the product is used in a correct way, as it is determined by the product information. However, in reality, in clinical practice, patients use many different drugs, and this may also affect the safety of the product. Well, this slide shows some important milestones in the history of pharmacovigilance, and as you know, some first information that showed the importance of monitoring of drug safety was arrived already in 19th century. However, there were some well, important moments in the history which allowed us to have good legislative framework, which we are still benefiting from. And probably the most important ones I would like to highlight the FDA Act, which was established in 1938. And there were then other several important developments. And for the European region, I would like to highlight the new pharmacovigilance legislation that was implemented in 2012. It's not new anymore, it's already 10 years, but that's legislation that is currently in place, at least in the EU, which determines the work of pharmacovigilance here. Just to highlight some important aspects of the legislation implemented in the EU, probably the most important ones is the introduction of good pharmacovigilance. May I ask everyone who is not speaking to mute the microphones? And thank you. Yeah, so the introduction of GVP modules is providing guidance to both pharmaceutical industry, but also to the assessors working in different national competent authorities in Europe. And these modules are being regularly updated to provide up-to-date information. There was also quite some important work to strengthen the Eutravigilance database. One of the important aspects was inclusion of non-serious adverse events that were reported outside of European Union. So previously it was not obligatory to report such events to Eutravigilance. Now it is a requirement. Probably I would also highlight the possibility with this legislation to involve not only healthcare professionals in our work, but also involvement of patients and citizens in European Union when there is an important assessment ongoing. And also with this legislation, the Pharmacovigilance Risk Assessment Committee was established previously before 2012. Pharmacovigilance was regulated by one of the working parties of the CHMP committee. So previously it was not a self-standing committee. Probably it's interesting to highlight what is the composition of this PRAC committee. So every member state in European Union, every European country is represented in this committee. There are two members from each country, one member and one alternate member. So currently, after Brexit, we have 27 EU countries participating and a small number of EEA countries. The last ones are not non-voting members, but they fully participate in the discussions taking place in Prague. So, so there are about 60 members representing the countries. That's presented on the left side of the slide and on the right side. So besides the representation of the individual European countries, we also have PRAC members who are experts in specific areas which are important for pharmacovigilance, and these experts are being appointed by European Commission, and they have a mandate for 3 years, which may be prolonged for one more time, and then there is a new election process when this period expires. Currently, we have 6 expert members in the committee, and they have important expertise. For instance, we have an expert who has a lot of expertise in use of drugs in pregnancy, but most of them have extensive scientific expertise in pharmacoepidemiology and clinical pharmacology. And also what is very important in this committee, there are members who are representing patient organizations, patient representatives. So there are 2 patient representatives, and also we have 2 members who represent healthcare professionals. On this slide, I have provided an overview of procedures which are being discussed or adopted by PRAC committee. So basically, it's everything. Any procedure which is being implemented and assessed in European Union is eventually being adopted by PRAC before the recommendations are being sent to marketing authorization holders. And the work of this committee involves both pre-authorization and post-authorization procedures. Before the product is registered in the European Union, our role in PRAC is to assess the risk management plan. So to see what are the most important safety concerns that need to be included in the risk management plan, and also to decide if the product is registered and brought to the market, what are the activities that we need to have in place, such as studies to further characterize certain risks, or what type of risk minimization measures do we need to have in place to ensure safe use of the product. Well, having said that, I have to say that most of the work is focusing on post-authorization. So products that are already on the market in the EU, and the safety of products which are already on the market is being issued through a number of procedures, which are probably very much familiar for the audience today. Well, we have Psur procedures, which is periodic safety update report, and there is a risk proportionate approach nowadays, which means that the products that have important ongoing safety concerns, they have a more frequent PSUR cycle, and we may see the review of safety of such products every 6 months or every year, while we have products which have very well-established safety profile that have been on the market for a long period of time. And for such a product, there is a possibility to have a very long PSUR cycle. For instance, we have products that have a cycle of 12 years. Then there are signals, which is a very important procedure for national competent authorities who review themselves the data available in Pharmacovigilance, but also in the literature, to evaluate if some preliminary information indicating a potential safety issue, if it is indeed a true adverse drug reaction or it was a false signal. We are obviously assessing also post-authorization safety study protocols and the results of such studies, making recommendation if there is a need to update product information or if there is a need to implement new risk minimization activities. And I would also highlight the referrals. Well, we do not have a lot of referrals, luckily. That's probably one of the most resource intensive procedures in pharmacovigilance in EU, which are being triggered in very rare circumstances when there is a very important new safety information which puts the benefit risk of the product at risk. And then there is a need to re-evaluate the benefit risk to see if the product may remain on the market, and if it may remain on the market, what are the updated conditions, what type of actions need to be implemented in order to ensure the safe use of the product despite of this new emerging safety information. And on the right part of the slide is just an overview, just randomly chosen PROC meeting. This one is from November last year, which shows the number of different procedures that have been discussed and adopted during this specific meeting. Just to say that PROC committee has monthly meetings. So every month there is a meeting which takes place, and it is actually four full days meetings. They're beginning on Monday and finishing on Thursday. And basically on Thursday, there is a need that all conclusions have been finalized, so the recommendations can be forwarded to the relevant marketing authorization holders. Well, I thought there are too many different procedures being discussed at PROC, but to give some more insights, I have chosen two specific types of procedures, being signals and referrals, to provide a little bit more insight during my presentation. So on this slide, I have provided some overview regarding signal evaluation at PROC level. And I would like to highlight that signals that are being evaluated at PROC level, this refers to slightly different type of signal than what marketing authorization holders are discussing, for instance, in their PSURs or DSURs. So At PROC level, the signals that are being evaluated are found by EMA or national competent authorities themselves. So these are not signals that have been identified by the pharmaceutical industry. And signals are mainly triggered either by member state, one of the European countries, or by the EMA. Most often the source of such signals is the review of hydrovigilance data. The data in hydrovigilance are being evaluated on a regular basis. Most of the time, this is once a month, and the review in hydrovigilance for centrally authorized products in Europe are being conducted by EMA, and the review for nationally authorized products, so all adverse events available in hydrovigilance are being reviewed by member states. So we have sort of work sharing. So every country in Europe is responsible for a certain number of active substances. So the country which is in lead for a specific medicinal product or actually active substance is reviewing this data. And if there is a conclusion based on the monthly review of hydrovigilance data that there is association that might be a potential signal, then signal is being triggered and evaluated and brought to the PROC. Occasionally, such signals arise not from hydrovigilance, but from review of published literature. But in certain cases, we also have signals that are based on emerging safety information from, for example, ongoing clinical trials. After this potential signal has been identified from one of these sources, most of the time, based on this initial information in hydrovigilance, it's not possible to make the definite conclusion if it's a true adverse drug reaction or not, and there is a need to review other sources of data. And in such cases, most of the time, we develop a list of questions which is being forwarded to the relevant marketing authorization holder, and in such request we draft request for review from additional sources of information. So, we have seen data from hydrovigilance or a specific study or a publication, and then we would also like to see, for instance, information coming from toxicology studies which are available in-house by the marketing authorization holders, information from the clinical trials, from the clinical development program, and in some cases we also request the review of company safety database, because in some cases we expect that company safety database may have some additional information on top of what we have seen in hydrovigilance. So after this additional analysis requested from the marketing authorization holders are being submitted for using a certain defined timetable, then the PROC reviews this data, and then only following assessment from all sources, the final recommendation is provided. It's forwarded to marketing authorization holders, and in principle, it's then legally binding and is relevant for all marketing authorization holders that have product containing those specific substances. And the recommendations for all signals are being published on EMA's web page shortly after that according PROC meeting. This slide shows the signal outcomes following evaluation of all available data, and shows what are the potential outcomes following signal evaluations. So the pie chart, you can see that approximately in half of the cases, there is a recommendation to update product information. And most of the time, this is update of section 4.8 of the SMPC, followed by update of section 4.4, which is warnings. Although we did have several signals which resulted in even update of section 4.1, which is the indication, or 4.2, which is the posology. So in half of the cases following signal evaluation, there is a need to update product information, and in well, approximately another half, well, the conclusion is that the signal did not show, the evaluation of the data did not show that there is a reasonable association between the adverse event and the product, and therefore there is no need to update product information, and further routine monitoring is necessary, for instance, within PSUR. And in very rare cases following signal evaluation, it's possible that the conclusion is that a different procedure is necessary, and you can see that for instance, in 4% of cases, there was a need to trigger a referral procedure, which means that the evaluation of the signal showed that well, it is a true adverse drug reaction, and it's so severe or important that it is impacting the benefit risk of the product, and there is a need to start a full re-evaluation of benefit risk of the medicinal product. On this slide, I have provided some well, tables and a graph which shows the characteristics of drug safety signal that may predict the need for product information update following the assessment. And not surprisingly, in the study that we have performed a few years ago, we have seen that having different data sources supporting the signal, so not only spontaneous reports, but also, for instance, availability of supporting data from clinical trials and from publications, and in some cases also from toxicology studies. So if there were at least 3 different types of sources supporting the signal, there was an higher probability that the product information would need to be updated than the other characteristics associated with product information update are mechanistic plausibility, seriousness of the event, and age of drugs being less than 5 years, which is also not surprising, as for, well, relatively newer drugs following introduction to the market and used by a larger number of patients. In the first 5 years after marketing, there is a higher chance of detecting new adverse drug reactions that have not been seen in the clinical development program. And in the graph, you can see the sections of the SMPC that are being updated most frequently following signal evaluation. As I have already mentioned, section 4.8 and 4.4 are the most frequent sections. And I can imagine that it is quite recognizable by everyone attending the conference today. And now I would like to move on to referral procedures. As I mentioned, although these procedures are not being triggered in European Union frequently, this is one of the well, quite critical tools that we have when there is a new serious emerging safety issue for a product which requires full reassessment of benefit risk of the product. Well, depending on the type of the product, if it is a centrally authorized product or if it's a nationally authorized product, there are different types of referral in place. I will not go to differences on procedural level, but from the content and scientific perspective, the assessment basically is the same. It is a request to the companies to provide both safety and efficacy data in this procedure in order to allow not only evaluation of the effect of the safety, but also to consider the balance of efficacy versus safety with this newly received safety information. And to further elucidate or provide an example of a referral procedure, I have chosen the case of valproate. I don't know if some of you are aware of this ongoing issue, but it's a very interesting actually and striking example of safety issue, because valproate is a very, very old drug. It has been on the market, since 1960s. And the fact that it's a highly teratogenic product is also absolutely no new issue. It has been known that it's teratogenic already from 70s of the last century. However, it's a striking example which shows that despite of being a very old product, and despite of the fact that this safety issue, teratogenicity, is also a very known issue, still we still, at least in Europe, we still have issues with the right use of the product, and we still have ongoing unresolved issues how to make sure that this product used in a safe way in clinical practice to avoid pregnancies while the product is being used in women of childbearing potential. Well, the fact that valproate can cause congenital malformations in 10% of cases during exposure during pregnancy was very well known, as I've mentioned, since second half of the last century. But the fact that it may also cause serious neurodevelopmental disorders, that's relatively new information, which became available late 90s. And as you can see, the magnitude of the problem, the magnitude of safety issue is quite high. So 10% for congenital malformations and up to 40% for neurodevelopmental disorders. And by neurodevelopmental disorders, we talk about quite serious issues like Asperger's syndrome, autism, serious learning disabilities. So summing up these two congenital malformations and neurodevelopmental disorders means that every second child who has been exposed during pregnancy will have quite serious health issues. And in 2014, there was a referral procedure triggered in European Union because several countries identified, through evaluation of nationally available data, that there are still pregnancies in women who are using valproate. And therefore, in 2014, Prague decided that the product information of valproate-containing products should be updated, and measures should be implemented to restrict use during pregnancy. And for instance, at that time, there was a warning introduced in the product information, which warned against use of valproate during pregnancy. There was an advice that all women of childbearing potential who need to use valproate need to use contraceptive measures, and only in such case valproate may be prescribed by the specialists. There was also a recommendation that valproate should not be used as a first choice drug for treatment of both epilepsy and bipolar disorder in young women. And there was a quite extensive risk communication to all relevant healthcare providers in Europe and to the patients in the form of dear doctor letters, but also implementation of so-called educational materials, which provided some additional information and called for additional attention from the relevant specialists. So this was in 2014. And following implementation of these additional measures after the referral in 2014, we continued to receive information from different sources that despite of these implemented measures, there are still pregnancies occurring in women who are using valproate. Information provided on the slide shows information from United Kingdom specifically, but it is quite representative what we have seen also in a number of other EU countries after the first referral in 2014. So some research that was undertaken by national competent authorities at that time in UK showed that up to 70% of women who are using valproate, they mentioned that they didn't receive any materials, any educational materials from their healthcare providers when valproate was prescribed to them. And only in UK, there were 400 pregnancies identified. It could be that the number was higher, but at least this number was detected in women who are taking valproate. And of course, if we extrapolate that to the whole EU, the number is much, much higher. And it's also striking that as late time and Jure published on their webpage that it's the scale of the harm is bigger than the safety issues expected with thalidomide. And despite of quite extensive risk minimization measures implemented, for some reason, women using valproate still experience unwanted pregnancies. So this was the reason that just 3 years after the first referral, the second referral was triggered. At that time, it was triggered by France. And in France, they have also identified a number of pregnancies in women exposed to valproate during pregnancy. So there was a clear need to assess the effectiveness of measures that were implemented to 2014 to understand why these measures were not sufficient and what are the barriers in clinical practice for implementation of such measures and what could be done to improve the safe use of valproate. And so this was quite an extensive referral procedure where different type of data and stakeholders were involved. So in particular, there was a survey conducted among healthcare professionals in Europe where the knowledge on risks and risk minimization measures was evaluated, but we also had a drug utilization study conducted in a number of different member states, which looked at the actual prescribing behavior. And this study showed that contraception use was not optimal in women using valproate. So this could be one of the reasons explaining why there are still pregnancies taking place. What was also very interesting that during this referral in the European Union, we had for the first time public hearing. And public hearing is, well, a procedure which was made possible with the new pharmacovigilance legislation in EU. Basically, it's a meeting where which is being announced in advance and which makes possible that anyone living in European Union, any patient, so you don't need to be working a certain organization. So really anyone could participate at this meeting and could provide an intervention, sharing their own experience, for instance, as a patient with regard to use of valproate and provide their opinion how their experience could be used in improving use of valproate in clinical practice. And of course, during this public hearing, besides patients, there were also a number of different healthcare providers who voluntarily came to this meeting and shared their experiences and their opinions. So there was a live broadcast, and I believe that it is still possible to find the whole broadcast on the webpage of EMA. For me, it was just individual opinion. It was very useful to hear the stories provided by different women who used valproate during pregnancies. I still can very well remember a woman with epilepsy who used valproate and who got pregnant, and her child was suffering from very severe neurodevelopmental disorders, and she was just sharing how a daily life looks like for a family who has such a child. What does it mean that even she can't have a usual babysitter? She needs to have a specialized caregivers who have understanding in managing this type of serious behavioral issues, and how it can impact the working life and the family life. And she highlighted that her doctor didn't tell her that valproate may result in serious health problems in in a child during pregnancy, and well, she was really advocating that information should be very well provided to the patients, so the patients can make informed choices with regard to having children. And on the contrary, there was another woman with diagnosis of bipolar disorder who didn't have a child, but she did have a wish to have a child all her life. And she was repeatedly told by her psychiatrist that, well, if she's using valproate, then having a child is a big risk, and well, she has chosen not to have children. But she was also, her opinion was that there are many other different products, especially for treatment of bipolar disorder. And there was a possibility, for instance, to consider an alternative treatment option for her, at least for some period of time, when she could become pregnant and safely have a child, and then she could be switched back to valproate. So, well, these type of examples, there were many other different type of interventions made, which really helped Prague to also take this on board when evaluating the data and making updated recommendations. Yeah, so I have talked already about these public hearings. That was the question that was sent in advance to anyone who wanted to participate at this meeting. What are your experiences with valproate and the existing risk minimization measures? And they were also invited to provide any opinion. What do the patients or people from public think how these measures can be further improved? So, well, I will not provide all the details of the review of all available data, which we undertook during assessment of this referral, but one of the important aspects was the medical need of valproate for epilepsy and bipolar disorders. It turned out that for bipolar disorders, there is no patient population, there is no specific type of bipolar disorder that may not be treated with another agent than valproate. So that means that for treatment of bipolar disorder, in principle, there is absolutely no need to prescribe valproate during pregnancy. It will be always possible to discontinue valproate to switch to another agent. But for epilepsy, it was different. We had also several expert group discussions where we invited neurologists, psychiatrists, GPs who had extensive experience in using of valproate. And it became very clear, but also based on the published literature, that there is a subtype of epilepsy, generalized genetic disorder epilepsy, where patients may not respond to any other agent than valproate. So it seemed that there is a small group of patients, but it existed, who would respond only to valproate. And if such a woman with this type of epilepsy would become pregnant, it would be impossible to switch her to another medicinal product, and it would be very, very dangerous for both the mother and the child to do such switching during pregnancy, which would mean that for this type of patient, using valproate would be necessary during pregnancy. So all efforts should be then made to prevent such pregnancy in such patients. But well, it was very interesting, also a little bit of ethical discussion. What if such woman, despite of being very well aware of all the risks of valproate, she still wants to conceive? And it was also acknowledged that there is also patient right to do so, but the most important consideration is that the patient should be fully aware of all the risks, and only then such decision may be taken. And that resulted in an update of product information where we have contraindicated use of valproate during pregnancy for bipolar disorder. But for epilepsy, there was a little opening, although a general contraindication was implemented also for epilepsy. There is a mention in the SMPC that it should be contraindicated unless there are no alternative treatment options. So this would then allow neurologists to go on with valproate if such pregnancy occurred in patients who do not have other treatment options available. Yeah, so So taking into account information from all the sources, the product information has been strengthened, and we have implemented so-called pregnancy prevention program, which had many different conditions. And here you can see on the left side, these are the a summary of the measures that were implemented using the update of product information, SMPC, and also some other additional risk minimization measures. The patient leaflet has been updated to provide careful information on all the risks, but also on the need to use contraception. And we have updated all existing educational materials for both healthcare providers and the patients. And there was a decision that a warning should be implemented on the outer packaging. So, so every time when a woman would receive her medicine from pharmacy, this warning on the outer package would serve as sort of reminder about the risks in relation to pregnancy. And well, it may not be necessary for all teratogenic products, but for a product such as valproate, it was considered important because valproate is a product that is being used chronically and sometimes lifelong. And sometimes it's being prescribed in young girl who is not who cannot have a child yet, and at that time it's not a big issue for her or for her parents. But then time goes on, she reaches the age of menarche, and then she may have a partner, she may have a wish to become a mother, and it is possible that the information about teratogenicity that was received many years ago when the product was prescribed could be forgotten. And well, 10 years or 20 years later, then still there is a risk to become pregnant while using valproate. So in such case, it's really important that there are reminders, that there are different measures that would make sure that there is a periodic discussion with the woman. So outer packaging warning is one of the tools that we have implemented, but on the right side, you can see that we have also requested that any woman who is receiving valproate, any woman of childbearing potential who is receiving valproate in European Union should have at least discussion with her specialist at least once a year, where it would be checked if she wants to become mother, if there is a child wish, and if so, then the specialist should make all efforts to try to switch her to another alternative treatment option, and guide her through that process by gradual discontinuation of valproate and introduction of another safer treatment, safer treatment, for instance, other safer anti-epileptic agents, and on the land pregnancy may be planned. We also try to define better what is the role of different healthcare providers, and we developed specific educational materials, for instance, for gynecologists who had a different material which was focusing on a type of contraception that is advised for women who are using valproate. The role of general practitioners was different. For instance, general practitioners should play an active role in referring the woman to a specialist on annual basis. And also in some countries, it was decided that there is a need for more frequent distribution to this target group. And here on this slide, you can see how the outer packaging looked in European Union. So we decided that there is a patient card necessary, which patients can carry with them all the time. And from experience, we know that patient cards are getting closed, or pharmacies run out of them, and then they do not distribute such cards to the patients every time when the product is dispensed. So a very interesting solution was to attach such a card to the packaging. So every time the product is dispensed this way, when it's attached to the packaging, then there is a sort of guarantee that the card would be received by the patients each time. And then this is the warning on the outer packaging that was implemented for all valproate containing products in European Union. On the right side, you see a textual warning. And on the left side, you see an example of this textual warning complemented with a pictogram. So the circle strike through with an image of a pregnant woman. So this was the preferred option for Prague. However, several countries said that for well, due to national policies, it's not possible to implement such a pictogram. So there was a need for some flexibility on this European level. So we decided that the textual warning should be implemented in all countries, and in countries where it is possible, it should be supplemented with the pictogram. And in the end, I know that most of the countries included both. So summarizing the lessons that we have learned from these referrals, is that input from different stakeholders reflecting clinical practice was very important, and also combination of various type of data was on one side complementary, but on the other side also confirmatory. And it's also became very clear for when one would need to have very detailed and elaborated risk minimization measures to be implemented in EU, there is a need for certain flexibility, because unless like the situation in United States, in EU, well, there are many different countries with different type of healthcare systems and different national legislative systems, and not all measures are legally possible. So there is a need for flexibility there. And also what is important, what we are still dealing with is, it is very important in such cases to identify what are the barriers of implementation of certain measures in clinical practice, because we can have the best set of risk minimization measures from both marketing authorization holder side and from the regulator side. So we can have a perfect SMPC and product information and perfect educational materials. But if for some reasons the specialists do not follow these measures, do not follow these recommendations, then our efforts may not be so fruitful. And it seems that there are a number of barriers which we are currently trying to identify to further improve the measures. And my last slide, just final remarks, just to highlight that spontaneous report and signals remain important source of evidence, despite of many exciting data sources coming from real-world evidence we are getting. Well, it has been also highlighted by other speakers today that collection and processing of spontaneous reports due to the large volume needs to become smarter, although it was not really part of my presentation, but this is obviously very important for everyone nowadays. And well, for Prague, well, it has been so in the last years, but there is more and more attention on transparency and communication. We try to share with the public the results of all assessments that are being undertaken by this committee. So due to the time, I will not highlight the last 2 points, but otherwise, I'm very happy to take any questions, and thank you very much for your attention.
Thank you very much, Liana. Really, actually, excellent presentation with bright example of the work of and importance of what is being done by Pharmacovigilance Risk Assessment Committee. So this really great work you are doing, and thanks for presenting this to us for a bigger picture of what is going on the level of Pharmacovigilance Risk Assessment Committee. Thanks again. Any questions from audience? Okay, so thank you again,
hi.
Liana. Yes.
Yeah, hi,
okay.
Liana. I have a question. This is Arshia from India. So I have a question. So it was, first of all, it was very enlightening to see the presentation from Liana, and the example that you shared from about valproate was also very, I would say, inspirational as to how EU and PRAC have handled the risk minimization measures regarding this drug. And it's also an example for us in India to follow steps with this. So my question was, So as you shared that there were the risk minimization measures that were taken, the communication was changed, and all the steps that you talked about. Has there been an assessment now after, I think it was in 2018 that these measures were taken? So has there been an assessment on the impact of these measures and how it has helped in reducing the congenital or deformations of abnormalities in patients receiving valproate?
Yeah, thank you very much for this question. Yes, indeed, following implementation of the updated measures in 2018, we have requested all marketing authorization holders in Europe for valproate to collaborate, and it was a very actually efficient collaboration. It doesn't happen a lot, that more than 18 companies collaborate together, but they had to conduct 5 different studies, and 3 of these studies were focusing specifically on evaluation of effectiveness of the newly implemented risk minimization measures. And we had a survey of healthcare providers. We had a drug utilization study to look at the actual prescribing behavior, and especially at the use of contraception following implementation of these measures. So the drug utilization study is still ongoing, and we do not have the final results yet. We will have it next year, but the survey, which was evaluating the knowledge of psychiatrists and neurologists about this updated risk minimization measure, showed very striking results, which showed that they do have a very good knowledge, but despite of this good knowledge, that they mentioned that they would still prescribe, for instance, valproate to women of childbearing potential, even if she doesn't use contraception. If, for instance, if the woman would say that she's sexually not active. Well, according to the product information and to the pregnancy prevention plan we have implemented, it doesn't matter if the woman self-reports that she's not sexually active. There is a need to prescribe efficient contraception. But in clinical practice, obviously, doctors have very good relationship with these patients who come to them for years. And it's possible that in some cases, they may trust the patient and decide that there is no need to prescribe contraception before valproate is used. And that was the reason that we requested the companies to come up with a new qualitative study. It's also very interesting. We do not use a lot of qualitative studies in pharmacovigilance in PRAC to evaluate the effectiveness, but this is the first time we are doing so, because now the question at the hand is, what are the really reasons, what are the barriers that the specialists decide to deviate and not to comply with the risk minimization measures. Well, I hope that this is a little bit providing information or answer to your question, but we do not have yet information on the harder outcomes in terms of number of pregnancies, if it really decreases following implementation of these measures, and how it is going with the number of adverse events reported in children. This data will come later next year. Thank you.
All right. Yeah, thank you so much. It really answers my question. And I really agree that, somewhere the implementation or the absorption of the communication that has been sent to the clinicians, this is where, we get lacking. And because of that, the prescriptions still keep happening. And I think if on a global level, there can be efforts to improve on this perspective, it can really help, as in the pharmacovigilance gets completely integrated with the clinical practice. Thank you.
Okay, thank you very much. Thank you, Liana. Let's move forward and go to the next presentation. Thanks again. Okay. So the next presenter is Dr. Jose Batista Ortega, America's Region Chairman, Global Pharmacovigilance Society in Mexico. So topic of his presentation will be benefit risk assessment in COVID-19 vaccines, lessons learned and challenges. So he's pharmacist, his experience is mainly related to pharmacovigilance field, SARS, PSUR, PWAR, signal management, risk management and risk minimization, audit inspections, legislation, vendor and CRO management, interaction with independent PV organizations, regulatory agencies, as well as pharmaceutical chambers. As part of his PV career, he worked at GlaxoSmithKline, Merck, and Pfizer, founder of the Global Pharmacovigilance Society, chairman of the Americas region and LATAM ambassador as well. In addition, he is also a member of the International Society of Pharmacovigilance, founder of GBA Pharmacovigilance NT, aim to build innovative scenarios to learn, share, apply, and build knowledge in pharmacovigilance across the LATAM region. Former president of the Mexican Drug Society, Safety Society, collaborator of the DIA Global Forum newsletter. So giving the floor to you, Jose, to present your talk today.
Thank you so much for this invitation. It's my pleasure to be here. Good morning, good afternoon, good evening for everyone. So let me start to share my screen, and if you please kindly let me know if you are able to see my presentation.
Actually, we can see your desktop. If you can share. Yes, we can see now.
Yeah.
Thanks.
Okay. Yeah, well, once again, thanks for this invitation, and congratulations to the organizer committee for this amazing event. And well, the topic that I will share with you, it's related with the benefit risk assessment of the COVID-19 vaccines, thinking or having the approach related with the lessons learned and the challenges. And that could be also very, I could say, challenging for the PV people, because when we talk about safety information, well, we mainly think or in just safety data, adverse event, but the process is more complex when we need to assess if the safety information that we are collected is considered not just acceptable, is considered that is not outweighing the benefit of the drug. So that's why this topic maybe could be very useful for the purposes of this event. First of all, I would like to express and to share with you that all the info that I am sharing now, it's mainly trying to have a educational purposes, and for sure only represent my point of views, my positions, my ideas about this topic. When we think and when we talk in medicines, and this means, you know, small molecules or vaccines, for sure, as my previous colleagues already shared with us, we should think in some of the scenarios that really those medicines are not helping us to really try to see how they are providing benefits to the people. There are different harms that these medicines could cause to the patients. But in the other side, there are things, there are disease, there are hopes that the medicines could bring to us. For example, the resolution of some disease that were affecting our children population, or for example, recently the resolution or giving us one important weapon to face one of the, you know, most important pandemics that we are facing recently. So how we are able to put in a context all of these harms, all of these risks that the medicines could cause to the patients in context of the benefits. How we are able to do this? For sure, most of those we could think in the vigilance of the medicines, in the vigilance of the drugs, especially in the pharmacovigilance. And for us, it's very common when we think in pharmacovigilance to also think in these three important process or three important stage of the pharmacovigilance. But the process that really help us to evaluate, to assess if the benefits are outweighing the risk of a medicine, is the benefit risk assessment. And through this process, we are able to conduct an evaluation of the demonstrated benefits and risks of a medicinal products. And for sure, to be able to conduct this process, we need to make some judgment as to whether the expected benefits outweigh the potential risks associated with the expected use of the medicines. And to do this, as you can imagine, we have several of different types of methods. There, we can use qualitative methods or also quantitative methods. And these are some of the methods that are currently used around the world, or that are currently using around the world to assess the risk benefit of the medicines. So as you can see, the alternatives are really huge, and we can use each of these methods depending on the info what we have. But when I say methods, it's not because I am just trying to give you some general information. When I say methods, it's because to have a method for the conduction of the assessment of the risk benefit, it's very important. Why? Because if we have a method, we can increase our transparency, because this facilitates the identification or that supports the decisions that we are doing in terms of this process. The other thing is that this having a method for the conduction of the risk-benefit assessment increases also the consistency. Why? Because different or the same evaluator is more likely to arrive at the same decision and giving the same data and value judgment. And finally, for sure, we also add value to the robustness, because less risk or forgetting or missing key steps or elements, knowledge gap are revealed. So that's why it's very important to have a method in place to really be able to conduct in a systematic way our assessment of the risk benefit of the medicines. So here is one of the examples that we can use just to try to elaborate a little bit more about how a risk-benefit method could be implemented. I am sharing the method that is used by the FDA. This is a qualitative method, and as you can see, jumping in five different types of info, they are able to conduct this benefit risk assessment. They consider the analysis of the condition. This means mainly the disease, the current treatment options, the benefit of this new drug, or the benefits of the drug that is under assessment, and for sure the risk and the risk management of the medicine that is under evaluation. And at the end, they are able to compilate all of this information to declare a conclusion about the benefit risk assessment. This is the way that the FDA suggests to conduct this assessment. As you can see, they tabulate all of this information on each of these aspects, but having in consideration two important conditions. First of all, we need to declare for each of these aspects what is the data available for each of these. For example, what is the data related with the benefit of this of the medicines? What is the data available? But also, what are the uncertainties? And when we think in this concept, most of us maybe we can think in lack of information. But yes, this is true. But it's not all the aspects that we need to consider as part of this type of assessment, because uncertainties also are related with contradictory information or with marginal information. And for each of these aspects, we need to elaborate a conclusion and to declare our reasons about these conclusions. So let's start to see what is the data generated and from and what are the sources that we have in terms of the safety information and the assessment of the risk benefit of the COVID-19 vaccines. So let's start thinking in the clinical trials. Let's see some figures around the most used vaccines around the world. And this means the Pfizer and BioNTech COVID-19 vaccines, Moderna vaccines, AstraZeneca, and Johnson & Johnson vaccines. Thinking in the population that was part or that took part of the clinical development, the early clinical development, I could say the phase 1 of this clinical development that is mainly related with adults. For example, thinking in these four vaccines, for the Pfizer and BioNTech vaccines, well, they collect information around 20,000 patients. In the case of Moderna, they collect information around 30,000 patients. For the AstraZeneca, it's almost pretty the same like in Moderna, and for Janssen, they collect information around 21,000 patients. So what we obtain in terms of this clinical development, at least considering this, I could say this stage 1 mainly related with adult populations. Basically, they collect information very similar in terms of what we expected, thinking in the reactogenicity. Most of the information collected were related with the effects associated with reactogenicity. And this info, well, for sure, was also related with symptoms at local level and also symptoms at systematic level. As you can see, I am sharing with you what is declared in both labels, one for the European Union and the other one for USA. And we can see how is consistent this info. Thinking in the other type of vaccines, those that are considered a viral vector, it's almost the same. Most of the information collected, the most frequent information collected is related with these events associated with the reactogenicity of the vaccines. Pain in the site injection, headache, fatigue, most of the events that we expected to collect as part of the development program, according to our previous knowledge for vaccines. But this was only the starting point of the collection of safety data, because after this amazing and incredible development of these COVID-19 vaccines, we started to generate real data. And for example, considering the population exposure in clinical development, and now talking about the population exposure in the real life, the numbers are really different, as you can imagine, and maybe most of you already know. Now we are really close to reach the 11 billion doses applied in more than 180 countries. And because and you hear maybe that I am based in Mexico, I am also providing some figures about how the Latin American region is contributing with these doses applied around the world in terms of COVID-19 vaccines. We are apporting as region, well, we are giving 10% of the worldwide doses applied for COVID-19. And these are the countries that also considering just data related with the Latin region, these countries are also covering the 80% of the total doses administered in the region. This is for my region, but let's see some figures about what is happening, for example, in the European Union. This information is considering a data log point of the 30th of January, and as you can see, well, the total number of doses applied, it's really big. This means that, well, in considering these 4 types of vaccines, they were able to administer almost 797 millions of doses. So what happened with the information, with the safety information that they collect from these figures? It seems that at least almost 1 million of suspected side effects were collected. And in proportion, just making some simple estimation, well, at least for each of the, for the doses applied in the European Union, well, 0.13% of the doses applied, well, were related or associated with a report also or a suspected side effect. But it's important to consider that as declared in the EMA side, that these cases, these reported cases, are not necessarily related or caused by the vaccines. So this is important to consider because at the end, sorry, could you please mute your microphone? Thanks. So this is very important to consider because basically what the health authorities are doing as the EMA is to share with the people, with the population, what is the safety data collected. But this is the primary safety data, because I'm as Liana also mentioned, then after this first process to get the information, there are several processes that are conducting under the health authority side in order to assess this information. So what happened in the Latin region in trying to get some idea about the number of doses applied, the available vaccines, because here in the Latin region, the scenario is a little bit different in terms of the availability of vaccines, because as you remember, for the US, they have 3 vaccines. In the European Union, there are 4 vaccines. And for example, thinking in Argentina, in Argentina, we have available 6 COVID-19 vaccines. Then in blue, we could see the number of doses applied in mainly the main, I could say, data log points are related with the end of 2021 year. And then in purple, we can see the number of suspected adverse event reports. And finally, we can see the proportion of cases that were considered severe or serious cases. Here we have figures for 7 countries that make available to the population the numbers about COVID-19 vaccines, adverse event collected, and also for sure the category or the proportion of the adverse event considering the classification of serious or non-serious cases. As you can see, well, just Argentina or, for example, Mexico have more COVID-19 vaccines available in comparison with the rest of the world. This means the European Union and also the United States. So let's see what is the information collected after this use of the vaccines in the real life. For example, thinking in those vaccines that are related with this technology, the ARM technology. Basically, what we collected in the European Union and in the United States is related with new information, basically related with severe allergic reactions and also with some cases of myocarditis and pericarditis. In the other hand, thinking in vaccines that are using the viral vector as the platform, well, we have this new information. For sure, reactions related with allergic conditions, coagulation disorders, capillary leak syndrome, and recently also neurological events such as this Guillain-Barré syndrome. This is the new information that we collect after the real life of the use of the vaccines. And for sure, when we think in reactogenicity events like pain at the site injection, fever, you know, pain in the muscles, maybe it's easy to us to try to understand how the benefits are outweighing the risk. But when we think in this kind of events, for sure that we could consider severe or serious events that could lead, you know, hospitalizations, life-threatening conditions, and for some of them also fatal outcomes, is not the same. It's not the same because at the end, these kind of events could lead to a fatal outcomes that are not considered, you know, that these events per individual's events are really awaiting the benefit of the vaccines. And for that purposes, what the health authorities are doing globally is sharing with the population data about the frequency of the events, because what we know very well right now is that all of these events are considered rare or very rare. And this means that, for example, if we consider an application of 10,000 doses of vaccines, the probability that I have as individual to experience one of these serious or severe adverse events is that probability that is expressing in that small red box. This is the probability that I have, that the population have, to experience this kind of adverse events, to this kind of events related with the vaccination. So this is one of the key elements that now we are using in order to transmit that even we know that this could happen with the use of these vaccines. Well, the chances that we have to experience these events are very, very rare or are rare. What happened with the information collected, for example, in the Latam region? Basically, it's the same information. There are no new relevant information in terms of safety data. Even most of the developments were not conducted in the region. Well, basically, what we collect at regional level, it's almost the same that is collected worldwide. So having this in consideration, well, now we are still facing very important, I could say, challenges in terms of the communication and the assessment of the risk benefit of the vaccines. Why? Because there is people that are still thinking that getting their shocks could mean to have this kind of adverse events, or even worse, getting the COVID-19 shot, they could also put their lives in a very, very high risk. So for me and for these PV professionals, some of the lessons learned and challenges around the risk-benefit assessment of the vaccines are related, first of all, with the importance of the spontaneous reports. Why? Because as we can see, this new data came from spontaneous processes of the pharmacovigilance, because most of these new adverse events now confirmed that are related with the use of this COVID-19 came from the spontaneous systems. That's why we don't we should not subestimate the importance and the participation of the spontaneous data, the passive pharmacovigilance, because through this process, we were able to identify something that we knew that we didn't expect to be related with the use of these vaccines. Also, one of the I could say paradigms that could have, or that we could have, for example, at regional level, is that because the studies were not conducted in my country, I don't know how the safety profile could be in my population, in the population at local level. As we can see, with this massive application of COVID-19 vaccines with a different technologies and platform, the truth, the official information posted by the health authorities show us that there is no new relevant information at local or regional level. And the other thing that I would like to emphasize is that the reported cases are not necessarily related cases, even signal or safety signals. Most of the people could be considered that a signal should be also interpreted and as another confirming harm that the vaccines could provoke or could generate in their health. This is not true, because as we already know, signals are related with safety information that should be confirmed, that should be put under the signal management in order to really confirm if the signal is true and how this signal is impacting the risk-benefit balance. And finally, maybe in line with the previous presenter, well, I could also emphasize that race of reports and traditional labeling are not enough to communicate risk. Because at the end, when we think in risk communication, we need to think in what the HCPs or the patients needs to receive, but not just from my perspective as PV professional, because we need to listen then. We need to be on front about the pros and the cons of the use of the COVID-19 vaccine for sure. And at the end, I truly believe that we need to give people the information that they need in a way that helps them to make a decisions. So thank you so much for your attention. These are the reference of the info that I shared with you, and it has been a big pleasure for me to take part of this important event. Thanks a lot.
Thank you very much, Jose. Very impressive. I always will remember the box you just shown with the probability of getting adverse events, the whole picture. This was really very impressive. I would ask audience if they will have questions, please. Okay, please, Liana.
Yeah, thank you very much for this interesting presentation. I have a question about the way you have dealt with processing of spontaneous reports. Well, I can imagine that also in your region, the number of spontaneous reports was very, very high, or at least higher than what we normally have for other medicinal products. In EU, for instance, many countries had quite some challenges in timely processing of these reports. And I just want to share that one of the lessons learned for us was that it would have been much, much more useful if beforehand we could think of a process to have a designated field in ICSRs to highlight if the report is related to the first dose or to the second dose, because when we were doing analysis, in order to find out if the adverse event was reported after the first or second dose, so the review would need to be done, either manually or using a search of the text. But I'm wondering what type of challenges you had and how you have overcome those. Thank you.
Yeah, well, thanks for your question, Liana, and I will speak in terms of a PV professional. I'm not part of the health authority side, but what we saw in terms of what the health authorities are doing to face this important challenge is firstly to put available to the population different ways to communicate safety info. Because previous to the COVID-19 pandemic, basically what we have around the Latin region are systems thinking in healthcare professionals and in the pharmaceutical industry. And this means that in the past we need to fulfill or to fill out, complex formularies in terms of safety info. Now, what we have are different approaches, different ways to stay connected with the population. And for some of the countries, for example, in the pages that are related with the vaccination campaigns, not in the health authority website. I am talking about in the website created for the vaccination campaign. The health authorities added a simple or a small formulary for the population that wants to report an adverse event. The lesson learned now is that for sure we need to collect information about the experiences of the patients using or receiving these shots. But one of the things that we discovered ahead this application of the COVID-19 vaccines is that we need to separate, or we need to provide information, or we need to provide a way to the patients or to the reporters to communicate the info in terms of the dose that they are receiving. As you mentioned, it's different to report, at least for the PV purposes, it's different to get information about first dose or second dose, or also booster dose. And to be honest, in the region, we have just 7 countries that put available information for the population. Of these 7 countries, just 2 of them also collect information in this detail, because they provide info about first dose, second dose, and also booster dose. So I believe that this challenge is, I could say, in a evolving state, but we are not, at this time, we don't have a final resolution of this important challenge.
Thank you, Jose. Any other questions from audience? Okay, thanks again. And let's go forward, and I will invite the next speaker for the talk. Okay, Dr. Tigran Uzunyan, Program Coordinator, Canada, Topic Clinical Pharmacology, Aspects of Drug Safety. Tigran is a physician, a clinical pharmacologist, studied and gained his professional and scientific designation in the Yerevan State Medical University, Armenia. Tigran has 20 years of diversified experience in the academic, pharmaceutical industry, and clinical research of drug development in various jurisdictions and phases. From planning and designing to administration and monitoring of clinical trial, phase 1 bioequivalence, pharmacokinetic, first to human and human abuse liability studies, as well as phase 2 to 4 clinical projects. He has been working at various American and Canadian clinics and research institutions as a physician, a research associate, clinical research coordinator, a research scientist, study monitor, and manage different research projects. Research interests include antihypertensive, antibiotics, psychotropic drugs, medical cannabis products, and many new agents in early and late phases of development. He is an experienced medical and scientific writer, scientific articles, research grants, reports, lectures, protocols, SOPs, etc. Reviewer and editorial board member in several scientific journals. Currently, Tigran is coordinating in clinical research, drug safety, and pharmacovigilance diploma program, teaching, coaching, as well as developing and updating the content of educational materials and modules for Canada, for US classes in the Applied Academy of Pharmaceutical Sciences, Toronto, Canada. Thank you very much for joining us, Tigran. Welcome to our webinar, and we are very glad to listen to you. Thank you.
Welcome everybody. I'm really also very thrilled to join this very nice webinar. This is my first experience actually presenting in the webinar, and I'm very excited to share my humble knowledge of clinical pharmacology. I'm feeling a little bit awkward here because I'm the pre-approval guy and maybe very far away from the approval, and my presentation would be a little bit not that much related to the pharmacovigilance, but I will talk about the clinical pharmacology methods and how the clinical pharmacologists involved in the drug development and what we can say in terms of the drug safety investigation. So if you don't mind, I can start sharing my screen.
Yes, sure.
Okay. Here it is. Okay. Couple words about the clinical pharmacology. I always am being asked what is the clinical pharmacology, and the answer is very simple. The specialty, the goals, what we do are just in the name, in the title of our specialty. Clinical means that we are dealing with the patients, right? Anywhere where the drug, the pharmaceutical product, or any medicine may be related to the human is in the jurisdiction of the clinical pharmacology. So we can say the clinical pharmacologists are. Pharmacology is a science discipline, scientific discipline in the medicine, which is like dealing with the drugs and their clinical use. It kind of connect that gap between the medical practice and science. The ultimate goal of the clinical pharmacologist is to find the right drug in the right dose. When we say dose, means the strengths, regimen for the right patient and the right time. So that's what we're doing, and what all our efforts are implemented. Objectives, of course, quite long. We can talk a lot about that, but I just identified three main of them. That's the first one, of course, is rational use of the medicines, no matter what kind of medicines there are, both for the individual patients, right, to find the best ultimate goal of the individual, for the individual patient, and for the special groups of the population, maximizing the efficacy and minimizing the safety concerns. The second main objective is research, right? And it's always translational, means that we're always trying to translate new scientific data of drugs into the patient care. And the third, teaching, training, we're always involved in the improving the qualifications of any healthcare professional who somehow related to the drug administration. No, this slide represents the story of drug development. I think that you all know this very well, how from the initial thousands of compounds identified, then tested for in terms of the screened for the target binding and other specific methods, right, pass through the discovery stage and enter into the drug development, which is divided on the preclinical, where we doing the toxicity tests, a lot of safety pharmacology tests on mainly on the animals, in vitro, in vivo, in experiments. Identify main PK profiles, and then the best drug candidates are entering into the long clinical phase of the development, divided on 3 main phases until it reached the regulator, and if everything goes well, it receives the approval. So, of course, in the post-approval, the science continued with our clinical pharmacologist participation, maybe not that much, but the data from the post-approval, of course, are very important for validation of those methods which we are using in the drug development. This slide shows you where the clinical pharmacologists are involved throughout the whole line of the drug development. For us, just the main principle, if we're talking about the drug safety, for clinical pharmacologists, the drug safety concern arises from the very beginning. For us, any unfavorable drug properties in the early phase of the development predispose the future drug safety and, of course, the efficacy concerns as well. So sometimes we are not involved in the direct safety problems, but what we're seeing in the early phases of the drug development, of course, will be reflected in the future efficacy and safety. So that's why in the very early stages of the development, we are trying to choose only those best drug candidates which will have more chances to receive the approval and be more successful and safe for the post-approval population. Here represented the main studies. Liana asked me to open some acronyms, and I will try to do my best to, I know that maybe audience not very advanced in the research, clinical research, early clinical research, but so we do the first in human studies after the finishing the preclinical, although our involvement in the preclinical also existing. First in human studies, we're doing the so-called single ascending dose and multiple ascending dose studies. It means that we are doing on mainly healthy volunteers, trying to test the different doses very early first time and escalate, stepwise escalation, see mainly safety problems, exclude any safety problems. Then, of course, we investigate the ADME properties, right? Absorption, distribution, metabolism, excretion. Of course, if we notice some concerns for the future specific population, or if the drug developed for that purposes, of course, we're conducting appropriate studies in the specific groups of the patients already in the future steps of the clinical development. A lot of drug-drug interactions, population PK studies. This is already a new world in the drug development, comparably new, of course, about 20, 25 years old. We use a lot of biomarkers in for different purposes, surrogate, safety, efficacy. The biomarkers are involved in our new models of the drug development, which I will describe a little bit later on. Pharmacogenetic studies. As I said, if needed, we're investigating the drug cardiac safety, so-called TQT studies, exposure response effects, how the concentration of the drug is reflected in the efficacy, the tolerable doses, minimal tolerable dose, a lot of BE studies, BA studies, so pharmacometric investigations. Also, clinical pharmacologists are involved. We're doing these studies either in the first stages or where we are developing the generic medications. Food effect, in vitro bioanalytical methods development, and of course investigation of the biological products, specific safety maybe and efficacy related studies. So this is the whole spectrum of studies, methods where clinical pharmacologists, of course, participating in the drug development. I won't talk too much about this slide because I'm pretty sure that many of you know what it is. This is a classic concentration over the time curve of the drug after oral administration, right? We see that right after the administration, the drug absorbed and accumulated in the blood, where we collecting the and measuring the concentration after reaching the Cmax. Then it goes down, and then we identifying so-called the main PK parameter, Cmax, Tmax, the time when the concentration reached the maximum. A very important parameter is the so-called area under the curve of this concentration. It shows the exposure of the drug. And just for your for the audience, I also would like to show the so-called therapeutic window, which is the difference between the minimal effective concentration and multiple maximum tolerable concentration. This after which we are experiencing unacceptable rise of the adverse events. So this is the limit where we are focusing for our future patients and population. Of course, for the drug approval purposes, we are interested for the drugs with the broad therapeutic window. If the window is narrow, we call them narrow therapeutic index drugs. And of course, you may expect that today regulatory agencies would not even expect that kind of drugs for the approval. We only are focusing on the broad spectrum therapeutic, broad windows therapeutic products. So now let me just show you what are those desirable clinical pharmacologist properties of the drug, which we are interested in for the future drug development, right? So as I said, if we have some problems to fulfill our expectations of the drug development in the future, in the late phase of drug development, and especially in the post-approval phase, we may have a lot of problems with the safety and efficacy. So that's why we want our drugs to be, first of all, very highly absorbable, right? So this is a main condition for drug developers. No, of course, we may have drugs with no oral administration, but the oral administration is always desirable method of administration. It is well controlled, it is expectable for the safety checks, and of course cheap and comfortable method of the drug administration. We want that the future drug exhibit the linear PK. This is a little bit maybe specific term. It means that the changes of the concentration in the blood are dose dependable, right? So it means that the PK is expectable for the physician, for the scientists. We don't want those fluctuations and non-dose related with other covariates effects, right? So we want the PK to be linear as much as possible. Although, of course, it's a big luxury for the drugs to be always linear, to have linear pharmacokinetics. AUC, Cmax, not significantly affected by the food, pH altering medicines, grapefruit alcohol, I think that you know that very well. And of course, the best choice, of course, the BCS class 1. This is the FDA classification of the drug, which shows how much the future drug candidate has high solubility and permeability. So those are the most important aspects of the drug absorption. So to be absorbed in the stomach and in the intestine, first the drug has to be solubilized, right? If the drug is not solubilized in the water, that's what we are interested in. It will not be able to absorb, and the bioavailability will be very low. And of course, the permeability. So the first class is those are who have high solubility and permeability. No, of course, we may have class 2, class 3, where the solubility is lower or permeability, but of course, these are the so-called desirable properties. In terms of the distribution, so distribution is where the drug, after being absorbed, reached the target. Or we want that process reached immediately and without any effective and at effective non-toxic concentration, never accumulated in the target organs, which may cause a lot of safety problems, right? And not significantly bound to the plasma proteins. It's also very important because we know that drugs are in the blood, and in the compartmental, in the organs, tissues. The drugs are bound to the blood proteins, and that's how the main storage of the drug in the body. And the effect of the drug and metabolism, excretion, always happening only for the free portion. So whatever we expect from the drug will happen only when the drug is free. So that's why we don't want the drug to be bound too much with the plasma proteins, because any fluctuations of the plasma protein or any interaction of other drugs which may replace the drug from the plasma protein bound may significantly change the effective concentration and create the safety concerns. The best example is the warfarin, the drug which has a lot of concerns in terms of the clinical pharmacology, but still used, and because it is sometimes we found, of course, something which maybe replaced the warfarin, but it's still a very cost-effective medication. So that's why we continue to conduct the clinical monitoring for when we administer the warfarin. But for the clinical pharmacology, the drug like warfarin, it's like a disaster. We try to, when we're creating the drugs, we're trying to avoid any warfarin-related PK drugs. In terms of the metabolism, excretion, so we are interested to have the drugs which will not be extensively or only or exclusively metabolized by the CIP enzymes, because as you may expect that drugs are always either substrates or perpetrators, inhibitors, or inducers of other CIP enzymes. So if we have at least two drugs administered, so they may both each other may change the metabolism of each other. So, and that's of course again may become a problem for the drug safety. We don't want the drug to be metabolized by most polymorphic enzymes. Those are 2D6 and 2C19, because it's yeah, of course we don't want, but a lot of drugs, especially the drugs from the psychotropic family, antidepressants, a lot of other drugs are metabolized here. So that's why we have so many different problems for the low and slow and ultra rapid metabolizers. So we don't want this, but we still are working on it. Clearance, not very highly variable depending on the other covarieties, right? So the age, race, and gender, right? So we want to be stable, not very much related, especially talking about the age-related problems, right? And clearance, not time dependent, so stable. And also other properties, not to be narrow therapeutic index, I think it was clear, have acceptable QT interval. If we have some suspicion that the QT interval is prolonged, especially when we see that it is prolonged more than 10 milliseconds, so we need to conduct the additional safety TQT studies to rule out any future possibility of torsade points, and those very dangerous ventricular tachycardias may arise from these type of products. Not a significant inhibitor inducer of the CYP3A enzymes, because they are the most, those enzymes which are metabolizing most of the drugs, and of course the P glycoproteins, which are the main transporters of the drug. Also, we don't want this to be very much related, because a lot of drugs may cause another drug-drug interactions in these aspects as well. Okay, so these are the main properties of the drug which we would like to have during the drug development. So what is the current state of the pharmaceutical R&D? What we have now? We have a very intensively rising drug development, although either for the pharma products or biotechnological products. But of course, we are stuck with a significant problem of the pharmaceutical industry, the price of the medication. You see that if like 40 years ago, 50 years ago, we spent about hundreds, 200 millions to make one drug, take one drug to the market. Today, it is the price of drug approval for one single medicine is more than $2.6 billion. So it's because the very significant increase clinical trial complexity, because the clinical endpoints becoming more stronger. The regulatory agencies are not satisfied with the very, very weak clinical endpoints. They want us to make drugs which will extend the life of the patients, have better clinical endpoints. For that purpose, we need to organize more complex clinical trials with a better, bigger sample size. That, of course, significantly increases the cost of the drug development and consequently the price of the drugs. So that's why this is a very big problem. And with all this, unfortunately, the failures of the drug approval, of the drug development, right, on every stage are still quite significant, right? So just imagine you spending wasting billions of dollars. The product reached to the late phase development, and it's not even progressing to the drug approval because of the unacceptable safety profiles. So this is what we have with the conventional so-called traditional drug development regular pathway, right, which I already described. So to solve this problem, yeah, and a couple of words, yeah, so as I said, still the level of the attrition of the clinical projects is quite high. It requires a huge amount of money for the problem solving. Very few drugs are making out to the clinical research pipeline, and many drugs are withdrawn even after the market approval because of the toxicities, right? So we have some problems in the early phases. We still may not investigate everything and produce the very nice drugs which would not have that kind of post-approval toxicities. You see the main reasons of the safety-related attrition of the products in the clinical development. Well, this is a preclinical stage. The cardiovascular, CNS, and reproductive toxicity are the most common reasons. In the clinical development phases, again, cardiovascular, hepato, and CNS toxicity, immunotoxicity. Generally, all about like 2/3 or even 70% of the drug failures in the late phase 2 and 3 are related with the efficacy and safety. Right, so we have a lot of problems with all these significantly advanced methods of the clinical pharmacology. We still have a lot of concerns of the drug, of the future toxicity. Right, so no, I think you all, as drug safety pharmacovigilance specialists, very well know how regulators are making the decision about the approval. Always the benefit should prevail the risk, and anytime after the approval, if the risk is balancing with the benefit, we always the future approval may be recalled and the problems may arise, right? So, but in the reality, right, it looks very simple. So you may prove that your drug candidate is beneficial, has less risk, so you will get the approval. The reality, but is not that clear. The problem is that it's not whatever we're submitting, whatever the sponsor submitting to the regulators in the dossiers, right? So those new drug application or new drug submission dossiers, the data are not simple and straightforward, right? So it's rarely we see the exact beneficial and exact unfavorable effects. Most of the data are have some uncertainty whether they are beneficial or we have some suspicion about them. That comes from the unfortunately validity of the extrapolation. The thing is that in the late phase of the development, or even in the early phases, for example, in the early phases, we do not enroll, for the example, elderly population, right? So the risk of safety concerns, of course, is very high. So that's why, for the safety reasons, we do not enroll them. Then we don't enroll them even in the phase 2 and the only enrollment happening in the phase 3, where we may not investigate all spectrum of the problems, or we may investigate, but the real reasons of that are unknown for us, right? So a lot of surrogate markers are involved, and they are not direct, so their variability is quite higher. So, but this is what we have, and this is what presented for the regulators to make a decision. Important sources of the bias still present in the clinical trials. Methodological deficiencies, limitations of the data set, right? So as I said, we never have enough sample size of the elderly, or not even mention pregnant female sometimes, or pediatric populations in our clinical study. That's why we cannot fully investigate them, especially in the settings of these very expensive clinical trials, right? So that's why it all mitigate the supportive information for the drug approval, and that all comes from the non-clinical and early clinical data. So we need a solution, and the solution actually came last 20-25 years ago. The first kind of proposal came from the PK population models. What it means that we are creating some mathematical models of group of people, population, what kind of specific PK properties they may have, and we use that mathematical model in the drug development. Then other data became available, so we collected very significant information about system data of different population, right? So intrinsic and extrinsic data of human body, the age, weight, cardiac output, tissue volumes, by the way, every organ tissue. So we know a lot about every different population, specific physiological and biological data. We know we have a lot of information about the drugs, right? So all the future, which will predispose their future behavior in the body, in human body. So their molecular mass, solubility, permeability, lipophilicity, the metabolism, transporters, right? So we collected throughout those 30 years of in the drug development, we know a lot how to investigate these, right? So we have all methods of this. We are processing everything about, and we know how to design the study. So what if we may combine all these data, right, into one single mechanistic model, the mathematical model, right? So we combine these three essential components into the one model, and this may become a very nice simulation prediction of PK, PD parameters, right? So PK, how the pharmacodynamic related from the PK. We call these models mechanistic, in vitro, in vivo, extrapolation linked, physiologically based PK model. So simply saying, it is a combination of mathematics, IT, and molecular biology, clinical pharmacology, pharmacometrics, we call it, where knowing all this data, we may import it in one single mathematical model and simulate the clinical trials. Instead of enrolling the patients with, especially the patients in the special population, where sometimes it's impossible to do, we may, knowing the specific, let's say, we know how the elderly Caucasian Nordic males, right? What are their specific PK and systemic data, right? So we are entering appropriate data, or Chinese population, or Asian population, right? Or American. So we may create a very nice model, and that model will be used in the simulation of the clinical trials, considering, let's say, for the elderly population, that in elderlies we have a lot of physiological changes, comorbidities, polypharmacy. These in vitro, in vivo linked physiologically based PK models may predict the impact of these variables on ADME, right? So we don't need to run the studies. If we have a very nice model, if the model is validated, we may use this model for different scenarios. The most important is that we, after validating the model, after knowing that the model is working very well, we may go further. We may create even the models of what so-called what-if models of worst case scenarios, right? We may modulate the overdosages, we may modulate the very unacceptable safety problems in the post-approval stage, right? And this is what we're doing now, by the way, and already developed throughout the last 20-25 years, and actively enrolled in the drug development. To be a little bit more specific, just let me show, it's maybe a little bit complicated, but I'll try to explain what it is. Just imagine the minimal physiologically based PK model when we are considering as human body as one single compartment, right? So just imagine the drug enters into the GI tract, then absorbed in the gastrointestinal system, to the portal vein, goes to the liver, right? So this story you know very well. In the liver, it happens the first metabolism, and then goes to the blood and enters into the body. So we are, if we consider the whole body as one single compartmental model, right? So, but we still have a lot of data to enter into the model and modulate and change it and update it. But the reality is not that simple. The reality is that our body is created by multiple compartmental models. Every organ, every tissue is receiving the appropriate specific blood flow, has different metabolizing and blood protein binding specificity, right? So we have to investigate and we have to account all these things. Unfortunately, when we're running our conventional studies, we're doing our PK profiles, we do not account this, right? We never investigate the PK in different organs. We just get the PK from the blood, and that's what we are. And then when we are extrapolating the doses on young population, main subjects of our clinical trial, to the elderly, we just extrapolating, and that extrapolation is very, very far away. So that's why if we have a mathematical model which will account every single aspect of the PK, physiological aspects of the PK, and if we know that population, how that differences may be related to the exposure, drug exposure and concentration curve, so we may have even more precise models, right? So that's why these models are always updated, always upgraded. The good point of that advantage of these models that we, if we have some problem, we may, if we see that model is not validated on the real clinical trial, right? So we have to validate, we have to check how this model works. Then we look what was the problem. We update it, we find the problem. Let's say we didn't account the enterohepatic circulation, we didn't account some other aspect. We update it, we approve it, and then improve it, and then use it for the next step of the clinical development. So the models are present everywhere now. We're using the models in every stage of the drug development, preclinical, clinical, in post-approval stages also, right? One more time, the representation of this physiologically based PK modeling. So as I said, all human systems, intrinsic, extrinsic, are enrolled in the this models, PK models, and this everything creates our for us the math model, which is used for the drug development. Okay, so if you see this is taken from the European Medical Agency conference where they were talking about the model and simulation in the drug development phases. You see, in the early phases, we have a lot of uncertainties. Then the more we go to the drug development, the less uncertainty and more confidence comes, right? And as you can see, we use the models in every stage of the drug development. So implementing this model will help us to develop the pediatric products for the pediatric population. You see that for other scientific purposes, it is very well working for all stages, throughout all stages, and even in post-approval space, we may have that implement, we may use these models. We simulate the disease, we simulate the clinical trials, instead of enrolling hundreds of population. And here, this slide shows how much we can earn by using this. Like, just imagine for the indication of thromboembolism, right? Using this model-based drug development approach, right? We may omit, right? So waive, right? Not to conduct phase 2 studies, some of phase 2A studies, early phase 2 studies, model-based dose-response relationship, adaptive phase 2B studies, right? And that what give us, the model gives us these designs. And see how much we can save. We can conduct 2,700 fewer population, and save 1 year of study duration, right? So this is significant contribution to the more cost-effective drug development. Okay, and the same examples of other indications and methods. As you can see, we have the regulatory agencies. Here we have FDA, but on the same is in European medical agencies and many other leading regulators. They started to expect involvement of the models from the very, very early, like even before 2000, right? So first scale up of the post-approval changes of the formulations, population PK guidelines is arised even before 2000. Then the more we go, the more regulators are encouraging the drug developers, the sponsors, pharmaceutical companies to enroll different methods. You see like how many guidances only related to the model-informed drug development. So this is the what we have now, and you can see that these are the cover pages of the last guidelines of EMA and FDA for the physiologically based PK models. So using this, we may overcome those challenges which we have, and the drug development may become even more accessible, more affordable, and of course, we're earning everywhere. We're earning in the efficacy and safety, and in the drug development costs. Right. This slide shows us how many projects or products were submitted for the FDA, where the PBPK models were used. Right. So if in 2014 it was just 96 models, in 2016 already like 217. Right. And the main contribution for the decision makers are coming for the drug-drug interactions and other concerns, right? So we may become more confident if we're using the models in the prediction of the future concerns of the main PK problems. That's it. That's the end. I don't know whether I exceeded the timeline, but I hope that not very much. Unfortunately, I wanted to talk much more, but because of the time constraint, I cut a lot of slides, and will be happy to answer, of course, any question. And if anybody wants, I can share these slides with the participants, of course. Thank you very much.
Thank you very much, Tigran. And I also would like to highlight that Tigran joined us from Canada, so it's early morning there. So thanks for that as well. Thank you for being with us. I have a question, Tigran. It was really impressive, this PK/PD modeling and advancement in drug development. my question a bit different. So we have two kind of overall stages: pre-marketed and post-marketed for drug. So pre-marketed, it's about non-clinical clinical data with several phases, and post-marketed, it's about post-marketing safety surveillance. So we're always thinking that overall, when drug is coming to the market and we are encountered with real-world data, it's a bit different from data during drug development phases, because in drug development, you always have restricted number of patients, restricted number of drugs, just one drug for investigation, etc., etc. So, and when comparing with real-world data, with co-medication, with other risk factors, or with a vulnerable population, children, elderly, etc., the data are a bit different, and that is the reason pharmacovigilance specialists are working while drug in the market during the whole cycle of drug marketed. So from the perspective of this PK and PD modeling, this is a very advanced technique, and this is very, as I understand, very cost effective because you are not recruiting patients, you are just simulating whatever you want to investigate and doing this all stuff. But what do you think? Will this even this gap between clinical trials and post-marketed real-world data, will this gap be even broader because you even are not performing tests on human, but you are using magical modeling? Will this gap be even how to say larger, or between real data and clinical development data?
Yeah, thanks for the question. It's really interesting, and it's always the first after you listening this PK model, the mathematical models, you becomes a little bit threatened. Like, what if in the future we will not, we will have that uncertainties even more increased? The answer is that these models do not replace the clinical trials. So far, what we have now, what I can say, they helping the clinical developers to gain the data from those specific groups for those specific situations where the clinical development is impossible or very limited. As I said, we don't have the luxury of involving a lot of females or pregnant population, or enrolling the pediatric population in our all stages of, or if we enrolling, the numbers are always scarce, right? So we are helping the clinical developers by this model, but we do not replace the drug development fully. Maybe it will when we will when the pharmacogenetics or personalized medicine, like the future AI development will reach the point that we will know almost like everything in our bodies, right? And we will be able to create even more developed population PK models. It may become a future, but so far we are very far from that. We are just using these models to facilitate the drug and help solving those questions which unfortunately are impossible to do in the conventional clinical development programs.
Okay, so the question about patient safety, it will not be, I mean, this is a separate process. So you still have to hire, to recruit subjects to perform clinical trials just for safety, for this. I mean, this is like an addition, not replacing, yeah? So you are still.
So far it is addition, but we have to account that, yes, when we're doing the clinical trials, as you said, we of course catching the most dangerous and most common safety concerns, enroll them in the databases, right, and make up all our labels. But still, it's impossible to find all of them, right? We have so many big gaps in the real life safety monitoring, right? So sometimes we are so much surprised, how come we couldn't identify these problems in the clinical? For that purpose, we may use these models, and model-based drug development will significantly help.
Thank you so much. Questions from audience? Do we have any? Okay. Thank you very much, Tigran. Stay with us. We have interesting talks.
Of course. Thank you also. And again, for me, it was very, I was very proud to participate here, and it was a very nice experience to be here. Thanks a lot.
Thank you. Thank you. Okay, let's move forward. And I will present the next speaker. Okay, Dr. Monica Tarapuez, member of Advisory Board, ISOP Ecuador. Topic of her today's presentation is importance of pharmacovigilance in emergency authorization of drugs for the treatment of COVID-19. Dr. Monica Tarapuez obtained her medical degree in Ecuador and pursued postgraduate studies in Spain. She has a Master of Science in Pharmacology and a PhD in Clinical Pharmacology at Universitat Autònoma de Barcelona. For 4 years, her pharmacovigilance training was at the Regional Pharmacovigilance Center at Catalonia, Spain. She has been a member of Uppsala Monitoring Center Signal Review Panel since 2016, and since January 2020, she has worked for UMC as a consultant for research and education section. Also, she has experience as a lecturer in pharmacology in medical school, a consultant for a Pan-American Health Organization, and currently is part of the advisory board of the International Society of Pharmacovigilance. Thanks for being with us. Thanks for agreeing to speak with us. I'm giving floor to you to start your talk. Thank you.
Hello, everyone. Many thanks for the invitation. I would like first to thank the organizer for inviting me. It's a real pleasure to be here this afternoon for you. I am going to share my screen. Please let me know if you can see my screen. Can you see?
Yes, yes.
Okay, yeah, perfect. Okay, so I divided my presentation into 3 parts. Firstly, I will discuss the difficulties in discovery of COVID drugs, COVID-19 treatments. Secondly, I'll give you a brief overview of the emergency use authorizations. And finally, I will talk about the pharmacovigilance methods in this context. Okay, so just to keep in the context, the COVID-19 pandemic forced the world to accelerate the vaccine and drug development and the evaluation at unparalleled pace. Nowadays, we know the importance of the immune dysregulation caused by COVID-19. So the pathophysiology, we can identify, for example, triggers, like you can see here in this figure, the mediators and the effectors. And nowadays, the drug developed these are the targets of the drug developed. But we need to have in mind that the pandemic also used the repurposed drugs, old drugs with other indications, and that can have a place in these phases. But nowadays, 2 years after the pandemic was declared, we know better the COVID-19 virus. We made a lot of efforts in different parts of the world to know better how the virus attack the human body. But at the beginning of the pandemic, we don't know that kind of information. Nowadays, 2 days later, we know more. But at the beginning, it was too difficult to know what is the virus and how to fight against the virus. So in different parts of the world, trying with the intention of fight against the virus, there were some wrong decisions regarding the treatment. But here you can see, for example, the case of hydroxychloroquine. And I take the information from the FDA because they have the archive of their decisions. And for example, hydroxychloroquine was obtained the emergency use of authorization in certain patients with COVID-19. But lately, all the world was shocking with the news about the potential risk. Sorry.
Please mute your microphones, please.
Okay, because many things, because the cardiovascular risk that was well known for hydroxychloroquine, but in the COVID patients, it seems that this risk was higher. And actually, that risk was potentially more deadly with the combination of azithromycin. For that reason, we start to see in different regulatory agencies some communications trying to mitigate this risk. However, at the final point, only two months or 2 months and a half of that authorization, emergency use authorization of hydroxychloroquine, in June, the FDA revoked that authorization. And since that point, the hydroxychloroquine was not longer used in COVID patients. This is an example of the trying to have the better decisions. We make sometimes decisions that we need to review at some point, because in this case of the pandemic, all our decisions need to be reviewed at some point and decide if we need to continue with that decision or we need to stop. But what is the emergency use authorization? This is a regulatory concept. I took this concept from the FDA because it's one of the regulatory agencies more, I would say, some countries rely their decisions in the FDA. For that reason, I take this. But I also want to highlight that every country has the opportunity to have their own concept, rely on their own regulation, because it's not possible to copy all the decisions from other agencies. This, for example, but all the concepts rely on the same principles, and the same principles is facilitate the availability of the use of some drugs or some treatments in the context of a public emergency. So, and that relies in articles, in the legislation and things like that. But I don't want to go in that way. I just to hear, I would like to point the importance of having this kind of concepts in our legislations, but with especially reflection. This authorization, this concept, these definitions are temporal and flexible. What means that? This means that we need to review at some point with the information that we need, that we have. For that reason, it's especially important the monitoring, because as the previous conferences mentioned, the drug development, it's difficult. And in that concept of difficulties, the monitoring is essential. But when we have an emergency use authorization holder, we need to keep more eye in the monitoring, because all the information, even though the limitations of the drug discovery studies, the limitation of the information is a key point here, and we need to be more careful about the monitoring. And here you have also the web page of the emergency use in the FDA web page. And here we have the last drug authorized by emergency use. And I have this slide because it's very important how to communicate with the general public and all the healthcare professionals, how to communicate the decisions. It's very important to have a repository of the information. For example, here you can see in this part, in the blue line, all the information, for example, for healthcare professionals, for patients. So it's very important. This is maybe not the best example. There is other examples, but I would like to follow the structure of my presentation, follow the FDA website. But there is a lot of options to communicate the regulatory decisions. And that is very important for the patients and for the healthcare professionals, because all the healthcare professionals need to have reliable sources of information, because nowadays with the pandemic, you know, the internet, it's with a lot of information, and when a patient needs an information, sometimes there is some places with wrong information, fake news and things like that. So to avoid that, it's good to have a good communication with the public. So here is another print screen, and here you have the other drugs that was obtained the emergency use authorization in 2021. And in the same way, we have all the information for patients. And even, for example, in this case, the FDA release information in other languages. For example, here is in Spanish, but they have in other languages. And that is important also if we have a big number of other languages in our countries. In the case of EMA, for example, the EMA have different initiatives to accelerate the access and the development also of new treatments. So I took this image from the website, and I want to emphasize the transparency of the communications here, and also the signal detection methods that they are developed, and not only with the regulatory agencies, they try to incorporate other research centers to help in these signal detection activities. So and here they are talking about vaccines, but it's almost the same for drugs, as we can see in the following slides. Here is a print screen of the COVID-19 treatments. How many are in the under rolling review? How many are in with the authorization application submitted, and the others that actually authorize it right now. And I want to emphasize the underlying rolling review is a process that they are reviewing all the documentation regarding the drug, but at the same time, they can be endorsed the use of beneficial drugs in, for example, in this case of emergency COVID-19 patients, COVID-19 treatments. And they don't have, EMA does not have the title of emergency authorization, use authorization. They use the endorsement. And that is the law from Europe. And that only emphasize that each country, each region needs their own regulation to make these drugs accessible for all people, or in other contexts, control the use of drugs, because the pandemic also demonstrate several cases of label use. And that is important to have in mind, because a lot of old drugs needs to use in COVID-19 patients, because we don't have any option at the beginning, especially. For example, at the beginning, we don't, the investigation about the virus was too little, so we need to start with the old drugs. Nowadays, 2 days later, we have other options. We have more drug developed in this topic, but at the same time, the world asking to continue the activities. 2 days later, we have still, we have some people, until today, we have people in the hospitals, but we need also to continue with the work. So we need to keep the balance. And one part of that balance is access to new medicines or access to reliable medicines for COVID treatment. As the previous presentation, I think Tigran explained very, very well the main limitations with the drug developed. And as you know, the clinical trials have artificial conditions in the patients. The population is selected. It's very difficult to detect ADRs, rare ADRs, because of the limitation in the small numbers, in the small patients, and also the highly select patients. No pregnancy woman, no elderly, in some cases, no children. So we have several limitations. But on the other hand, we have an excellent monitoring of the patient, and that is not so possible in clinical practice sometimes. For that reason, we need to keep the post-marketing surveillance, and one of the methods is the pharmacovigilance, the spontaneous reported, as we will discuss in the future slides. But one of the challenges right now with the drugs is having access, first of all, and monitoring. In this diagram, in this graphic, I want to show you how many fail or maybe are inactive investigations right now. Because if we have emergency use in US or outside US, that is the result of other investigations maybe that fail. Because at the beginning, we started, for example, with several vaccine candidates, and right now we have a few of them approved or in the final phases, because the investigation needs a lot of steps that Tigran explained, even though it's medicines or drugs. In this context, I would like to back to the pharmacovigilance, because pharmacovigilance is design and activities related with 4 steps: the detection, the assessment, the understanding, and at the final point, the prevention. So the pharmacovigilance is an activity to contribute with the patient safety, and that is a public health activity. The pharmacovigilance include several definitions. We know the definitions, we are more familiar with the adverse reaction, but also include the interactions, the new effects, the new adverse events of the medicinal products. Sometimes it's not the best method, but it can be used to investigate the lack of efficacy, sometimes the misuse, and sometimes also always the abuse. With the pharmacovigilance, the key point is have a better knowledge to make better decisions. And for example, the misuse, as I mentioned before, the pandemic have a lot of cases of label use. And that needs to be regulated, needs to be carefully monitoring. In that case, the pharmacovigilance could be a method to use, but not only the spontaneous report, as we will see. I want to go to the new drugs, new antiviral drugs for COVID-19 treatment. Nowadays, we have, or maybe the world is discussing about the new antivirals, because we have 2 new antivirals more target to the COVID-19, to the virus. And for example, the molnupiravir made by Merck is a competitive nucleoside analog in viral RNA-dependent RNA polymerase. And these drugs cause multiple nonsense mutations. That is the main point of molnupiravir. But at the same time, we have another new antiviral, and that was made for Pfizer. And this is an old figure, and you can see here the code, but right now we have the name for this drug, and it's a combination with ritonavir, and this is another antiviral with a lot of action, or the investigation is made against the COVID-19. However, as you can see here in the BMJ in June, I think, in October of the last year, some of the experts discussed the limitation in the information available about the research of these drugs. And even though we have some limitations, last week, the molnupiravir was included in the WHO guidelines. And now we have more information compared with October of last year, but still there is some concerns about these new antivirals. And for example, the molnupiravir is the first oral antiviral drug that WHO includes in their guidelines. This is a new medicines, and WHO mentioned there is little safety data. For that reason, WHO recommends the active monitoring for drug safety, along other strategies they mentioned in their press release. In the same day, it was released by the BMJ, another editorial making a reflection about the differences in the information about the efficacy and also in the safety, because we have several concerns about these drugs. Not only molnupiravir, the Pfizer drug, it's also have a lot of safety concerns. If we are going to use in our countries this kind of drugs, we need to think what is the best option, what is the best strategy to follow the patient, to monitoring the patient. And for example, here is the authorization in the FDA about the Paxlovid, for example, the Pfizer drug. And they emphasize that it's not used, it's not authorized for patients that require hospitalization due to severe conditions, and is not authorized to pre-exposure or post-exposure. That is another point. And no longer use, no authorized for longer use than 5 consecutive days. This is very important because we have all the data, all the investigation, all the research have only 5 days of treatment. For molnupiravir, we have that is not authorized for people less than 18 years old, and they have other considerations regarding the assessment of the benefit, because we have more safety concerns about these drugs. One of the main concerns is regarding the mutagenesis that this drug can cause. And also one concern of both drugs, it's about the variants of the virus. For that reason, they explain this is not the first option for patients. The patients need to be carefully selected. If they are appropriate, if they are a higher risk, they need this drug. And also with other options are not available. Some people mention the patients need to be carefully assessed if they are good candidates for these drugs. In this context, the pharmacovigilance methods need to be assessed. For example, we have the spontaneous reports. We have 3 methods in general for pharmacovigilance. And we have the first one is the spontaneous reporting. So we have the yellow card. However, the yellow card maybe can help in some stages of the post-marketing surveillance, but not in all cases. For example, it's not the best option for long-term surveillance. For example, pharmacovigilance, as you know, have a lot of limitations, but at the same time, it's good to think how looks our yellow card. For example, here you have a print screen of the yellow card from the UK, and they have developed a lot of ways to report. A lot of, for example, they use some apps to send the yellow card, and they adapt also. Some countries adapt the yellow card for patients. It's not the same information, the same questions compared with the yellow card for healthcare professionals. So we need to think how to improve our ways of spontaneous reporting. If we use the yellow card, maybe it's good to think in new technologies like the apps in the mobile phones, or maybe a website when in which the patient can have a register, or maybe the healthcare profession have a registry, and they don't need to put every time the same reported identification data. So we need to think more about how to improve our spontaneous reporting methods in our different settings. In this context, for some of you, maybe it's well known the BG Access. The BG Access is the open source of the Uppsala Monitoring Center to visualize the reports that we have in BG Base. This is an open source. However, they only show numbers without causality assessment. And that is very, very good to have in mind. I want to emphasize this part because remember, the spontaneous report and the pharmacovigilance start with the suspicion of an ADR. We are not totally sure, and that is the purpose of pharmacovigilance. It started with the suspicion. So here you can see all the numbers of suspicion, not the causality assessment, not the 100% of causality. Other resource that we have, and if you want to check, is the FDA. For example, the FDA have a public dashboard about the COVID emergency use authorization drugs. And you can select, for example, I select the molnupiravir, and you can see all the numbers. But remember, this is just numbers. And what is the objective of having this? Communicate, try to communicate in the best way of the transparency, because right now we have information and at some point the information it's not released in some countries to the public, and that makes the process of pharmacovigilance, the process of surveillance, that makes not that process is not reliable for some people if they don't know the results. If we don't communicate in a good way the results that we have, the information that we are gathered, the people maybe don't trust in the process and they don't report. They are under-reporting. It's a big issue in pharmacovigilance. So giving feedback to the reporters or the community, it's good to have. Maybe this way of the FDA, for example, it's too complicated to understand for some people, for the public in general, but it's at a starting point for the healthcare professionals. So we need to keep the balance. This is maybe is not the best example, but we have some examples from other agencies, and maybe we need to think in our country how we communicate the information that we ask, because we are asking to keep monitoring in the patient, but how the healthcare professional knows what happened with that information. So this is some steps to communicate. And we have other two methods. These methods are related with the target reporting and active pharmacovigilance. And those methods engage more the healthcare professionals. For example, the target reporting is a variant of spontaneous reporting, and that it focuses on capture ADRs in a well-defined group of patients and on treatment. The healthcare professionals during the consult, during the visit of the patient, they fill in some formularies, the application forms, like an application forms, they ask for some information about the safety. And that makes the healthcare professional that needs, the healthcare professionals needs training in that activity, and they have more time maybe during the consult. because this is regarding the treatment, regarding the new drug that we need more information. And in some settings that it's sometimes difficult, we need to think how to apply, how to adapt in my concept these strategies. Another is the active pharmacovigilance, and the active pharmacovigilance is one of the most advanced methods, and sometimes they use the electronic medical records systems, and that needs the support from other institutes of research. For example, if our hospital don't have that facility, but also needs some people trained in not only the healthcare professionals. They need more people like researchers, specialists in use of big data sets about electronic medical records, and also this kind of method sometimes need in hospital the authorization by the board, by the safety board, the commission. But these methods are not new for COVID-19 treatments. They were being used for the public health programs in several countries, especially in countries that we have, for example, tuberculosis. And it's well known that in India, there is a very strong capacity regarding the monitoring of this kind of drugs like tuberculosis. In Latin America, we have also some experience in this, like a public health program. So in these programs, we have the experience about monitoring with different pharmacovigilance methods comparing with only the spontaneous reporting. And I want to emphasize also the concept, the new concept of real world monitoring, because this is a new concept, especially used in Europe, and they have the project about international cohort monitoring, and that is that seems very expensive, but I think since my point of view, this is more a political decision, because if we have, I think all the countries have the electronic medical records, maybe not a national level, but in some hospitals. And it is that the cohort studies start with that point. So the political decision is needed to have more access to that information, because sometimes the problem, or maybe the highest limitation, is the access to the information. But if we have that resources in our countries, maybe it's a good idea to think how to access to that information and how to make this monitoring more easy, more accessible for the researchers. This can be applied for the vaccines or for drugs. For example, right now we have the introduction of these new antivirals. We need to think how to monitor at long term, because one of the main safety issues in vaccines or in new antivirals is the long-term safety. So with cohorts, if we have these cohorts event monitoring, that is the official name in pharmacovigilance, we can have more information in the long-term period. So we need to think more in this kind of activities. Well, to finalize my presentation, I want to resume some key message. One of them is, in the world, there is a real need for new treatments. We need this access, also investigation, research, but also the access of these new treatments. And also think the monitoring and the surveillance in the short and the long term should be done in these drugs, especially if these drugs start with the these special authorizations. There are several methods of pharmacovigilance for monitoring the adverse drug reactions of new drugs, and at the same time, we can evaluate their effectiveness in the clinical practice. So we have some methods. We need to think how to adapt in the best way possible to our settings. Finally, I want to invite you to the annual meeting of ISOP. This will be on Verona in Italy. So the abstract submission now is open to our meeting. So I would like to have that invitation in this time. Many thanks. And this is the social media channels for the ISOP. I like you invite to follow us. Many thanks.
Thank you very much. Thank you, Monica. Just wondering what time is it your city now? It's morning?
For me, it's, yes, it's 8 a.m.
Okay, great. Thank you very much for joining us in this early morning and for I think this is really very impressive, conclusive presentation of today's webinar with an important point to raise out. You raised out one small question regarding endorsement from the EMA of COVID treatment, COVID drugs. So in my understanding, emergency use authorization is the FDA term and conditional approval is more EMA term, but I'm not sure about endorsed. So endorsed, it's like it doesn't approve in European Union region, but they are just giving other member states the possibility to be approved in their national level. Yeah.
Yes, because they have, as you mentioned, they have the option of conditional approval, but the endorsement, it's previous that. And with that level of endorsement, they give the opportunity to different countries to decide also, because you know the EMA includes more than 20 regulatory agencies, so it's difficult to, and there is some pressures from different countries to have the access to some medicines. For that reason, the endorsement is a first step previous to the conditional approval, for example, because they have that view. For example, remdesivir at the first point, and nowadays have the conditional approval, and the conditional approval is another regulatory measure that EMA have.
Yeah, just to gain time because of this pandemic situation, and to give member states to go forward with approval if they
exactly.
Thank you so much. Please, questions from audience. Thank you again, Monica. It was really very impressive and interesting. Thanks for joining us and for speaking for us.
Thank you so much.
Thank you. So I am giving the floor to Chinmaya.
Thank you, ma'am.
Dr. Chinmaya.
Thank you, ma'am. So I would like to introduce Dr. Raghunath Jinosian. So she is the director of analytical laboratory branch at SCDMTE Armenia. She is pharmacist with PhD and MBA in healthcare management degree. Over 20 years of work experience at local regulatory and international QA and QC laboratories under GXP regulations. As a QC lab director, she was responsible for drug QC during registration, import, export, and expertise of unknown samples from different government agencies. And direct work responsibilities as QA manager at US-based Spanish CRO include implementation of integrated QMS and ISMS or PIMS certification, and total quality management, including regulatory and vendor audits. She's having more than 8 years of multilingual teaching and supervision experience at ISCC, NASRA, and YSU. So welcome you, Dr. Raghunath Jinosian. So we both, I think we can have a panel discussion. So the topic that is here for today, we can discuss it, but let her talk first.
Thank you very much. Thank you, everybody. And first of all, I would like to thank you again to organizers for such an interesting and useful presentations and the knowledge that today we had a chance to share with all of you. And as from my experience, and I would welcome everybody to go discussions and share your questions or concerns or maybe even suggestions. So for future, we have a chance to work on that and cooperate together and have better improvements, especially for such difficult times, including the COVID situation that we experienced last 2 years. And plus like the international communication and globalization, because even from Armenia point, currently local regulations are slowly changing, and we are working through Eurasian Union, and now we need to adopt all those regulations, and which are of course based on European regulations, but still like we face some challenges. So you are welcome, and maybe you can continue too, Doctor.
Yeah.
Sure.
So first of all, let's start with a first topic, that what could be the strategies to help the patients who suffers from a ADR or adverse drug reaction. What could be the strategies to help the patients to get protected from the ADRs? Because we all are working for pharmacovigilance field, right? So we are doing a lot of job and lots of work we are doing, but still one question comes, like how can we protect a single patient, particular 1% or 2% or 3%? How can we protect them with our knowledge? So that is the first question. Anyone in the audience, they can join us for discussion. This floor is open for all.
Please, you have such a great opportunity to speak out. Maybe Dr. Chinmay, you can start from what is coming on in India, and you can share your experience in this.
Yeah, so we thought that suppose a patient suffers from an ADR, right? He discuss this topic with his colleagues or friends, but still he doesn't know how to report it to the regulatory body, and still that's why all the training processes are going on, and we want patients to participate in the PV system, right? So once they start doing that, we will get the data. That is one part, but how means my point is, if we can stop the re-administration of the drugs that is suspected drugs that may cause adverse events to the patient, then by that way I think we can stop the ADR, because the patient knows what drug he should not take, right? Suppose I am allergic to penicillin injection, so I know that during my childhood I have taken some time and I got suffered from rashes and also I know that, but doctors, physicians, they don't know. My family members or my friend circles, they don't know. So it is better to let it document it in as we are having now digital platform, and we can create some kind of our profile, and we can keep it with ourselves. No need to disclose it to others, but we can keep it with ourselves, so that this message will be communicated to the physician during our next visit or whenever we go to the physician. So by that way the physician will get to know that yes, the patient is suspected to this drug, and he won't rewrite the drug, right? So that way we can stop in a small scale, we can start this movement, and later on I think it will go on, right? Now coming to the second topic, is pharmacovigilance audit is mandatory for all the pharmaceutical companies or generic companies or regulatory whatever it may be? So is it mandatory for all the companies or not, or only for the PV, or only for the companies that are having the PV office, means PV system in place? So that is another question what we have to address. So anyone can join. It is an open discussion and you all are free to discuss. No need to be worried about it.
Thank you. And as we today also heard that now the main approaches are being changed, and more patients will be also involved in this procedure, which is very important. Because as you mentioned, like for medical personnel, sometimes it's like, especially when what we learned during pandemic, that medical people were busy because of overwhelmed. Right. And yeah, in this case, like patients' acknowledgement and their support is very, very important. At the same way as the PV contact person, and meantime connections with the regulatory affairs, because like only working together we can achieve our goals, and like to keep all this changing information stay updated, and medical staff is informed properly about the information maybe within narrow professionals is already there, but to spread and to make sure that patient safety and patient-centric approach is all over during the whole care is the most important thing. So thank you, everyone. And I don't know, Liana, if there are more questions or no. Yeah,
only one,
really
last one.
For this chance. Yeah, thank you.
Our last one is that, see, we have WHO EMC scale for causality assessment. We have other scales also for causality assessment, but still, ultimately, all the reports are going to WHO, right?
Yeah.
From different regions, different countries, it is going to the WHO. So why not we follow only WHO scale, and why we are going for other scales in some parts of the country, some other companies? That should be addressed, right? We have to streamline these things. Otherwise, someone will say that yes, this kind of causality, and another will say this kind of causality. So it will create another conflict. So it should be streamlined, because ultimately the report is going to be reached to the WHO. So why not, though they are using WHO EMC scale, so why not all the persons are adopting that scale? That is a one question, single question, not a big one. So anyone can say, anyone can comment on it.
I totally agree, Dr. Chinmaya, because actually sometimes pharma companies use just binary assessment, like related and not related. But the best approach is to be, in many fields within pharmacovigilance, everything is standardized. But regarding causality assessment, which is the most of importance, it's not so much standardized. I agree with you. Yeah, and so one pharma company uses binary, another WHO scale, the third one Narenger scale, etc., etc. But yes, this is the final result of the overall pharmacovigilance activities, just causality assessment, and I agree that it should be standard. And regarding communication, communication with healthcare professionals and patients, we know that case reporting is the most important thing, because we are gathering data from real world. So as Liana from Prague mentioned, now orientation is a bit different. They involve patients, they involve HCPs in their decision making, yeah? And this is really very, very, very good to be patient oriented and HCP oriented in their decision, and to involve them in the processes.
Yeah, Dr. Liana wants to say something. Dr. Liana wants to say something.
Sure.
No, thanks a lot. I really enjoyed following all the presentations. Very interesting topics. Again, thank you to all organizers. I just have two short comments. One with regard how we can improve communication and ensure safety. And well, it was an interesting example you have just mentioned with penicillin allergy. That, well, it's just a simple example. I understand that, and we have many more. And one of the solutions you have suggested is why such adverse drug reactions, or actually history, allergic history, could be documented in patient records, electronic health records. Of course, that would be the best way to ensure that, well, if the patient goes to another specialist, then another specialist can retrieve the medical history. That would be an ideal world, but we know that in many countries, electronic health records are not widely used. That's one thing.
Right.
But even if I talk about the well, situation in the Netherlands where everything is digitalized, still the medical history of patients may not be shared equally between different hospitals. So if, let's say, if I go to another hospital where I don't go usually, my well, GP records may not be shared and readily available there. So it remains very important that patients also proactively communicate to healthcare providers about medicines they are using. Of course, it's primary responsibility is not with patient, it is with the healthcare provider who needs to check any medication that the patient is using. But just an example, what we do in Europe for products that have important risks that need to be communicated to other healthcare providers is that we implement so-called patient alert card. It's a small card of credit card format, which patient can easily carry all the times. And in case of emergency, it can be easily found, and then another healthcare provider can see, oh, this patient is using this drug. And one example is a number of different immunosuppressants which are being used for treatment of MS. And for instance, sometimes there is a contraindication to use live vaccines in such patients, and then this information is mentioned on the cards. So another healthcare provider will be made aware via information on that card. And one another comment with regard to the criteria to assess causality. I have to say that I am personally a big fan of WHO UMC causality assessment criteria, because it's just, well, it allows us to make a clear conclusion, even when we have very limited information in spontaneous report narrative. Well, Naranjo, for instance, Naranjo criteria may require really a lot of clinical data, laboratory values, which very often we do not have. Yeah, I think that it would be really good if there would be more standardized approach used by many different companies. And well, I think WUMC may really have, well, a very good chance to be used on a wider scale. Well, unless we are dealing with very specific adverse drug reactions, such as, well, scarce serious cutaneous reactions, such as DRESS, etc. For this type of reactions, at least in Europe, we have developed a different causality assessment algorithm called ALDEN, and we need for this a bit more clinical information. But I think for such serious adverse drug reactions like DRESS or Steven-Johnson, we really want to be sure what is the causative agent, especially if the patient is using different products. And if we use WHO criteria, we can very easily conclude that the association is possible. I think for this type of reactions, we need to be a little bit more certain before we are going really to update the product information and allocate that side effect to a specific drug. Thank you very much.
Thank you, Doctor. So,
can I ask a question, Chinmay?
Yeah, sure.
Yeah. So I have a question, and this is because this is an open discussion. I would be happy if everybody could contribute to this discussion. So generally, pharmacovigilance is a retrospective science where it is about identifying, detecting, and managing the adverse effects. For cases where there are delayed adverse drug reactions to any medication or vaccines, what is the strategy that we apply to identify those as adverse drug reactions? Because a particular condition, an unusual symptom could arise months or probably years after a drug has been administered. So how is it really diagnosed, whether this was due to the vaccine or the medication that was administered? Or I'll give you an example. I have a friend who, and I recently shared this post, I have a friend who who's in his 60s, and he has a history of a congestive heart failure. And he very recently in the last year developed photodermatitis, a condition where he was experiencing severe allergy to severe skin allergic symptoms to exposure to sun. And he was diagnosed by the dermatologist as a case of photodermatitis. He was started on treatment on that. His condition improved a little, but then after a point of time, there was no resolution. He lost his sleep, his mental health suffered. And so he was just talking to me about that. And as we discussed, I asked him, why would such a condition suddenly develop after so many years? A photodermatitis, an allergic reaction to sun. And the first question that I asked him was, what is the, if there was any change of medication in the past year? And so he first, the answer was negative, and then he thought about it and he realized that he had taken valsartan, sacubitril and valsartan, Zio, very recent, in the last year. And after that, probably the symptoms had started, and these symptoms had been continuing. So I did a literature search, and the adverse drug reaction of photosensitivity glared to me with valsartan. And I informed him about it, and he discussed it with his cardiologist, and there looked to be an association, and eventually his that drug, the new drug, was discontinued and was switched back to his old medication for hypertension, and his symptoms actually resolved. So here, because I had that lens of pharmacovigilance, because I'm a pharmacovigilance, I have a lot of experience in pharmacovigilance, I could directly relate it to a new drug that was introduced. But that drug was also going on for about 1 year, but that was not kind of identified by the dermatologist when they were diagnosing the condition. So, as we were discussing here about the effective communication to the healthcare professionals, I feel, that integration of pharmacovigilance expertise in clinical diagnosis and in clinical practice is also as important, so that, any new conditions that arise, they can be related to possible side effects of a drug that has been continuing for a long time. So what do you think about it? Do you think it's, do you have similar experiences, or do you think being in the field of pharmacovigilance gives you that lens, which probably a healthcare professional as a clinical clinician might not have at all times? And I think the pharmacologists can also contribute there, because they have been working on the pharmacology of adverse drug reactions and have much more experience.
Anyone wish to answer?
In understanding of the adverse effects.
Anyone from the audience wish to answer? All right. So as our previous speaker spoke about the curve, that when we take a drug, it goes on increasing in our blood circulation, then it comes down, and so that is the area under curve, and something he has explained just before a few minutes. So when we are taking a drug, it is not going completely cleared from our body. Still, a small fraction of it remains in our body. It is the theory, whatever it is there in our book, whatever we have studied. So it is the theory that a little bit part of the drug, it remains in our body, right? So we don't know, it may cause the adverse event or it may not cause the adverse event, but the patient is the main person who can suspect the drug, that yes, I have taken this drug in this time, and then I got this kind of adverse event. It may be a mild to moderate, but he would have got some kind of indications in that case. But it could not be a severe one. And for a long-term study, what you are telling that it is a long-term adverse event, means it comes after long-term administration or something like that. So it may be due to your it may be due to the accumulation of the drug later on. And
no, I understand that. What I'm asking is, do you think, that
yeah, it should
there
be
has
monitored.
to be,
Yes, it should be
that the
monitored.
monitoring has to happen, and probably that kind of communication and understanding in clinicians needs to come, that they have to, the AE review of medications should happen before they prescribe a new condition. Yeah,
let me complete it. So as we go and read the guidelines for medications error, how to protect a patient from medication error, in that it has been clearly written that whenever you are going to take a drug, you just ask your patient, ask your physician that why you are going to take this drug, and maintain a record of it, means for which disease you which drug you are taking. So that is the main thing that has been regulated already, but no one is following that, right? So you have to maintain a diary that yes, I am taking this drug on this date and for this disease, for this indication, right? We have to maintain that record, and then we can whenever something goes wrong, we can come back and we can see that whatever drug we have taken. So that is the thing that should be done by the patient itself. No one will do for him, right? In this world. So that is the thing, is for a long-term study we can do that, but the system has to be changed in this way. And it is there in the guidelines. You can check it out in the medication error, some WHO guideline, it is there. Yeah.
Okay, Dr. Shirma, we have questions, I guess, from audience. Can we give a floor to Grigor technician to ask a question?
Yes.
Please, while listening, you, Grigor.
Hello, hi everyone. Can you hear me?
Yes.
Yes.
I'm very pleased to be here. Thanks for organizing this. I'm from Armenia, and I'm actually new to the field and have maybe just a little bit over a year in the experience in the pharmacovigilance. So obviously, I had a great interest in participating, and thanks again for gathering so many people from actually so many like various domains and brought them together. This was a really nice experience. Throughout the day, I actually gathered some questions, and I thought maybe it's actually more appropriate to ask them all at once. Let's see how we can manage this. So the first two will be like more current status questions, and then the other two are more like future oriented. So
I'm sorry to interrupt you. Grigor, are you addressing your question to particular, I mean, to speakers or to whom? Maybe we can clarify this, then they will.
Actually, I would like anybody who is willing to step in, just go ahead and step in. I don't have any specific preferences. I'd like some questions about AI to be like maybe responded by Nicole Baker if she's still on the call, but anybody can step in, no problem. So yeah, the question about COVID pandemic, like we all know that well, prior to the COVID pandemic and after and during the pandemic, the approaches in pre-marketing and post-marketing were like they drastically changed, and some things were really accelerated. There are some new methods coming up, and I had a natural question of whether or not all these new methods sort of were in place and were going to be launched in the industry, like by regulatory authorities or by many stakeholders, or all these new approaches are sort of like like retrospective and just maybe sitting there and waiting when the pandemic will strike and only then be implemented. So I just want to understand like what was the balance there.
Okay, saying new approaches, you mean handling pandemic and regulatory some stuff regarding pandemic?
Yeah, more specifically, let's say, you know, that vaccine development has been changed and maybe accelerated, and it's not like we don't have the same pace as we had, let's say, 5 years ago, or the reporting of adverse events after immunization. So this reporting fashion has also been changed. So I'm just curious whether such implementations were already in place, like they were in the agenda of stakeholders, or this is just like some spontaneous approach that only got implemented after the pandemic.
Yeah, okay, understood. So I think it would be better if Liana is still here. She's from regulatory. She can address this question maybe better.
Yeah, of course. Yeah, it's a very relevant question. And now that the pandemic seems to a little bit, well, there is a hope that it is coming to an end now. Many different countries are now busy with looking back and thinking about lessons learned to implement in the future. But of course, it's not something spontaneous. I know, and I'm quite sure that in any country in the world, there is some sort of pandemic preparedness plan, especially after we have had one pandemic, which was, of course, much less severe in 2009, 2010 with H1N1 influenza virus. And so it is not very different for regulatory agencies which are responsible for regulation of medicinal products. Of course, there were plans in place how to deal with well, assessment of marketing authorization for vaccines or any other products, because there could be emergency situation, not only with infectious diseases, but it could be also different, and also how to deal with an increased volume of spontaneous reports that is coming in. But I think you can never be prepared fully in advance, because every type of emergency is different. And now when we look back, we see that a lot could have been done beforehand. And one of the things I have already mentioned is that at least for processing of spontaneous report, it would have been great that before any vaccine was for COVID was authorized, that would be a requirement in all systems. So not only for national competent authorities, but also for pharmaceutical companies to have systems in place to well, make obligatory requirement in the fields to record if it was dose 1 or dose 2, because that would really greatly help all stakeholders who are busy with processing of the reports. So it's both. Yes, we were prepared, and yes, there are lessons learned. Yeah, thank you.
Yeah, that's great to know that there is preparation in place. Thanks for the response. Thank you. So maybe going forward to my next question, I actually couldn't resist asking this because this is like from like current realms, and the question is about consequences of pharmacovigilance activities in the face of like modern warfare and like heavy sanctions and virtually isolation of a certain population. So, well, currently it's the example of like the war in Ukraine and like heavy sanctions put on Russia. But I mean, I don't want to get like political by any chance, but just the mere fact of such thing happening. I was really worried about like what would be the consequence for a vast majority of like for a vast like population in terms of, say, well, we know that sanctions are affecting well, they are like directed towards the government, but fundamentally and ultimately this affects the population itself. And so by, for example, restricting the access to digital media or some other means, that these populations, and we're talking about millions of people, are virtually isolated. And I was thinking, what are the consequences on in the pharmacovigilance domain, let's say, or in the clinical research? I've heard that many clinical trials have been paused in Russia. And so I was curious, how is such, is there a mechanism to tackle such situations? And maybe if I come up with a simple example, like if someone was conventionally reporting some ADRs via Instagram, and now there is no Instagram, like what are the other means maybe, and how can the WHO or other regulatories reach these people and maybe carry on the conventional pharmacovigilance activities in these tough realms?
Okay, I will try to address as much as I can. So actually, when I was just trying to prepare for this webinar, I decided to be apolitic because the topic is very painful indeed, and to speak even difficult. But you raised this from the pharmacovigilance point of view. In our everyday, we see the challenges in pharmacovigilance activities. For example, me and my team were doing literature monitoring activities for CIS region, and including Ukraine, and we advised not to open their websites and not to do anything with Russian and Ukraine sites from the point of view of monitoring of literature. This is one small part of pharmacovigilance. Globally, if we will see the whole picture, of course, it's influencing badly as other spheres, so including pharmacovigilance, and you can understand the importance of this field. It's about safety of drugs, and the sanctions and this war, it's of course influencing. Coming back to your question regarding WHO, some actions on this, I don't think that they for this moment they managed to do something with this. Maybe in near future they can think, but not now, because it was really sudden happening, all these things. So hope it will be, and praying for peace and finishing all this stuff as soon as possible. It's really difficult to speak about.
Yeah, I totally agree. I mean, if I could maybe derive a message out of this, is that so a state that's currently or in future, if the state is getting isolated, it's sort of mainly the responsibility of that government to to close up and not to to basically shut down all these cooperations. Is that the case? It's I guess it's not much other regulators.
You cannot to be isolated in this, because it's broad actions you have to. So you cannot just think about your regulations and that's all, because I don't know. Let's see. But saying again, this happened very suddenly, and that's why I don't think there are any pharmacovigilance actions in place, proactive, to avoid any challenge in this field. So let's pray for peace.
Yeah. All right. Yeah, let's see. All right. Okay, then maybe moving on to some more positive and prospective aspects of pharmacovigilance. I was actually very thrilled by the speech of Nicole Baker, because I'm also naturally very interested with the AI, machine learning, and all of this new developments that are happening virtually every day. And I was curious how widespread is this AI modeling in the pharmacovigilance industry? Was the work that Nicole presented a pioneer work, that like a single example, or it's because I just don't know the market very well. So I was thinking if like how widespread is this, and whether so that's one question. And the second question is whether or not the AI modeling is an approach of currently is an approach of private sectors, or some like FDA or EMA are also into this, and maybe they are also developing some AI tools to then share with the industry, and then maybe everybody can subscribe to that and implement it at their own company. Like what's the situation of the market, basically?
I'm not sure whether Nicole is here.
She's not there.
Nicole.
She's not here.
No, that's fine. I mean, if anybody can provide any insights, I would be really
regulatory part, I will advise Liana to accomplish. Regarding overall artificial intelligence and pharmacovigilance, it's coming more and more. That this artificial intelligence and pharmacovigilance is like going together, because you know when you will be involved more in pharmacovigilance, you will see that so many efforts, human efforts, are required for example, literature monitoring activities, for reviewing journals, or just human efforts. So that's why, yes, this is a tendency, a fast tendency to involve artificial intelligence as much as possible into pharmacovigilance. This is, in my knowledge, private sector. I mean, some CROs are suggesting AI services to pharmacovigilance companies, to other CROs, to pharmaceutical companies, because they develop this and they are suggesting to facilitate the work of pharmacovigilance departments or CROs, because you know regulatory is very strict, and you have to do so many stuff, and using artificial intelligence, it's really facilitate, and you can do the same kind of activity, not with 20 people, but with 2 people, and have the same results. So this is going fast to be involved in pharmacovigilance, and this is private sector, I would say. Regarding regulatory, I will pass the floor to Liana again to accomplish this.
Sure.
Yeah, thanks very much, Liana. And I would, of course, probably we don't know what is the exact, widespread, how widespread is the use also by pharmaceutical industry, but I absolutely agree with Liana that it's nowadays almost impossible to imagine a pharmaceutical company, especially company who is dealing with brand leader innovator products without AI tools in place. But with regard to regulatory agencies, I can say that at least in West Europe, as you may know, it's not only in West Europe, in any country, basically in Europe, there is a pharmacovigilance center who is performing activities which are very similar to what the companies are doing with regard to receiving of spontaneous reports. Because in Europe, according to the law, patients and healthcare providers may report suspected adverse events to either the company responsible for the product or the National Pharmacovigilance Center. So at least I can speak for the Netherlands. The reporting culture is very, very active. It's collecting most of the highest number of reports in Europe. So the number of reports has been increased exponentially after introduction of COVID vaccines. And so the Dutch Pharmacovigilance Center had to really come up with new artificial intelligence tools in order to process with the validation and correct coding of all the reports that are coming in. And it was a great success because it was actually one of the few countries in Europe that doesn't have any delayed reporting. There are some countries that still have well, some lags, so quite some number of reports that still need to be processed. But of course, it's on the one hand, it's a costly process, and as you have mentioned already earlier, it's something that you need to anticipate, because you cannot prepare something like this in few days. So yeah, preparation.
Right. I was actually surprised that this modeling. Yeah, I was actually surprised that this modeling takes a year, like for a simple task to. Well, it's not a simple task, but yeah, like it takes a long time, sure.
Yeah, and now currently we have a project ongoing where there is a sharing of expertise and knowledge on use of such artificial intelligence tool between different European countries. So countries that were more successful in implementing such tools could share their expertise, and so it could be taken into consideration by other countries.
Mm-hmm. And all right, yeah, thanks. So do you think, like, if we think like 3 years or 5 years from now, like, are we already close to discussing the question whether or not we are going to retrain / replace some of the stuff that was doing well, really like low echelon, close to mechanical work in terms of let's say ICSR processing or literature review, or they're still far from discussing that.
Let me try to answer. Today, Luca, if you remember, he said very important thing that for doing this artificial intelligence stuff, you have to properly think about how to do all this stuff. So, I mean, you cannot do artificial intelligence without human brain, because if you will, at the beginning, will set up in a wrong way, all your work will be meaningless. So to set up in a right way, you still have to have brain. So, I mean, artificial intelligence is not replacing human brain.
No.
Answering on your question, yes, maybe in the near future we will have less team involved in certain activities. But, for example, regarding signal detection, causality assessment, or still setting up this all stuff, still we will need appropriate expertise to work in the field. It's like both together, artificial intelligence and expertise working together. Artificial just simplifying the process, but still we will need properly trained and qualified staff to work in this field. I'm not sure whether I answered that question.
No, that's fine. That's fine. I mean, it's actually very hard to forecast because things are changing in an accelerated manner. So yeah, you might not know actually what happens. That's natural. And I also agree on the approach. That's one of the, I guess, main like ethical principles regarding all of this field, that it's indeed not replacing a human, but it's going together. It's just helping the like assisting the human brain, or assisting the human capacities in their daily activities, right? Thank you. Thanks again for the responses. And yeah, well, naturally, my last question as will follow. It's about the like the job market and trends in the pharmacovigilance. So as a new guy in the field, I was also interested in like how the like what would be the required skill set for someone that is wanted in the pharmacovigilance domain in the face of this changing, in the face of all of these tendencies. Like, do you think that maybe the recruiters would like to see someone not only with like a strong life sciences background, but also with like AI or data analytical like skills, or well, I can't answer my question. Yeah, basically. So I would like to know what's the maybe are there changes in the required skill set? Because as a new guy, I would like to like work on my, like to make my profile stronger in the field. So I was thinking, in which areas may I improve?
Okay, good question. Are you a doctor, physician?
I'm coming, yeah, I have a medical background, but then I was, my background is also, like, I did a master's afterwards in molecular biology and immunology.
Okay, so nice to hear. Actually, you know, pharmacovigilance is, as I said, it's very, very broad science, broad field. And for some activities, you have to have just life science. I mean, you can do these activities, but for other kind of activities, for example, the same signal detection, it's better to have a medical doctor degree. Why? Because you have to have just medical background and medical kind of judgment. Yeah, it's in our it's within us if we have medical background. So it still depends. Regarding the artificial intelligence kind of activities or background or qualification, you know, the tendency is now CRO, overall CRO has different departments. For example, pharmacovigilance, data managers, SAS programmers, biostatisticians. And here I would emphasize the teamwork, because the same person cannot be, I don't know, data manager, physician, or program developer. It's impossible. You can understand, yeah? But working together and doing stuff together will help. So it depends. But mostly for pharmacovigilance, you have to have pharma degree, a medical doctor degree, or life science, and you will be allocated for kind of activities which are fitting with your background. That's all.
All right. Yeah, thanks. So basically to choose what I like and then to see the specifics of that field. Yeah, gotcha. Thank you very much. And again, thanks for doing this. Hope to see more of this in future. Thank you.
Thank you too. Okay. So Dr. Chinmay, are you there?
Hello. Yes. Yes, ma'am. So
time already, 50 minutes. So it means it was interesting, I guess. So maybe we can conclude. Yeah.
Yeah, we can conclude now. Yeah, we can conclude this session now, ma'am. And you can give a vote of thanks to all the participants. And I would like to thank all the participants also who have joined today, and the speakers from different countries. So you can give the vote of thanks, ma'am.
Okay. Thank you.
Yeah, and one more thing, we have one more upcoming event in April 7 and 8. So it will be in hybrid mode. So I will be sharing the agenda soon. Very soon I will be sharing the agenda with all of them. And after that, we have Global Pharmacovigilance Congress in June 10 and 11. So the events are upcoming one by one. So it will be helpful for all the participants. They can join and they can learn something from PV perspective. So, and thank you so much all the persons who have joined today. And yeah, ma'am, it's up to you.
Okay, thank you. I would like to thank, first of all, Dr. Chinmay, who helped and who actually advised to do this webinar. It was his proposal, actually, and I am very glad that I hope we succeeded, and I hope audience and participants gained, and we all gained knowledge, gained more, and this was really bring more value, and you will use all you learned today during your everyday activities, everyday work. I would like to thank our speakers for invaluable support and input and contribution to this event, for their time, for their efforts, for being with us. Thank you very much. And I would like to thank our audience for being patient with us. We're out of time, but I think this is because it was really interesting. I hope so. I would like to thank our moderator, Dr. Maya Jinma, for helping us with this, and I would like to thank our panelists, Agavni, also Dr. Agavni. Thank you for being with us. So thank you all. I hope, and I'm very glad, I'm happy that we succeeded. This was actually my first experience of doing kind of activities, and thank you very much. Thank you all, and I wish good luck to all. I wish, again, I wanted not to speak about this, but just to say no more and just to pray for peace all over the world, because it's really very painful, and I hope very much soon we will see this peace. Thank you.
Thank you so much, Dr. Liana, for your all support and contribution. And I know what you have done at the back end, what work you have done. Really great. Thank you so much for bringing all the persons together and organizing. Helping me in organizing this event in a successful way. Thank you a lot.
Thank you. Have a nice rest of the day. All the best to all. Bye.
Thank you.
Thank you. Bye-bye.
Thank you.